delay, but it is going to start within next maybe 1 or 2 minutes. We are going to start, sir. Thank you.
A very good morning, everyone. I welcome you all to the fourth session, Perpetuity, Genetics and Society. On this important day, I, Harshita, PhD scholar, feel delighted to begin the session by inviting our Honorable Chairman of the session, Dr. Arun Kasingh, to grace the dais. I would like to extend my warm welcome to the speakers of today's session, Dr. N.K. Singh, Dr. Velu Govindan, and Dr. Arun Kasingh to grace the dais. I would also like to welcome the conveners of the session, Dr. H.K. Dixit and Dr. K.K. Vinod. Then I would like to welcome the reporters of the today's session, Dr. Johann, Dr. Rajkumar Junjare, and Govind Rai Sharma. May I now request our Honorable Director, Dr. A.K. Singh, to welcome the chairman and speakers with a bouquet of flowers. Thank you, sir. May I now request Dr. H.K. Dixit to introduce the chairman.
Very good morning to all esteemed dignitaries, invited speakers, and delegates. I thank organizers for allotting me this pleasant duty of introducing chair of this session. Thank you, sir, for agreeing to chair this session. I welcome you, sir, with brief introduction. Professor Singh was born in 1942 in Ghazipur district of Uttar Pradesh. He obtained his BSc and MSc AG degrees from Government Agriculture College, Kanpur, which was affiliated with Agra University. And then sir did his PhD from Florida University. He served as postdoctoral fellow in University of Florida for 2 years during 1996 and 7, and then joined Government Agriculture College as pool officer in 1968. He served as lecturer at BHU and also served as plant breeder in ACRIP of dryland agriculture during 1972-73. Sir was elevated as reader in March 73 in Department of Genetics and Plant Breeding. Served as professor from 1983 to 2004. During this period, he served as head of the department for 2 full terms and dean faculty of agriculture. Sir taught several courses in genetics and plant breeding and guided 28 PhD students and published 182 research papers and edited a book, Advances in Cytogenetics and Crop Improvement. Received several awards, including Fraternity of Alpha, Zeta, Gamma, and Delta from University of Florida. Served as president of Indian Society of Genetics and Plant Breeding from 1990 to 1994, received ISPRD Recognition Award in 2003. Developed 15 widely adapted varieties of cereals and pulses, and his teaching and research contributions are commendably recognized far and wide. Major research accomplishments include cytological and biochemical differentiation of different Sisal species, cytological analysis of pearl millet, chickpea, pea, and sunflower, development of rust resistance in wheat and downy mildew resistance in pearl millet, genetic investigation on induced mutations and monogenic analysis in oats, induced mutations in wheat, rice, barley, and peas, pesticide-induced mutations in barley, and genetic analysis of yield and yield components in cereals and pulses. Sir, I request you to kindly start the proceeding. Yes, sir, please.
Am I audible? Am I audible?
Yes,
Okay.
sir.
A very good morning to all of you. At the outset, it's my great pleasure to be a part of this very important symposium on Mendel, where we have definitely learned during the last 2 days many very authoritative lectures. I see Dr. B.D. Singh joined online. His lecture was appreciated far and wide. Similarly, yesterday morning, Professor Balram gave a very nice lecture, and I hope the others would follow the suit. I see in the audience Professor R.B. Singh, Padma Bhushan, who has been my teacher at the college, and also Professor A.K. Singh, Director of the Institute, Dr. B.B. Singh, Shiv Kumar, to name a few. I might miss many names. I see Dr. Renu Singh also, wife of my younger brother, Dr. A.K. Singh. Ladies and gentlemen, particularly the students. First, I would like to introduce the speakers for the morning session. Here we have Dr. N.K. Singh, who happens to be my student. Very gratifying and satisfying for me to see students doing so well in different areas wherever they are. Dr. N.K. Singh, he did his BSc AG and MSc AG from Banaras Hindu University during the period 75 to 80. And he won gold medals in both these examinations. Then he moved to University of Adelaide, Australia, where he did his PhD from 81 to 85 during that period. And then he was a postdoctoral fellow, CSIRO, Division of Plant Industry and at the University of Adelaide from 86 to 91. He is National Professor, BP Paul Chair in Genetics and Plant Breeding, National Research Center on Plant Biotechnology, Indian Agricultural Research Institute, New Delhi, from 2010 till date. Earlier, he served as a Scientist C, the Central Food Technological Research Institute, Mysore, 91 to 94, Associate Professor from 94 to 2000 at the G.V. Pant University of Agriculture and Technology in Uttarakhand now, and then Principal Scientist Biotechnology, NRC on Plant Biotechnology, IRI, New Delhi, 2010. He, as I mentioned, that in honors and records, he has many. I mentioned about the gold medals of BHU, then KP Barreley Prize, University of Adelaide, 1983, CSIRO Postdoctoral Award, Australia, 1986, Queen Elizabeth II National Research Fellowship Award, Australia, 1988, DBT National Bioscience Award, 2001-2002, World Technology Award for Rice Genome Work, 2003. As you all know that he was the leader here to sequence the long arm of chromosome 11 in rice, which was completed in 5 finally. The Rafi Awad Kidwai Award, 2007, ICR Narbonne Borlaug Award, 2014, Dr. O.P. Bhasin Memorial Award, 2019. He is Fellow of Indian National Science Academy, National Academy of Sciences, Nasi. Research areas: plant genomics, genetic engineering and molecular breeding in rice, wheat, pigeon pea, and chickpea rhizobium. And I'm also told that he is working on mango also, and he has tried to see the number of genes from the common parents in Amrapali, which is much more than the 2 parents involved in the variety. So with that, I move on to the next speaker, Dr. Velu Govindan, who is from CIMIT. He has been engaged in wheat improvement research for past 15 years or so. During this period, he has pioneered partnership-based impact-oriented research, making significant contribution towards development and release of more than 20 biofortified wheat varieties with enhanced zinc and iron concentration. This is, as you all know, very, very important that if we can biofortify the varieties of different crops, they definitely are going to serve the masses rather than giving it, in an artificial way. Then Govindan is passionately committed to advancing genetic gains in wheat yield potential, improved nutritional quality, and yield stability across diverse environment by combining traditional breeding and cutting-edge genome tools. He has published more than 70 peer-reviewed journal articles and 15 book chapters. He is a member of Crop Science Society of America and Indian Society of Plant Breeders. Govindan has received the Jawaharlal Nehru Award for Outstanding Doctoral Thesis from Indian Council of Agricultural Research, as well as the Young Scientist Award from Society for Plant Research based in India. Now, the third speaker to my left is Dr. Arun Kasingh. Professor Dr. Arun Kasingh is a professor and head of neonatology at AIIMS Jodhpur, and is credited with opening the first department of neonatology, the first institute of neonatology, along with early interventions. Okay. He was the first to conceptualize and implement the idea of SNCU, that is Sick Neonatal Care Unit in this country, which is now a model for the rest of the country. His original work was published in Lancet. He is also the first to integrate a detailed early intervention with newborn in care unit and SNCU for improving cognition, which is also the basis for DEIFC under the Rashtriya Bal Swasth Karyakram. His original work on physiological cord clamping, designing of labor room and its impact on cognition. He has published more than 100 papers in various national and international journals. He had set up the Institute of Neonatology as head of the department at Institute of Postgraduate Medical Education and Research and SSKM Hospital. He designed the program RBSK, which I mentioned above, National Child Health Screening and Management Program for entire country, and was the national advisor to RBSK under the Ministry of Health, Government of India since its inception. He was involved in conceiving and implementation of this universal screening for more than 30 health conditions. Fewer than 4 Ds, including childhood diseases, childhood deficiencies, defects at birth, and developmental delay and disability, along with their management, including rheumatic heart disease. So with this introduction of the 3 speakers in the first session this morning, I would like to call upon Professor Arun Kasingh to deliver his lecture. Professor Arun Kasingh, please.
Both on the stage and off the stage. Indeed, it's a privilege to be invited to this historical place to deliver a lecture on the relation of genetics and society. Could I have the first slide, please? Now, when we talk about genetics and society, the beauty is that human beings, when they started settling down, they initially settled down with agriculture. As a group, they started having a faith in God, religion. At the same time, as a Homo sapien, one of the key differences with all other animals, that it is not your height, it is not the beauty of your skin, it is not your body weight which is essential. What is important is that Homo sapiens universally are the Z of the evolution. They are known for their brains. So we don't have the height of a giraffe. We don't have the weight of an elephant. We don't have the beauty of a peacock. Today morning, I could hear the beautiful peacock's voice. But what human beings have is the brain, its own height, its own color, and its own weight. Next, please. So when I think about Gregor Johann Mendel, Gregor was the title which was given to him. Interestingly, whenever you talk of society, and as a medical science student, I was always in dilemma. My mother was a great believer in God. She used to practice religion. And I had the problem that whatever I read in science didn't permit me to agree with my mother. Yet I was confused. I was partially religious, partially scientific. That means when I was in problem, if I had exam to pass, I would become religious, but at other times, it was not. Much later, I quote that the father of modern pathology, when he said, where science ends, belief starts. And where belief starts, science ends. But now I add a line. It is not the duty of science to hurt the believers. The duty of science is to convert the unknown into known, so that the domain of unknown, the need for calling for other powers becomes less, because you can predict better. You can predict today the plants, you can predict the weather, you can predict the lot of diseases. So Mendel was in a unique position. On one side, there was a church. He was a German speaking, coming from a poor family. Parents were agriculture. Today, if you have to visit his place, you have to go to Czechoslovakia. Earlier, it was in Austria. On the other hand, his desire to understand nature. Let me quote you that to me, God is nature. To me, if you cut a plant to make a temple, I would quote that you are destroying the nature, the same very God which you would like to worship. So who am I? What is science? What is music? What is plants? It is all about nature. And in Bihar, the place of Nalanda, there is a festival which doesn't worship any statue or god. Chhat Puja hoti hai. And in Chhat, we respect the nature where both the sunshine and the sunset is there. So I can understand that the Gregor name which was given means a person who is vigilant. I was just thinking, उसी दौरान हमारे देश में भी तो एग्रीकल्चरल साइंटिस्ट रहे होंगे. साइंटिस्ट किसको कहते हैं? जो प्रिडिक्ट कर सकते हैं, जो ईमानदारी के साथ, सच्चाई के साथ प्रश्न रख सकते हैं. क्यों नहीं मेंडल का जन्म हमारे देश में? हमारे देश में भी तो लोग मंदिर मस्जिद में काम करते होंगे. क्योंकि one of the things which was permitted by the church, they allowed you to question and put forth your views. That I think was the word. So you are a watchman to observe the nature. You are a watchman to observe what is happening. Why is Dr. Singh not tall as some of you? Is that very important for his contribution to the society? All these questions had to be raised. Next slide, please. Yeah. Okay, you can from here. Yeah. Okay. So if this is the place where you would go, it's about from Prague, if you go, it's about 3 hours journey from there. But what is very interesting about his place, that the place in which he was born, the place where he studied, is surrounded by a diversity. On one side, you see Germany, south, you see Austria, Poland, Slovakia. And if you see, these are all different languages, different cultures. He himself was a German speaking, but placed there. This diversity is very, very important. And I must say that somewhere I wish we start respecting the diversity of nature. So all of you know this famous garden. You have far more gardens than I can think of. I wish I was introduced to this institute earlier. I would have not become a medical science, but I would have taken up agricultural science. It's still far more exciting. So just see how things happen. He loved teaching. Some of his students have contributed much, but he was always failed as a teacher. Twice he sat in exam for becoming a qualified teacher. Both times he failed. Sometimes our examination system cannot identify a genius. His family, Gregor Mendel, though he was a person of genetics, he didn't marry. So the gene transformation in his own family never happened. He had no children. He had parents, as you can see, and the place where he did his work, there's a statue. Earlier it was known as Brno, now it is Brno. This is the university, the current university from which he had graduated. These are the gardens. What was very important was, we all have eyes. I keep on telling my students, I teach newborn. And I say in the science of newborn, forget all the machines. Do you look at the child's face? Can you understand the language of a child's cry? Once I did a research with IIT in which I collected all the cries of a newborn. And a database was prepared, and I would invite all the professors, both from our country and outside of neonatology, to listen to a cry and tell me why the child is crying. Do you know who scored the highest? If you understand Mendel, you could always say it was the illiterate mother who was giving birth to the second or the third child. She could predict far better than any professor. And that day I understood that what Mendel taught you, that forget all the equipments. The 2 eyes which have been given to you, keep on observing every small thing, the color, the shape, everything is so important. And if you have the patience to do so, all of you can become a Mendel. Perhaps we, as a Homo sapien, lose our patience. We want quick answers, and we confuse academia with science. Academia gives you degrees. Science is always a lone voice. It is not the voice of majority. Academia is majority. If I'm elected a professor, then there would be a group of people who will say, are you suitable to become one? But in science, a person who has never visited to a school can be the most scientific person, and his contribution can be far more. So let's not confuse academia with science. I keep on telling my students, are you come for a degree or are you want to do science? Both are not the same. So the beauty of his work was he could wait for 2 generations. The beauty of his work was that though his eyesight was not good, He respected nature. He broke all the walls of religious beliefs and dogmas. He wrote and observed what he saw. That's the first principle of science. There are no Bibles and there's no holy priest. Whatever I'm talking today, I may be completely wrong. You have 100 billion wires known as neurons in your brain. You have to decide whether it makes a meaning to you or not. So before that, so he made a law of segregation, which all of you know, I would not waste your time, and law of independent assortment. But today, and he died of a disease. He died of nephritis, which is known as a Bright's disease. Today, still we don't have a treatment for Bright's disease, where you start bleeding through your kidney and you die at the age of 61. Now, today we only should not remember Mendel. We should remember all the people who have contributed, which has made you do all kinds of experiments. Life has been made easy over the last 300 to 400 years. So you have Charles Darwin, just before Mendel's work. You have Watson and Crick, and of course the genome mapping. I'll just take you to a quick timeline. Well, after 1865, you had the chromosome. So people were asking, what is a chromosome? Before that, cell was discovered. And it was, well, the Fleming who showed. Before that, microscope had already been discovered. And you know who discovered the microscope? Father and son who were working in a spectacle shop. They discovered the microscope. There was further improvement on microscopes. And finally, people saw within the water the first bacteria. So all this was going on. What was interesting was that one of Morgan's student, Alfred Sturtevant, he was the first one to map out the entire linkages in Drosophila. So the genetic mapping of chromosome was done in a fruit fly. You can understand how medical science with the diversity is developing. Incidentally, he was colorblind. And I do not know how he completed his studies. So sometimes things are, because one of his areas of work was to see the red color tones. One person I think should have been, so I've just arranged it that the two Jansens, it is said the son got it, but son was a teenager. He couldn't have invented a microscope without his father. So the Zacharias Jansens, Robert Hooke further improved on it. Leeuwenhoek, he was the first one to find the bacteria in the water. Charles Darwin, of course, came in 1859. 1878, it was, well, the Fleming who identified chromosome. 1902, these two people, Walter Sutton and Theodore Boveri, they independently proved the theory of inheritance through chromosome. Then, of course, the American geneticist Thomas Hunt Morgan, who found that genes are linked in a series on chromosomes and are responsible. You can continue this. 1923, Painter, who counted the number of chromosomes but made a mistake. He thought human chromosomes are 48. It was not till 1956 when that was corrected to 46. Now, I have, and this is the very famous experiment which shows that it was a semi-conservative way in which DNA. Remember DNA, after Thomas Crick got it, the structure, it's a double helical structure. So people thought, and even Watson and Crick thought it, that this two double helical structure would separate and each would act as a basis for developing a next one, because you know the ATCG, they can keep on matching. That's so the complement stance will be a new one. Some people thought, no, this is not possible. So that was a conservative approach. And some people thought it is fragmented. Anyway, it was proven the semi-conservative way. And then if you see from 1944 onwards, genetics moved away from cytogenetics to molecular genetics. And of course, you have the genome mapping. But one story I would like to tell you. It relates to my experience also. Oswald Avery, if ever a Nobel committee has done a mistake, the mistake was done by Oswald Avery. He was alive, suffering from 1955 from liver cancer, when he found that instead of him getting Nobel Prize when he was nominated 30 times, his own work contributing to Watson and Crick DNA structure is getting the Nobel Prize. So one of the biggest mistakes ever done by the human committee is not to have given him the award. So what he did? He was doing an experiment with a bacteria. And this was what you say after the match, the extra time. So he had already retired. So again, a theory, extra time is not a wasted time. He had already retired and he was looking at certain bacteria which was leading to pneumonia and death. And what was that? So he had 4 pairs of rats. One, he injected one rough strain. This rough strain was not so virulent. The newborn survived. The second, he injected a smooth strain which was very virulent. The mouse dies. The third, he takes the same virulent strain, he heats it, now the mice lives. The fourth one was a magic. He took the one which was non-virulent and mixed with the semi-virulent, and the rat died. How could it have died? Because the heat killed part never could kill a rat. So there was something which was transformed from one area to the other bacteria. Very similar to this, all of you have heard of NDM? Anyone? N stands for New Delhi. You are sitting in New Delhi. So in bacterial resistance, somewhere 2010-11, you have read in the newspapers, there was a specialized bacteria which was resistant to everything, named after New Delhi beta-lactamase, NDM. I was in Calcutta and I was working with a group of scientists. We were actually working on fungal infection in newborns. So the microbiologist who went with me, she said, sir, it was a wasted day. We collected skin samples from the hands of the doctors and from the patient, newborns. There was not a single fungus. There was some bacteria, E. coli, which is a common thing to get from the perianal. 7 days later, she said, sir, 5 children died there. All their blood cultures were growing E. coli. When we did the DNA and matched it, they were not matching. They were partially matching with 2, they were partially matching with 3. At that time, no one in the world had heard of NDM. So Sulagna Basu, who was one of the scientists working at ICMR, I told Sulagna, try to think, there must have been some exchange. Today that paper is published, the first bacteria to be isolated in newborn NDM gene, resistant gene. And that came where we showed in the lab how bacteria are made in hospital, virulent bacteria. They do not come from outside. If we don't maintain hygiene, one bacteria transfers its gene to the another, so the non-virulent bacteria becomes virulent, and there was death of 5. That paper is in BMJ. So, And how did he come to know that this is DNA? Why was he not getting a Nobel Prize? Because someone sitting in the Nobel Committee believed it was the protein which was the basis of transfer and not a DNA which has only 4 nucleotides. How can such a simple ATCG carry the entire genetic code? What experiment did he do? He took the material, gave proteases, found that the rats were still dying. Then he used ribonucleases. So obviously it was not RNA. Finally, he took deoxyribonuclease and the rat were no longer dying. So deoxyribonuclease is an enzyme which gives. The simple experiment changed the whole story. I think after Mendel, this person who was so silent in his work. In medical science, even as a student, we were taught it is protein which create antibodies, basis of all vaccines. He said, no, it could be carbohydrate too. No one believed him. Today we know he spoke the truth. So I will take you to the negative aspect of genetics. And this is eugenetics. After Mendel's work, etc., and I find this in everyone, all of us in some way take great pride in our lineage. Taking pride is one thing, but trying to think it is superior to others is a very, very negative sense. Because the genome study has shown that the blue-eyed American and the black-colored Negro have 99.9% same genes. Nature has not discriminated. The 0.01% difference is the color of the skin, the shape of your hair, the color of your iris. As a doctor, if I cut the skin, if I take the brain, I cannot differentiate. Is this a Brahmin's brain? Is this an American brain? Or is this a brain of a Negro? Nature has been very kind not to have any of these discriminations. So eugenetics was a racial improvement and plant breeding which happened. And these were the posters of the Nazi regime. Can you understand? One of the posters in Nuremberg says that maintaining of the purity of blood ensures the survival of the German people, and we are the Aryans. The second paper says the feeble-mindedness and alcoholism are passed on from one couple to their 4 children and their families, so they must be finished off. You will be surprised to know that IQ was discovered not for a scientific purpose, but to show the superiority of the white race over the black. And today, let me be categorical that if I take a 5-year-old white boy and I take a 5-year-old black boy, of course, the white boy is more intelligent than the black boy, but that has got nothing to do with genetics. That has got to do with environment. If a child doesn't get a positive environment, if he's out there on the streets, Sir was mentioning his own students, but supposing there was no university, supposing a student didn't have the ability to come to him, can the brain get developed? So, ladies and gentlemen, we require institutions. We require the support of a country. We require the support of a family to form a brain. Don't blame the God if your children's IQ is less. We have not understood how to make an IQ. So, कई बार लोग जय हिंद बोलते हैं. मुझे समझ में नहीं आता है कि आप लोग समझ के जय हिंद बोलते हैं या बिना समझे. क्योंकि हिंद क्या है? आबिद ने जब नेताजी सुभाष चंद्र बोस को कहा कि एक ऐसा टर्म कॉइंट करें जो सबकी जुबान पर एक जैसा हो, चाहे आप सिख हो, चाहे हिंदू हो, चाहे मुस्लिम हो, तो आबिद ने कहा जय हिंद. I tried to understand what is Hind. I'm at Jodhpur, and if I go straight, it will go to Karachi. So, very simple term is 2 mountains and 2 seas. One side is the Hind Kush, other is the Himalaya, from north to west and from north to east. The 2 seas, the Bay of Bengal and the Arabian Sea. That is the Hind. And when you say victory to this geographical place has a huge common sense, has a huge similarity in its approach, in its thinking. So, am I proven right? Now you know. How many of you are aware that we have already published the Indian Genome Study? So the Indian Genome Study talks about the entire country. This genome study, which is done partially in Delhi itself, collecting samples from all over the country, we find only 4 traits. 78% of people sitting in this hall are Indo-European, which means you carry all the genes of Europe. One group of people who migrated from so this is the group which came from Hind Kush, the Khyber Pass, migrating from the north. The second group is Dravidians, which were supposed to live in the north, migrated to south. The third group was Indo-Tibetan, which came from 40,000 years before the birth of Christ from the east. And the fourth one was Austro-Asians, which are the tribes. What does it mean? Bajar शब्द एक टर्की का है. Chai is the Turkey word. What it tells us, this study, that your genes have moved with languages, not with religion. This study was evolutionary, and it shows that India is a melting point of all the genes of the world. The strength of India is not in its gold and diamond. We never had gold mines and diamond mines. What we had was, who said this? 100 years back. So this is all, if you are interested, you can read about, can this be? You can read about all the diversity of the genes from all the places of the Europe. But majority of us are mixed population. I think it is hang ho gaya. So I am remembered of a poet who was born 100 years before, never went to an academia, got a Nobel Prize for Gitanjali. Just see the few lines of Gitanjali. No one knows whose call made streams of so many population come from whereon turbulent currents lose themselves in an ocean. Here Aryans, here non-Aryans, here Dravidians, Chinese, Sakas, Huns, Pathans, Mughals, all dissolve in one body. The doors are open to the West today. All brings gift from there, give and take, meet and merge. No one turns to leave on this seashore of great humanity. These are the lines of Gitanjali. 100 years before the genome study shed the same thing. Sometimes I think poets are far ahead of scientists. And this is try to understand the Hind. All kinds of things, they merged. Aapka sitar is not coming from one place. Urdu is a language which is not spoken anywhere else, the subcontinent. Science has grown here, arts has grown here, mathematics has grown here. We call it Indo-Arabic, that this cultural meet, this diversity is so very important. So one of the things which genes teach you, remember Mendel was a German speaking. He was working in a different area. Even he was a Catholic Jew, Catholic Christian. But does it matter? As science can be understood by those who are believers and non-believers. And if you have any idea, बिहार में यूनानी मेडिसिन कहते हैं. मुझे समझ नहीं आता था यूनानी मतलब, एक दिन समझ में आया यूनान मीन्स ग्रीस. When I saw the genes You have so many Greek genes in all of you, you cannot think of. The beauty is, in Greek, there are temples, hundreds of temples. Sani Bhagwan India में भी है and Saturn temple वहां भी है. महाभारत जब लिखा जा रहा है, रामायण जब लिखा जा रहा है और इलियड जब लिखा जा रहा है, एक तरफ सीता का एब्डक्शन है, दूसरी तरफ हेलेन ऑफ ट्रॉय का एब्डक्शन है, लीडिंग टू ए वॉर. You see the timelines. It's too much of a resemblance. What does it tell you? It tells you I and you both are the same. We are all Homo sapiens. And all of you can become a Mendel, a scientist, provided you fight your own dogmas, you relieve of yourselves. You are not a Keralite, you are not a Tamil. At the end of the day, you are a Homo sapien, where you are observing the nature in its best beauty. The more you allow this mixing, the better it is. That's what the genes taught you. This was not understood by eugenetics, and this led to disaster. My few 4 minutes I will take. Can you improve the development of the brain of a child? And I was talking to Sir that One Health concept is a very unique thing. Today, medical college cannot develop medical science in isolation. I wish the agriculture university, the fisheries, the anthropology, all would join hands in one place and understand nature and not divide it into organs and areas. Because most of the diseases, including COVID, is a combination of humans, wildlife, and pets. And there is one thing which is impacting every one of your children is the digital toxicity. Please be very careful. Most of the studies. Tell your children stories. Tell them the stories of Mendel. Tell them the stories of Newton. But don't show them the same thing on a TV or a smartphone. It doesn't mean the same. How genetics has proved disastrous in India. There's a classical example. The first day of my medical college, my physiology teacher, Subhash Mukhopadhyay, a class of 100, only 5 people were sitting in his class. Unfortunately or fortunately, I was one of them. He took only 3 classes. Up till now, I have not met a greater teacher than him. Who was he? Subhash Mukhopadhyay was the first person in the world to have done cryopreservation and formed the test tube baby as early as 1978 on the day of Durga Puja known as Durga. 2 months earlier, Robert Edward also had made the first test tube baby, but he was not successful in doing cryopreservation of the ovary. His method is not followed today. We follow Subhash Mukhopadhyay's method. One person got a Nobel Prize. The other also got a prize. Can you say what prize? He was ridiculed. He hanged himself to death. He was transferred. I could attend only 3 days. Today, what did he say that people went against him? He said, if you are making IVF, don't try to become God. Let not IVF be in private. It could cause serious disaster. It should be very carefully planned. Today, when I see the results of the Cochrane review, it has shown there are multiple births. There are more chromosomal abnormalities than you can think of. You have played with nature. Nature allows one egg to select out of 1 million sperm, 1 sperm. In a lab, you take 100, 10, whatever examination you do, the birth defect is going to be very high. This is the meta-analysis. Increase preterm, increase low birth weight, increase. What is the commonest cause of IVF? We have not addressed. We have become a commercial country. We want problem to occur. If you go home today, understand infertility is nature's not provision. It's our provision. What did we do? We allowed our daughters to become obese. We allowed them, no one told them what kinds of plants to be taken, what food to be consumed. And this leads to a thing called PCOD. I incidentally had a picture taken by one of my classmates when he was teaching. And the first lecture he was talking, at that time he had not yet he had not disclosed the world that he has already discovered test tube baby. He said, stress is the most important cause of infertility. Most important cause of obesity. Which IVF center tells you this? So we do require. Stress has led to more preterm children. Don't quarrel with a pregnant woman, even if she makes a mistake, because her genes require an environment to reflect. This is known as epigenetics. I have a fan. Fan doesn't move till the switch is made on. The switching on is done by the environment, which is the epigenetics. You may have good genes. That doesn't mean you'll become good. That switch on and off mechanism has to be there. That starts in the mother's womb. If you are quarreling with a mother, pregnant mother, you are quarreling with her, your future. You are impacting the genes which will reflect into protein synthesis. This was a beautiful paper, the white paper on maternal newborn in Europe. A small thing called dental infection can infect the child. And this too is with the release of certain chemicals known as cytokines. You have heard of cytokines now. What does famine cause? The only country of the world which has got the data of all people in the last 100 years, even if the baby died one day or you survived 100 years, what kind of diseases you had, what food did you take, what about your grandchildren, all the record is maintained, and that is Netherlands. 1944-45, there was a famine. This is the most important genetic study, the impact of famine. This famine was only for 1 year. It was because of Hitler. They have even documented the amount of food which the mother was taking. It fell very short. They are now publishing the 4th generation data of those mothers who suffered from famine versus those who did not. What happened to them? The first they found, and let me tell you on record, genetics teaches you, nature is more female-centric than male-centric. I'll give an example. When a girl is in her mother's womb, all the eggs are formed when she is still in her mother's womb. Not a single egg will be added. This doesn't happen with male and sperms. So if I take a medicine, I give it to a pregnant mother, not only her daughter is impacted, her granddaughter is also impacted. 2 generations straight off. But what they showed, that in the 4th generation also, for a famine which happened in 44-45, there was more heart diseases, there were more lipid disorders, there was more obesity, there was more asthma, there was more diabetes. Sometimes people say, यहाँ पे तो खाना ही नहीं मिलता है, कैसे diabetes हो जाता है? They don't understand genetics. This is the kind of genetical change which happens. If you suffer from malnutrition, just like Mendel said, the next generation will not develop obesity, but after that, you'll find obesity, etc., everything. Because now the food has increased, but the genes, they know that there'll be less food, so they can't match it up. And this is what happens. So comes the beauty of epigenetics. The same thing happened in Bengal. I'm yet to see a research paper. What happened after the famine of Bengal in terms of genetics? If you get one, please do help me. The Dutch winter has shown just 1 year earlier, that was caused by Hitler, this was caused by Churchill. What happened to them? This extreme degree of famine, which I collected these photographs from the newspapers kept in the archives. This is a famous experiment done on the desert mite in which where I'm working. They took these mites from Rajasthan. They were they were not obese. All their cholesterol lipid level was okay. When they took it to Boston, And they start feeding that food which they were giving to their own rats. All of them developed obesity and etc. Which shows that your genes are affected by environment. So healthy food can change your genes. That is the environment which you are in. So what are the things which is impacting epigenetics? Breastfeeding impacts genetics. Yes, I can give food by talking to Nestle company and giving it. But the epigenetics will get defeated. I can get delivery by cesarean. But a normal delivery, the bacterial flora allows the genes to express themselves. So nature is the biggest scientist you and I can think of. Don't fool nature. I can produce a baby in 5 minutes. Nature takes more than 24 hours for a newborn to travel a distance of 30 centimeters called birth. There must be a reason for it. Today's lecture is not for that. I have a lot of work done on that also, which I learned from the tribes of this country in Andaman. So the feeding type, the home environment, the maternal microbiota, your food, all this impacts. If you want your children to be intelligent, protect the nature with all your might. Remember, the God you worship and the God you destroy are one and the same. This is, when I talk of Hind, I talk of this statue. This is the last one slide. What is this statue? In Peshawar, which is the entry point to, as if someone has cut a pathway in Hind Kush mountain, the Khyber Pass, there's a small place called Peshawar, from where all your heroes and heroines of that famous movie Mughal-e-Azam come from. They got a statue, they got this metallic thing buried by the Britishers in 1917 or 1920. That is now kept in the museum in Burma. What does it show? Buddha is sitting. On one side is the Indra and the other side is the Brahma. Why should Brahma and Indra standingly salute to Buddha? I was very intrigued by it. The language is written by Kanishka. If you remember, Kanishka was an emperor at that time. My reasoning to it, Buddha is knowledge. To knowledge, even Brahma and the powers, the rulers, so-called Vishnu, also have to bow down. Thank you very much.
I'm sure you all must have enjoyed this very interesting lecture of Dr. Singh. If there are some queries, clarification, questions, probably we can take up few of them. Please raise your hand. Mike, please.
Sir, I think, I don't know whether you knowingly or unknowingly skipped surrogacy. It's a very serious business, particularly in India. It has a lot of involvement, legal, societal, genetical, and can you please say something about it?
So I think I opened the broad topic. Let's not play with nature, which I was taught by my teacher. Remember one thing, when I ask a parent to become a mother or a father, I always tell them you are going to be a parent of a Homo sapien child. Anything which impacts, when a mother touches the skin of a child, the information goes to the frontal lobe. When I touch the same newborn, As a neonatologist or the father touches it, it goes to a different area of the brain. Surrogacy is a joke. Don't do it. If it is necessary, please explain. All good things also have limitations. I'm not saying that without explaining, don't subject it. Explain everything. If the parents are still ready, go ahead with it. But our duty is to tell the truth and let the parents decide. I'm not the God of the child. I can only place the facts. And the fact is, in the last 1 month, I'm doing experiments where if a newborn is born preterm, he's on ventilator, I allow the mother to touch the baby. That very touch itself improves, the ventilation time becomes less, the sickness improves. Close your eyes. When your mother touched you, it is not only an art, it is also a science, because that goes to the emotional area of the brain. I'm wanting to do research. I'm wanting to develop an MRI for the fetal and newborn brain. The operculum, which in Latin names as the lips, when you separate it, lies the incus. So this area of emotional brain is, when the mother speaks to the child, the left side of the brain gets stimulated. When the grandfather, grandmother or father speaks, the right side of the brain gets stimulated, because the baby knows only one person before birth, that is the mother. Smell is such an important thing that I have seen, if I give body spray to the mother, the baby refuses to go to the mother. The baby smells his mother. The baby's vision is very poor. In nature, any animal which has a bad vision, the smell is very strong. Dogs come and smell you because their vision is poor. Babies have an excellent smelling power, which decreases, and a very bad vision power. They only see their mother's face with the outline of black and white, what you in computer do, 100% contrast. So I think I have been trying to tell you, I'm not passing a sermon. Please don't mistake, I'm a scientific person. I may be wrong. These are my views. I'm talking of a broader philosophy that plays the truth which is known today. The truths may also change, but one truth will remain: the laws of nature. What Mendel did was the law of segregation applied to one and all. That was the beauty of his work. That is the beauty. You could apply it to plant genetics, you could apply it to human genetics, you could apply. So these laws of nature allows that you become a mother not the day you conceive. You become a mother the day you dream of becoming a mother, because your neurotransmitters change at that particular time.
Are there any more queries? Yeah, there is. Can you?
Sir, it was one of the enlightening presentations, sir. It's just the extension of earlier question. In case of, we are talking about GM and transfer of genes from fish and any other animals, bacteria to plant system, and also expressing even for vaccination, we are transferring. In that kind talks, what is your opinions? Yeah, because in the main message of your presentation was that's not play with the nature. Just I wanted to know your opinion.
But it has another meaning also, that we should not stop the development of science. Had we not used bacteria, a lot of answers would not have come. So I'm only trying to say, whenever you do some experiment and you escalate it in the society, please remember that all the facts should be placed on the table and the limitation should also be. When my student says, this is the best medicine, I say, have you also understood the limitation of your medicine? So I think the point I'm trying to raise is that limitation should also be raised. But again, let me tell you my ignorance. I honestly admit I'm not the expert to pass a judgment on it. I have not done enough research to have told you that the vaccines developed by there are a lot of Lancet papers which talk of autism, etc. I'm trying to tell you, truth is something which takes decades to know. But the broad principle I was trying to tell you, that please also tell the limitation of things. What my sir was telling that day also is the extension, that Test tube baby may be useful, but have we understood the limitation? That is the point I'm trying to make. हमें दुकानदारी नहीं करनी है, हमें साइंस करनी है.
Thank you, Dr. Singh. Thank you very much.
Thank you, sir. Now it's tea break.
Good epigenetics.
Into the session.
So after tea, I think you all are refreshed. So we can go for the next lecture. Now I would call upon Dr. N.K. Singh to have his talk on accelerating breeding through genomics. Dr. N.K. Singh.
Good afternoon, sir. And welcome everyone here. Respected Chairman, Professor Arun Kasingh, my teacher, Professor R.B. Singh, also my teachers, and I have so many students who I have taught, I know, who are here. All students of genetics. We are all students of genetics. And you have heard really stimulating lectures for the last 2 days and just before this wonderful lectures. So I don't know whether I can do justice to that. And I'm very thankful to the organizer, Dr. A.K. Singh and his team, to include me in this illustrious list of speakers. What I'm going to do today to talk about the science of genomics and how it has been applied for developing varieties for the farmers. So I will not go into too much detail of the science part, but I will just give you some examples. Anything? Yeah. So at the outset, why those things are coming on the top? At the outset, the title is hidden actually. I would like to acknowledge actually to start with, because usually we run out of time in the end. Whatever work I'm going to describe, mostly I will talk about the general principle of genetics and genomics, but mostly the examples I will confine to our own work. And this is not my work alone, because this work for so many things, so many scientists through large networks of scientists. Yes, go back. Can you go back to the first slide? It's not working. Anyway, can you go back to the first slide? I would like to acknowledge the financial support from Department of Biotechnology and ICR, who have been mostly supporting my research. And there are some large network projects, MPTC, FGGM network, and QTL to variety network. DBT has funded rice, tomato, and wheat genome projects in a big way, and also a QTL to variety network project. So I want to acknowledge their financial support, without which it is impossible to do this kind of work which I'm going to describe. And the large number of scientists have been associated with me, very well-known prominent scientists. Once again, it is because of their contribution what I have been able to do something. And the even larger number of the students and RSRF in the project. So I like to put on record their contribution, which I'm going to speak. This is the outline of my presentation. Somehow the heading is hidden by that panel of things. I don't know, but this is the outline. I will just talk about why we should sequence genomes, then give very briefly overview. I'm not going into detail. The genome sequencing work in India, how it started, and these international partnership for rice, tomato, wheat, and flax genomes. Then functional genomics, once again, very selected example of gene discovery and its utilization in rice breeding. Then a little bit about a major translational genomics project from QTL to variety. Just a few years back, Professor Gupta, P.K. Gupta was asking in several meetings that has any variety been released with QTL, because he was a bit skeptical that these QTLs are not so important. But now I will show you that not one or two dozen varieties have been released and several are in the pipelines also. Then search for new genes or some exploration work which we have been doing. Genes are in the germ plasm in the nature, and I will show you that how important it is to protect that germ plasm, explore it, and make utilization of that. And BPGR representative were here in this morning. Then we started our own genome projects after learning how to do it. Pigeon pea, jute, and mango, which are uniquely important for India. Nobody will put their money for that. So I will just very briefly give an overview of that. And some other genomes where we have supported, and few genomes where other scientists of the country have done good work. I will just give you an overview of all those things. So more detail will be on the rice and how because the rice is the forerunner. It is the first crop plant to be sequenced. And then its applications are now on the farmers level. Others will follow in the same direction. Well, there are some terms and concepts we have been hearing about that right from the first lecture by Professor Arun Kasingh and so many luminous lectures. I don't want to repeat any one of those things, but I want to start with the definition of the genome. That's why I put in the perspective here. You have heard that the Mendels did not coin the term genes. It was coined by Johann. Alleles term was coined before the genes by Bateson and Saunders. Mendel called them factors. And the gene, genotype, and phenotype were called by Johansson. See, all these terms were called before we didn't even know that what is the genetic material, what is genes are made of. At that time, the science was not that advanced. Now we know that what genes are made of. And the genome which we are going to talk about, that is also 100-year-old definition by a German botanist, Hans Winkler. He defined the genome as the complete set of genetic information present in a single haploid cell of an organism, including its protoplasm. So he knew about the maternal inheritance, that something is in the protoplasm, something in the nucleus, and he defined the term genome, the entirety of all the genes. The Mendel was talking about the single gene and monohybrid and dihybrid ratio, and he very meticulously, through his very hard work, he came up with the principles of genetics. Many people criticize it, that manipulation and all those things, but definitely his work, that's why we are celebrating after 200 years of his birth. So I want you to focus on the definition of genome and how it is useful, why we should spend money and our resources to try to decipher, to decode the genome. Just few years back, 20 years back, the genome decoding was very expensive science. To buy a sequencer and to develop a lab, many people were so skeptical that why these people are doing air-conditioned science, sitting in the lab, spending crores to buy sequencer, doing sequencing. But not only we were doing, the whole world was doing. So this answer is for the entire world. Why the whole world is doing sequencing? Of course, you will know that in the COVID epidemic, how useful it was that this strain is coming, that strain was coming, and all the COVID variants were sequenced. Only by sequencing, we knew that which strain, whether it is alpha, delta, or omicron, whatever it is, and the RT-PCR techniques were developed. So immediately it had application to the society in case of medical. But in agriculture, I will be just giving you examples. The very first is that the human curiosity, our basic knowledge. We all want to know that who we are, where we have come from. We just heard that all races of human, they have the 99.9% similarity. But when we get the complete genome information of different species, we come to know that not only humans are brother, even plants, animals, bacteria, they all belong to the same family. They all have the common lineage, and the life has originated on earth, and everybody has, all the living beings have common ancestors. Only the how far back they had the common ancestors. So it's a degree of relatedness. So once we have the complete physical map, genome is actually physical map. Physical map is actually reality, which you can see with your eye. You can even hold in your hand. You can hold the DNA in your hand. You cannot hold the gene in your hand. Gene is an abstract term, and genetic map is also an abstract term, but DNA is real thing. You can take the DNA in your hand, you can see it, you can feel it. So once we have it, we can compare the sequences, and then we can find the evolutionary and phylogenetic relationship. Then the second part, we are coming more towards the practical utilization. We can do molecular characterization and high density fingerprinting of germplasm resources. Now we know that after the acceptance of germplasm as a wealth, as a resource under the biodiversity treaty, mostly the developing country, they are rich in biodiversity, they are poor in technology. So this acceptance of biodiversity as a resource, as a wealth, is a great thing. But how are you going to claim your right unless you can fingerprint it, unless you can mark it, you can prove it that somebody has taken your material. So unless you have a high density fingerprint, you cannot prove. People will take you, and you can just simply blame it, but you cannot prove it in court of justice. So having genome information allows you to devise very high density fingerprints of your material, at least the important material, and it can be traced back. Whoever steals, even 100 years later, it can be traced back. This material comes from our germplasm, which our farmers have protected. Then genome is very useful in gene discovery also. Once you have the genome, complete physical map of every chromosome from telomere to telomere, you know the complete landscape, where are the genes, how many genes are there, what is in between the genes. So this is just like having a GPS in your hand. Now this is a very practical example, everybody knows. You hire a Uber taxi, and you don't even have to talk to the driver, and you can know that where you are going. It's all because we have the complete map of the street and everything. So gene discovery, when you want to find which gene is responsible for a particular trait from amongst thousands of genes which are in the genome, typically there may be 25,000 to 50,000 genes, out of which is the gene which is making the difference. Genome is less like having a GPS when you're trying to search somebody on the street or somebody in a particular city. So it is that map, that important it is now. Without the genome, it's like looking for a needle in the haystack. Then actual breeding process itself, the selection, having genome information, you can practice marker-aided selection. Earlier, you were doing just simply phenotypic selection based on the trait evaluation and your own good eye of the breeder. You are selecting, but now you can do, with the help of genome sequence, you can have markers for the trait and the background. So these are the increasing order from the very basic to very applied. Having the genome information is very useful, and you will see with the example that how rice breeding has revolutionized, and now we are getting variety, better and better variety every day. Not only with higher productivity, but also with better nutritional quality, better resistance to environmental stresses. And I will just go through very quickly through our journey, because quite often it is criticized that what damage Green Revolution has done, but that was important for improving the production. But now we can go back and restore all the quality traits and resilience which were lost during the Green Revolution. Now I think unfortunately those title is the half the message and it is hidden. Can you put at the bottom that thing which is the bar which is hiding the title? Here I want to show that the genome sequencing is easier said than done. Why genomes are so hard to sequence? It has become quite cheaper, at least 2 orders of the magnitude since we started the RISE Genome project. Now it was about $1 per base pair. Now less than 1 cent per base pair, you can decode the genomes. But the plant genomes are actually very, very large in size. They are huge. If you take 1 picogram of DNA, that means 10 raised to the power minus 12 of a gram is 1 picogram. That will have 10 raised to the power 12, 1 followed by 12 zeros, that many base pair. So you can see how fine it is and how informative it is. So wheat, for example, it has got 17 picogram of DNA in a cell, applied cell. So it's got 17 billion base pairs, which is 40 times bigger than rice. Most of the microbes or bacteria will have about 5 million base pairs. So there's enormous range of size of the genomes, and the bigger the size, very difficult it becomes to assemble the genome, just like the jigsaw puzzle. If you have big jigsaw puzzle and small pieces, it will be hard to join it together. So that's why rice was started first. We are lucky that the rice has very small genome, and Arabidopsis was the model genome. But wheat, 25 countries and some private company joined hand to sequence wheat, and it is not complete. Still there are gaps in the genome because it requires a lot of efforts and money. So very quickly, I will go through here the genome sequencing in India through international partnership. So here we are talking about the, looking at the complete overview of the entire genetic information, not a single 1 or 2 genes, because now as a human being, we have developed the capacity to decipher the entire genome. So this science of genomics were developing around the world during the 1990s when Human Genome Project and Arabidopsis Genome Project were being worked on. Unfortunately, India was not a member at that time, but China was. So as a result, China was at least 5 years ahead of us in genomics at this time. And at present, because there are large investment in science, they are still much more ahead than India. We like to compare with China, but the kind of investment we have in R&D is very, very minuscule compared to China. But nonetheless, it was a very good beginning that DBT funded liberally for the RISE Genome Project. And Professor R.P. Sharma is here. He was the director of NIPB. I was at Pantnagar, and there were a series of meetings, and they called me from there. I consider fortunate enough. Dr. Sharma called me in a corner and he asked me that, would you like to be PI of the RISE Genome Project? So I was very happy. It was a privilege for me. And then we started writing this project, and then, we have to establish all the laboratory right from the scratch, learn how the genome sequencing is done. But Delhi University South Campus and our institute, which was part of the IRI at that time, we developed all these things. And we did this before the actually schedule, before the deadline. In 2002, the genome was launched. Earlier, the deadline was for 5 years. In fact, the whole RISE Genome project was to be completed in 2008, but it was finished, draft was finished in 2002 and finished quality reference in 2005 because of the international collaboration, and people really worked very hard on that. This is the commemorative event launched by the Japanese emperor in Tokyo in 2002. And after another 3 years of finishing is a hard work actually. You can generate draft very quickly, but if you want to finish it means without any gaps, very few gaps from telomere to telomere. The human genome telomere to telomere was just published just few months back. Otherwise, the first draft was published in year 2000. So these are very enormous challenges. Small gaps needs to be closed. So this reference genome of rice is still a really gold standard. It was published in 2005, and it is used very, very extensively by scientists around the world, both in other species also for the rice breeder and geneticist. This is like a GPS of rice. And now the genome has, now you have the concept of pan genome. Means one variety doesn't have all the gene of that species. All the variety will have some unique genes, and when we combine all of them together, that is the pan-genome, the complete set of genes for that particular species. So now the concept of genome is being replaced by the pan-genome. And once again, the purpose is the same, but say single genome will not have all the information. But when we combine the genomes of several varieties, that will have more, that will be more complete. Since then we followed, now we have gathered the expertise and we become competent. Then without training, actually, we were actually called to join the tomato genome project. And then Dr. Kallu was DDG at that time, and he was very particular. India should participate in the tomato genome. We took the chromosome 5 and we completed it. This time we took the longer South Campus again, and NIPGR 3 institute joined hand together. And we again completed this one. But something happened at this time actually. The sequencing technology was also evolving. So when we started this tomato genome work, we were following the same technology as was done in the rice genome. But that was quite expensive. As I told you that $1 per base, it was costing too much. That's why 10 countries joined hand to complete it. Similar thing started in tomato. Tomato genome being 2.5 times bigger than the rice genome, it was decided to sequence only 20% of the genome. Once again, the economics factor, because the euchromatic region is rich in genes. So it was decided we will sequence only 20% of the genome, which is rich in the genes, and we leave out the 80% of the genome, which is in the heterochromatin. This is the gray area here, contains 80% of the genome, highly condensed heterochromatic, poor in genes, highly repetitive. And the orange part 20%, even though it looks bigger here because it is less condensed. And that was decided to sequence that. But something happened in about 2006, 7, 2008. NGS technologies came to fore. I think Dr. Gupta summarized very beautifully all the development, how it happened. So these 3 independent technology, when this came, so you don't know in what is lies in the future. Only once this technology came, then we realized that whatever we were doing, it was a very slow, long, arduous path, just like a walking. As compared to the new technology, was like a super highway. Now in India, we have so many super highways. Even in the most backward areas, you have big highways and you can go at the speed of 100 kilometers. So that is the difference. These technologies, why they were like a super highway? Because you did not need to clone the DNA anymore for sequencing. Earlier, we have to break into pieces, clone in bacteria, purify. So every piece of DNA which was sequenced in rice, it was cloned in a bacteria and then sequenced and then put it back together to make the genome. But with the NGS technology, cloning was not needed. You can take the DNA, bring into pieces, and then PCR amplify. So cloning was, instead of doing in bacteria, it was amplification of DNA and a piece of slide, glass slide, and a well, micro well. So that made it much faster because now you could sequence thousands of DNA segments in parallel together and at a much cheaper cost. But bioinformatics cost became more. You need more people knowledgeable in the bioinformatics. So this time, these are the 3 techniques: 454, Illumina, and solid. Again, I will not go into detail because it's a general lecture. The basic concept is that you don't need to clone the DNA, and you can sequence large number of DNA fragments at a time. The read lengths were smaller, but the advantage of generating large amount of DNA overweight this thing. And then the Sanger sequencing has been now become thing of the past for genome sequencing. Nobody does Sanger sequencing now. And as a result, we could sequence more than 90% of the genome, and it was published in 2012. In the tomato, each chromosome from telomere to telomere, where are the genes, where are the repeats were also sequenced here, where are the different kinds of non-coding RNA, telomere, centromere. You could see the complete landscape here. So in the beginning, it was a revealing thing, but now we know that every species you will sequence what kind of things to look for. You have the repeat elements, you have transposons, you have the genes, and you have all the features and landscapes are known. Every genome we sequence, we find the same thing, more or less, with some minor variation. Of course, the variations are very important, but the basic features of genomes are now understood. Now, the third one is the Wheat Genome Sequencing Consortium. Again, seeing our potential, even we had so many people coming from USA and Canada seeking the partnership to sequence the wheat genome. See, why people were coming after us? Because wheat is a open pollinated crops, largely in the public domain, very little private sector involvement. So these genomes should be sequenced to the public funding and made available in the public domain. As a result, mostly the public funded projects were involved in this thing. And genome is so big, 40 times bigger than rice genome, that no country alone is going to put that much of money for the research. So they wanted that better all the people who are using wheat, they should contribute. India is the second biggest producer of wheat. So they always thought that it is important for India. And eventually, Indian government also realized that we should contribute. So DBT funded this project. And this time we got chromosome 2A. In mind that this chromosome 2A, a single chromosome is twice as big as rice genome. So once again, PAU Ludhiana Institute and Delhi University, we joined hand to take up the challenge and sequence this chromosome 2A using the NGS technology now. Now we are not going to clone and sequence. So we have evolved with the evolution of the sequencing technologies. Sequencing became cheaper. In the wheat was something very interesting, that once you have this NGS technology, you don't need to work together to sequence, because it can be sequenced in one place, in one lab, because you cannot share the back clones which you used to share for sequencing of rice and tomato genomes. But in case of wheat, it was possible to share, because chromosome specific DNA could be isolated by flow sorting. In wheat, we have wonderful cytogenetic stocks thanks to Arun Kasingh from University of Missouri, Columbia. University of Missouri, he has developed stocks where each of the chromosome is in the two pieces, two telocentric chromosomes. So the telocentric chromosome size becomes half of the full chromosome, so they can be easily separated out from the rest of the chromosomes. So this way, this chromosome 2A short arm and 2A long arm, we got the chromosome specific DNA of wheat for all the 42 chromosome arms, 21 chromosomes, 42 arms, and these arm specific DNA and back clones were shared around the world in 25 laboratories. And you know who did this work? Once again, the Czech Republic comes into mind. There's one scientist, Yaroslav, in Czech Republic. He meticulously, it is hard work, actually pure hard work and good hands. He germinates a lot of wheat seedlings and then dissects out the growing root tips, then prepares chromosome separation, and then meticulously sorts the chromosomes to prepare chromosome specific DNA. He prepared back library for each chromosome, and the number of clones were so large that it was shipped into a big truck from Czech Republic to the France, where it is being stored in -80. A big truck, just like we have the truck cargo going. This back library was shipped there, and we received the back library from Czech Republic, and the arm-specific DNA also. So these are wonderful examples of international collaboration to work together to, decode the genomes of the crops like wheat, rice, maize, which are the, you can see the main food security for the whole world. And having the genome sequence is it is helping actually marker development, gene discovery, and other things. So in each of the three papers which I described you, two in the Nature and one in Science, India's role was very substantial, because we have 25 authors in each of those three papers. And 25 authors, nobody will give you in the Nature and Science unless your contribution is substantial and important. So we got training also. Many of the young graduates got trained, we got trained. In fact, Dr. Mahapatra, Dr. Tia Sharma, they went to USA, Clemson University, Cold Spring Harbor. Everything we had to learn and come here and apply. But we again, when we learned it, then we trained a lot of graduates, bioinformatics particularly, and they are now at the key positions around the world. This is one area we need a lot of human resource development. I don't have the time to go into detail, but that's where this started. That's where the shoe is actually pinching. We don't have the large number of trained bioinformaticians to make use of the genome information or to assemble the new genomes. So I don't know if somebody is listening who matters to take care of this problem. In Flax genome, we did a minor role, so that's why we have only 2 authors here. This was a draft genome. Now, sir, let's come to my third point, is the gene discovery and utilization, process flow and timeline. And that's how I have tried to summarize here that how genomics can accelerate the breeding process. Genomics is not doing anything new. It is only providing you tools to speed up your breeding. Earlier, we used to cross in, then manually select it through visual selection. We had some understanding of genetics of some traits, and the breeder has very keen eyes to pick the plants which will perform better. But now we can do selection through markers, because we can get a greater understanding of the relationship between the DNA markers and the trait. So once again, I don't know, can you hide this one somewhere? There should be a way of hiding this thing. This is summary is out down here. If you do the conventional breeding, by the time you make the first class, it may take 10 to 15 years to release a variety, which reaches to the farmers. But with the molecular breeding, it can take 3 to 7 years. So you virtually half the time, or even 1/3 the time taken to release a new variety, because you have the more efficient selection tools. And I will give you just some examples of our own examples of that thing. So here is the nature. Germ plasm sets, land races, and wild relatives of the crop plants. You take any species, any crops, all the genes are in the nature, in the germ plasm. Nobody, with all the development in science, nobody has created a gene de novo. We have copied the gene and synthesized in the nature, and did little bit of mutations also. But the genes have been evolved in nature, and they are there in the nature also. For all kinds of work, genes are there. And they are present in the germ plasm, traditional variety, land races, wild relatives. These are there. So first step is to do trait discovery. We have to do high throughput evaluation of the germ plasm. Most of the time, we have not evaluated our germ plasm thoroughly, because we need high throughput technique. So for example, more than 100,000 rice accessions are there in the MBPGR gene bank. Only now we have started evaluating, but still a lot to be done, because for every trait you have to evaluate, and then you can find a variety having a particular trait value. That is called trait discovery, or you can discover the donor, and then next step is the gene discovery. Because a germ plasm, a variety, a farmer's variety may have a good trait, but it may have hundreds of other traits which are not so good. So all the good genes are not in one place. They are scattered out in different varieties. The job of the breeders is to find that and combine them, put them in one place. So slowly and slowly, it is a very arduous task. It's not a simple task to identify a gene. That's why the cross breeding and selection is trying to take the genes from different varieties and put them in one, so that all the good things come in one. Just like the people also, somebody may be good at one thing, other may be good in another thing, but nobody is perfect and good in everything. So, but the same thing applies to variety also. So you want a variety which will be highest yield, it will have best quality, it will resist all the environmental stresses, but unfortunately, there is no such variety, and breeders are moving towards that. A long way to go. This is an infinitive goal, and the sky is the limit, but we are moving in that direction. Genomics is helping to speed up the process. That's how we can do it faster. So I think I have not finished with that slide yet. Can we go back? You are going forwards. So once we identify the gene, to cut the very long story short, for gene discovery, we need a structured population, not a random germ plasm set. That that the parents should be known to you how this is segregating. And then we use a series of functional genomics tools. There are so many tools are there, no time to describe. And then when we combine the two, the trait will segregate and the marker will also segregate. So this co-segregation analysis allows you to find out which particular DNA segment is associated with the trait, and that eventually leads to the map-based cloning of the genes. There are several examples how genes have been cloned like this. Once you know the gene, you can again go back to the gene germplasm and your varietal sex and land races and things, and you find the different versions of that gene. Allele, the definition of allele is the alternate version of the gene is the allele. So each gene has hundreds of alleles. Mendel only described 2 alleles, tall and dwarf, right, round and wrinkled, because at that time, at his time, he was ahead of his time, but from the comparing the present time, it was much behind. Now we know that there are several thousand genes in each species, and each gene has got hundreds of alleles. It's not only there are few genes with 2 alleles each. So the situation is much, much more complex. So you can do allele mining and find the best allele. Then you can do molecular breeding in real terms. So real accelerated breeding you can do when you go with the knowledge. Which gene you want to put together, you have a tag on that. Which background you want, you can select the whole background. I will give you an example. Then it goes to the society. So all society need right from the farmers to the consumer and in between the industry and also scientists. We are also part of the stakeholders. You know, we are developing better variety which a farmer will grow, then traders will trade seed and the products, they will do processing, consumer will eat. We are all part of the value chain and we are there. So the whole value chain, it goes to the society. And our role as a breeder is to find the good genes and then deploy them to in a better variety. Now, there are thousands of genes. And we targeted these 3 traits. The traits were discovered here at IRI in Division of Genetics with Dr. A.K. Singh, Dr. V.P. Singh. You can see one of the best variety, longest 1121. We don't know what are the gene here. We did not know at that time. Now we know. What makes very high grain number in some of these lines? This is green revolution variety Jaya. This is some of the new plant type which are more than twice the grain, and they are all very well filled. You know, it's not that when you increase the grain number, then they will be shriveled, and it is not always true. All the grains are very well filled also. And they are not in small size. So what is the gene behind that? What makes that? If you know that, we can make a variety which will have better quality, better yield, and it can have stress. This is salt tolerance. There is one variety which is tolerant to salinity, but this yield is very low, its quality is also low. So once we identify genes for this, for that, that is the breeding's goal, to develop variety which will have higher yield, better quality, and better resistance to environmental stresses. This is definition of plant breeding. And I'm just giving you one example of each which we focused 10 years back, 15 years back. It has taken 10 years to arrive at the genes which are responsible for that. And we are deploying in the breeding. Similarly, there are hundreds of genes, hundreds of traits are there. And many institutions are working, including ERI and all the advanced laboratory. So this particular high gain number gene, unfortunately, it is hidden here once again, chromosome 4. We identified the gene here on the long arm of chromosome 4. All these things have been published in peer-reviewed journals. And we identified this gene here, protein kinase domain containing gene, which is 5 times more expressed in the variety with the high gain number as compared to the normal variety. This is the nature has all this in this gene. We identified this and we try to deploy it in the breeding process. Now let us very briefly, sir, I will just talk about what are the molecular breeding strategies, different strategies. Dr. P.K. Gupta summarized, Dr. Prasanna also talked about that. Molecular breeding we have been hearing for a long time, but there are a number of approaches where molecular breeding can be used. And but one of the most successful for the time being has been the marker-assisted backcross breeding. I think more than 90% of the variety which has been released through molecular breeding are as a result of marker-assisted backcross breeding. And I will come to that, why it is so important, why it has been so successful. Although we can do mass also, pedigree breeding can also be done through marker, they are doing in the maize. You can take select a series of markers and you can select use for the selecting the lines having it. When you cross two varieties, they give a lot of different combinations. Typically, the breeding size, population size has to be bigger. Then only you can find the right combination. Then mass marker assisted recurrent selection in the cross pollinated crops. In the maize, it has been used. Then marker assisted backcross breeding. It is good for the open pollinated crops because here the variety is fixed. If it is lacking some trait, you know that the genes for that, you can within 2-3 backcrosses, you can transfer it. I will give the example for that. Then genomic selection, it is a new upcoming thing. We are talking a lot about that, but this is under the process now. You have to estimate the breeding value of each marker, and then you can deploy it in the selection. Then haplotype selection, again, new thing is coming. Instead of selecting for one marker, you select for the allele, that is a set of markers which define a gene or define a genomic region. So you can select that. So these are new things. Marker assisted breeding has been effective. One thing in this, sir, I think, and Dr. Kasingh is not here, but quite often he has asked this question, that no matter which tool we use for selection, either visual or any excellent tool like genomic selection, it depends on your breeding population. If your breeding population is only 2,000 individuals, if it does not have the right combination, whatever tool you use, you cannot select the right material. And I can tell you what is the science of combination. If you can move this bar somewhere up or down, can you move it, this black bar from the side? Move it up or down, this calculation I showed you here. I have just enumerated the rice genome has got 37,000 genes. Pigeon pea has got 47,000 genes. Human have 25,000 genes, but their alternate splicing, etc., made many more projects. Wheat has got 1,25,000 genes. I have taken very simple thing here. In the Mendelian fashion, the gene will segregate. If the genes are in different chromosomes, or they are not linked, there may be one gene on one chromosome arm, the other one on the other chromosome arm, then also they will freely recombine. So I have taken simple here. In the rice, there are 12 chromosomes, and you take each chromosome 2 genes, one on the short arm, one on the long arm. So how many genes becomes? 24 genes. When you cross two varieties, each 24 genes will have 2 alleles. You can't have more than 2 alleles because in the sexually propagated thing, bisexual, even there are multiple alleles. In any crossing, you can employ only 2 genes at a time. Even in hybrids, you can employ only 2 genes at a time. Now, when you cross two varieties differing only for 24 genes, mind you that it has got 40,000 genes, but we are talking only, making it simple, only 24 genes with 2 alleles each. How many combinations of alleles are possible? 2 raised to the power 24. So 16 million different combinations of alleles will be there. And only one of them will be identical to the one of the parents. Suppose you take your best variety like Swarna or Samba Masuri, cross with Samban. So only one of the 16 million alleles will be similar to that Swarna, which is your golden variety like Sachin Tendulkar, MS Swaminathan, or Lata Mangeshkar. The rare combination of genes, rare assortment of genes. Once you disturb the apple cart to restore it, you can't do it unless you do back crossing. Back crossing is such a wonderful tool. Just by one back crossing, this 16 million becomes 8 million. Second back crossing, again 4 million reduced without knowing about this thing. So undesirable combinations you can eliminate through back crossing. So whenever we start with a good, very highly adapted variety, that's why most of the breeders, they will cross elite with the elite, so that you don't have a lot of undesirable things. And whenever we try to transfer resistance from a wild relative or very weak performing things, invariably you will be doing back crossing, 1 or 2 back crosses. Why? You want to eliminate this, 16 million. You don't want to waste your time, trying to understand what are those things. But as a process, so that's why marker-assisted breeding, back-cross breeding has been successful, because we have fortified the best of our variety. Next slide, please. किधर पॉइंट करने पे चेंज होगा? साइड चेंज कीजिए. नेक्स्ट.
So see what Green Revolution did actually, we all know that they developed a set of varieties which were very highly adapted, very highly productive. They were high yielding. They were bred actually for the high input condition. That's why when the criticism comes that Green Revolution has done a harm, because we had to apply a lot of nitrogen, a lot of irrigation, all those things. Because that time selecting for the yield was the prime target to improve the productivity. And because these varieties were selected for high input condition, they don't have those traits which are required for the marginal conditions. They were not selected for quality. That's why we lost the quality. They were not selected for drought prone areas. So that's why they don't have the drought tolerance. Not because deliberately we have tried to exclude those things. But now we know that they were selected for the high input condition. We give them optimum dose of fertilizer, nitrogen, all those things. So we selected lines like Swarna, Samba, Masturi, IR64, MTU 1010, which made a difference that we stopped importing food and now we are exporting. So the Green Revolution has played that role. But whatever was lost during Green Revolution, now we can restore it. Now we have the science to restore it back. And I will give you some examples here. Through marker-assisted backcross breeding, through nothing else. So you can see here, mega varieties are called. These varieties are grown in millions of hectares. Even after 40 years of their release, even your extension worker will tell that don't continue this variety. I have released a new golden variety. Use this one, don't use old one. In front of me, farmers has tell the extension worker कि सर आप बता रहे हैं कि ये बहुत अच्छी है, लेकिन मैं जानता हूँ मेरे लिए पुरानी वाली ही अच्छी है, मैं उसी को लगाऊंगा. मेरे सामने बोला है, क्यों बोला है? तो हम क्यों इंसिस्ट करते हैं कि नहीं इसको हटाओ, ये वाला यूज करो? Because we want to promote our variety, that's why we tell that. Varietal replacement, so called. But why don't we replace the variety itself, the variety which farmer likes? We see what is lacking in this, and if you can go back to traditional and fortify it, then they will start liking. And variety like Swarna, Sub-1 and things are typical examples of that. So I will just skip this one. This is the Green Revolution high yielding variety. They lack the climate resilience. They lack the quality. That is fact. When people are pointing out, that is fact. But they have the high productivity. That is also a fact. If that was not there, we will be begging for food from around the world. But now we know that where are the genes? They are in the wild relatives, in the traditional land races. The genes are there. If you can find it and put it back, that is the game. See one of the examples here. And Nirja is sitting there. She actually pioneered this work while working at Erie. You see, this is FR13A. This is a land race of rice. And it was identified in 1970. Flood resistant 13A from Orissa. It took 20 years to develop breeding lines and do some mapping work. Never saw light of the day in the form of a variety. When the rice genome was published, very quickly the gene underlying a single gene, just like SD1 gene for the Green Revolution. This flood tolerance was governed by a single gene, Sub-1, it was cloned, and within 3 years, she transferred in Swarna and released a variety Swarna Sub-1. Now the same Swarna, which will die in just 2-3 days of complete submergence, it can tolerate 2 weeks of complete submergence. And most of the floods, they don't last more than 3, 4, or 5 days. After that, the flood recedes. You will get 2 to 4 tons per hectare, whereas you will get 0 yield in the original Swarna. So this is the typical example where how this Green Revolution variety has been added a single gene here. And this is the marker-assisted backcross breeding. You have marker for selecting the foreground, and you have the markers for selecting the background. And within the three backcrosses, you can restore a variety like original Swarna, except it has got one more added feature. Now it is flood-proof Swarna. Here, they did the same approach to transfer in several mega varieties like IR64, BR11, all those things. This picture is used by many people. It is from Erie. See, without the Sub-1 gene, they will die. But with the Sub-1 gene, they will survive and eat. One gene can make the difference, just like one gene made the difference for Green Revolution, in wheat also and in rice also. Now we developed tool in our laboratory for background selection. for genetic analysis, for QTL mapping, high density fingerprinting. We have developed high density arrays like 50K SMP chip in rice. And there's a story behind this also. We were forced to develop this chip because Cornell University will not give their chip to us for using here. They rather say that you send your germplasm, we will do it there. So our graduate students and we got together and developed this chip, which is better than Cornell chips in many respects. It is patented. It is gene-based chip. It has got multiple SNPs per gene. There are several features have been patented, which are better than any available chip. It's a calling rate and all those things are much superior. I'm not going to go into detail here. It is based on the single copy genes, but also agronomically important genes have been. When we applied this gene for the background selection in the lines developed by Erie, we found that when you do the background selection with the low density marker, like SSR marker, maybe 2 or 3 markers or 4 markers per chromosome, then you might think that you have recovered the background. But when you apply the high density chip, then you can see this Swarna sub1 has 97% background recovery, but Samba sub1 has only 79% background recovery. So there is still some variation, which you can, recover more like Samba Masuri. And why do we need to recover? I gave you the logic. Because Samba Masuri, Swarna, Aya 64, they are like Sachin Tendulkar, Lata Mangeshkar, MS Swaminathan. They are rare combination of assortment of alleles. Once you disturb that, then you will lose something. So you just add what is not there, but retain that whole thing. Of course, not to say that we don't need to make another Sachin Tendulkar. So we keep on doing that, breeding for new varieties. But meanwhile, just correcting the defects of the what is lacking in the mega variety, it is paying dividend much faster than that. So we developed a program with the help of DBT. This is major funding, 17 crores for 5 years, then another 10 crores. And there are 15 different institutions, Agriculture University, ICR institution, and our institute playing the coordinating role. You can see here mostly the eastern part of the India where it's the rain-fed rice. It depends on the mother nature. So you have the drought also, you have the flood also, you have the coastal salinity also. So it was a very relevant program with the help of the Erie. We developed it and we published a concept paper that within 2 to 3 back classes, of course, it is inspired by the Erie's work, what Nirja did there and the Bill Gates Foundation, they were funding the stress tolerance rice. But the key here was through networking. I alone could not have done this thing, and any of the partner institute also could not have done alone. So with the coordinated network, people who are working in a particular niche area for a particular trade, and those who have expertise in genomics, if they join hand with this kind of funding, I will show you what the results we have got. So we took 7 QTLs for drought tolerance, 1 for submergence tolerance, 1 for salt tolerance. And we mobilized them into these high yielding background, not Swarna IR64, but the locally adapted variety also. So what we did at NIPB here, we standardized these markers, the peak marker like RM21, which is linked with this particular QTL 2.1, which gives drought tolerance. You just mind that we don't know the gene yet, but what we know that so we standardized these markers for foreground selection. We broke the linkage between drought tolerance genes and also the dwarf height, because when we developed the dwarf varieties, we got this susceptibility allele here because they are very tightly linked with that. This is called linkage drag. So when you select for the dwarf rice, you inadvertently select for the sensitive rice. But this linkage was broken because of the knowledge of genomics. So now we have this, see the original Swarna here dies, but this dwarf Swarna, which has recombined that region, we restored the original allele from M22 and thing, this survives. And this is a summary here. In this program, we have already released and notified 11 varieties. These are the stress tolerant version of this DRR DAN 50, released by DRR here. It has got 2 QTLs for drought tolerance and submergence. So it's 2-in-1 submergence and drought tolerance. Similarly, many of these regional varieties like Ranjit Sub1, IR64, DTY1, it has now reached the farmer. This was released in 2018, and breeder seed indent is in good demand for this one. Similarly, there are more than 6 are in the pipeline. So this is very fruitful applications of genomics to develop varieties for the society. Now the gene which we identified by high gain number, we have transferred using same approach to 12 different backgrounds, and in each background, this is background selected, this is the foreground in the red. Then you can see in each background it increases the grain number, the pancake size increases. I will give you example here in Samba Masuri. This is the original Samba Masuri. If you just add one that curious, the number of grain becomes more than twice. The tiller number is slightly reduced. This is the panicule size you can see has increased. And in the 4 years of station trial, it is 15% higher yielding than original Samba Masuri. So this is actually traditional backcast breeding supported by genomics. And this is now getting under evaluation at the ECRIP trial, and Dr. Sundaram is here. He is very hopeful that it will perform better. Now search for new genes. This is the great Dr. B.P. Paul, after which this is symposium in this particular hall is named. He is the Dian of Indian agriculture. And he wrote the article search for new genes. The genes are in the germ plot. So I went around the country to collect the wild rice from its natural habitats. These are the abundant habitats where it is found in abundant. There are habitats where it is rare found in the upland areas. This is throughout India I have gone. This is the coastal salinity, sodic soil, drought, flooding. The wild rice grows everywhere. And that means it must have genes for the survival into those conditions. Then it is threatened by development. We talk about the development versus nature. This is wild rice growing here, but the home is getting built, a school is getting built, industrial park is there, road is getting widened. So this is its natural habitat which is getting destroyed, and we are losing it at alarming rate. We have collected 1,000 of them, developed a database. I am skipping a lot of these slides because through our genomic study, we disproved a Chinese claim that all rice originated in China. They started claiming that earlier it was not, earlier it was a polyphyletic origin, but they started claiming through genomic data, publishing in Nature. So if something is published in Nature, don't go that it is going to be correct only. It can be misleading also. Then we published that our Indian wild rice actually has originated for Indian wild rice. Our cultivated rice, Aus and Indica type, they have originated from our wild rice. So it is a polyphyletic origin. And there are alleles which can give white pericarp color, which are unique to our germ plant. Then another paper came in Nature Plants from the UK workers, reanalyzing that China data, and then proving that it has 3 independent origins. Same data. So you can be biased and then conclude something according to your things. So the wild rice has been evaluated for drought, for flooding, for salinity tolerance. Allele mining we have done. Time is short, so I'm not going. I'm just giving you one example that one of the lines which are tolerant to salinity, we have developed population and we have mapped it through using this gene chip. And we found these alleles in chromosome 1 and chromosome 11, which make them salt tolerant. So this is being backcast into IR64. So once again, it can contribute to developing stress tolerant variety. Underlying genes are also we can scan using the genomic knowledge. Then in pigeon pea, mango, and jute, I think I have prepared according to 1 hour, but I will skip now because he has to present one more topic is to go. We took pigeon pea, mango, and jute because it is important for India only. Jute is important for Bangladesh also, but there are several crops and several traditional, medicinal and plants and the fruit trees which are important unique only for the India. Like jamun is only in India, haldi, all those things, and they have great value. $10,000 US worth of mango is produced, and only in the season it gets lost. So having genomic knowledge will help in the same way as it helped in the rice. Rice is one of the example. The same thing can be repeated in every species of importance. So that's why I spend more time in the rice. We developed the draft genome of pigeon pea. This is the first genome sequence entirely in India and first pulse genome sequence in the whole world. Then pigeon pea also can have the same story. We have the dwarf lines. We can identify the gene. We can develop the variety where farmers can do the operations, mechanical harvesting and all those things. 3-4 varieties have already been released. The early versions, just like rice and wheat, we have the early dwarf varieties were not so high yielding if you compare with today's version. So this is the green revolution is making for pulses. Dwarf pulses is just a knee height. It will produce the same yield as the taller than knee. There already IRI is having trials for them. These major genes are there for those traits. Then we have developed chip for pigeon pea also. In jute, again, jute is very important for India, and we have selected the best variety for decoding. We have already decoded it. Here, JR524, in collaboration with the Jute Institute. So collaboration is key here. If you work in our silo, we cannot make the impact. So we have to join hand and harness our synergy. Then mango, you can see more than 1,000 variety. Mango has originated in India. And this is a history, this is not science. In historical time, mango has gone from India to China. Lychee has come from China to India. That is also recorded, but mango has gone from India to China. So the truth is sacrosanct. We should not claim something which is not true. Then mango has a very typical problem of high heterozygosity. But this new third generation technology where PCR is also not required, you just simply isolate the DNA and you can sequence. Even PCR amplification is not required. You remember in the first generation, you need to clone the DNA. Second generation, you need to PCR amplify and sequence. Now the third generation, just isolate the DNA and sequence. No amplification. You can take one piece of DNA, one polymerase and DNTP, and you can read which base is incorporated. Now this slide is very important, sir. The mango slide which I've been taking, which I shared with the Prasanna, because we talk about heterosis and hybrid vigor, all those things we talk about a lot. The basis of heterosis, what is the basis of heterosis? What is the dominance theory, over dominance theory, or dominance into dominance or dominance into additive? When this new information came to our knowledge here by Amrapali, Amrapali is a hybrid of Dasari and Neelam. So when we sequence Amrapali, heterozygosity did not allow us to construct the genome of Amrapali because the maternal and paternal were assembling separately. And which part they are similar, they are assembling together, which part they are different, they are assembling separately. So this new technique called trio-binning, when you have both the parents and the progeny, you can separate out the chromosomes of Amrapali into what came from Dasari, what came from the Neelam. So now we have Amrapali has 2 genomes. 20 chromosomes coming from Dasari and 20 chromosomes coming from Neelam. This is chromosome 1 selected, chromosome 20 tak hai. The crux of the unique thing about this is that Dasari has got 36,000 genes and 9,600 of that, about 25% of genes are unique to Dasari, which is not present in Neelam. Similarly, Neelam has got similar number of genes and 9,000 plus are unique to Neelam. So what Dasari has, each parent has contributing only 36,000 genes because 9,000 genes are unique in each. So Amrapali turns out to have 45,000 genes. So that is the hybrid advantage. Substantial gain in the number of genes. This is the present substance variation. And this is the new data which we have got. We are preparing the publication. And this gives one of the basis of the heterosis. People have talked about this thing, but this is experimental evidence that this is one of the major contributors. And the dominance, also at the molecular level, there is no dominance. Molecular, everything is co-dominant only. And 4 of the 7 genes of Mendel's has been cloned. And the recessive allele is nothing but the inactive allele of the functional gene. So recessive allele is that knockdown genes or destroyed genes is recessive. Now we have done all this phylogenetic. The mango is closely related to kaju here. If you have seen the mango seed and the kaju seed comes outside the fruit. Mango seed is inside the fruit, but their shape is same. You can eat kaju, but you cannot eat mango. I think if you know what is the difference, you can make the mango seed edible. Kaju seed also gets treated before eating, but mango seed much bigger than kaju seed. They look very similar in size. If somebody has seen the mango के गुठली में से जो निकलता है उसका शेप काजू जैसा ही होता है, तो उसको हम अगर नॉक डाउन कर दें जो टॉक्सिक कंपोनेंट, you can eat it. Now through we developed chip for mango also, 80K chip this time, and we have identified, we have fingerprinted 384 variety. We can fingerprint all the 1,000 varieties of mango. If anybody steals our mango, they have to pay. We can prove it that it is ours, they have stolen. And several states we have done GWAS. I'm showing you only one here. Fruit acidity. Acidity index means what makes mango sour. Similarly, TSS, what makes mango sweet. And size, pulp percentage, all these things, many states we have mapped. This is a very high large score here. Then very quickly skip to Varaja coarctata. Halophytic rice has been sequenced. Coconut has been sequenced. This is all Indian work through collaboration with our institute. Then tea genome has been sequenced. Reference genome of guar has been prepared. Then chickpea rhizobium, if you want to reduce, nitrogen. Then draft genome of wheat leaf rust. This genome information also will be important in genome editing if you want to reduce the off-target effect. You have to design a guide RNA which targets only a particular target. Then other institute like chickpea at NIPGR, NEEM at IBAB Bangalore, and Tulsi at CIMAP and Bangalore, and Haldi, once again one of the ISR institutes. These are getting sequenced. This is my last slide here, last but one. Bio-Earth genome, Dr. P.K. Gupta talked about yesterday. We had planned to sequence 1,000 genomes, 500 plant, 500 animal, but unfortunately, the funding did not come for that. Funding had died out for some reason. I think the investment in science R&D, I can tell from this platform that there is a need to increase investment in R&D of science. Only less than 0.4% of the GDP goes for scientific research. And 90% of the budget goes only in the salary. If you don't have the budget to work, then why pay salary to people? It is a waste of time, waste of money to pay salary. So the budget for working capitals must be increased because it pays dividend. And then the challenges is there, sir. I will share this slide with you. This is summary in the summary form that we have to conserve the germ plant. We have to choose the plants for economic importance for sequencing. Then we have to go through the whole thing, just what I described. For every other species which is important for us, we decode the genome, we conserve the germ plant, we mine the genes, markers, we deploy for developing better genotypes, both for yield, quality, and the resistance. Using the genomics, of course, the tools. And then, in the end, thank you all very much for your attention. And this is really a memorable event for all of us to commemorate the birth bicentenary of Gregor Mendel. And somebody said that the similar thing we should do for the many Indian scientists also. I shared in the Facebook and many people commenting that we should do this kind of honor to our great Indian legends also, so that we start respecting the science in the society, because science delivers to the society. Scientists work hard. Before this, a medical scientist was there. So we should have that passion. And sir, I will just, I just want to quote Kabir Das that in Gulat in Panya, Gare Pani Packs. So Mendel ने कितनी मेहनत की थी, 50 साल तक इग्नोर किया गया. Many people, average was ignored. So many people, you may have complained that people are ignoring my work, but you don't have to worry about that. You should be satisfied because you are doing good thing. Then you don't worry whether you are ignoring, because after 50 years also, if the truth is there, real satisfaction should be yourself. You should be satisfied with what you are doing. It's for your good, for the good of the humanity, and that should satisfy you, not because who is accepting you or ignoring you. That is not the thing. With this, I would like to thank because I have taken more time and one more presentation. Thank you all very much for your attention.
Please give within time to nice presentation. Wonderful presentation. Here we have seen science and its implication, applied implication. Not only doing very good basic science research, but how this has helped in development of the number of varieties which Dr. Singh has mentioned. So I'm sure there may be number of queries. Of course, we are short of time, but still we can take few questions if there are any. Yes, please. Can you give the mic there, please?
Please turn off your mics.
बोल भाई बनते तभी बोल रही थी अभी आराम करते खाते फिर से वहां दौड़ जाना था दौड़ना ये पता है अब अच्छा थकावट मुझे थोड़ा सा ही तो देना है
Yes, I think you are very much right. NBPGR, I think Dr. Rakesh Singh has used molecular markers to develop the core in case of rice. And he found that when evaluated in the field, that core is much better than the core developed through the morphological markers, because the morphological markers are only a small set of genes which are only visible. But when we use the genome-wide markers, we can develop a better core. And for the parent genome, we don't have to sequence, 3,000 or 5,000. If you select the really good core, that will cover the large number of entire 99%. So this is a very good question. I think we should adopt that thing.
Any other queries, questions? Students, I think.
Good afternoon, sir. Thank you so much, sir, for this kind of fruitful session, sir. I have two questions, sir. The first question is, like, if we want to sequence a large genome. So, as you mentioned in your presentation, sir, there are some few methods like 454, Illumina, Sanger methods. So, in terms of that, like if we focus on two objectives, like first is time management and second one is qualitative results. So which method is, as per your experience, is the best method for future generations as for now time? This is my first question, sir. And the second question is like, if we want to sequence a genome and we perfectly sequence up a genome in a perfect manner, but later we want to manipulate a codon by which we can manipulate a protein also. So what will be the changes in a genome history we will get? It is possible to we get a trait which is never be discovered. It is kindly possible that what kind of result we obtain by the manipulation of that codon. So that is my query, sir.
Okay, your first question is which technology, which platform we should use for sequencing large genome. So in present day, because technology is evolving all the time, in present day, the PacBio seems to be on the top because it gives you long read and also the high base quality. BioNano also gives you Nanopore, Oxford Nanopore also gives you large reads, but the base errors are a little bit more, but they are refining. So these two, we have to go for long read single molecule chemistry and then supported by Illumina. And second thing, that gene editing, genome modification you want to do, why you want to go for a trait which is not there? We don't need those traits which are not there. Just we need to improve the traits which are needed, which are there. just like the yield, quality, and resistance. And definitely you can do gene editing now. Government has allowed it that if you do the minimum gene editing, which is equivalent to the normal mutation, you can do a much faster thing, and the SOP is being developed. So you can get the desired result through gene editing without disturbing the genome too much and without introducing any foreign gene. In fact, gene editing can be a substitute for backcrossing. You can just take the allele and replace it. You don't even have linkage drag. So in future, I predict that gene editing will replace the backcrossing. If you find, say, 20 good genes within rice itself, if you do through recombination breeding crossing, it will take a long time. In the one step, you can just replace it. You just can put them all together in one background. So these are things for the future.
Thank you so much. Thank you so much.
Okay, sir.
Okay, thank you very much. Dr. Singh, for a very nice presentation. We must give a big hand to him. Now we move on to the last lecture in this session. And I invite Dr. Velu Govindan from CIMIC. The role of wheat genetics and breeding in mitigating global hunger.
Good afternoon, everyone. Thanks to Chairman Dr. Abhi Singh, and also special thanks to the organizers for giving me this opportunity to share some of my experience in wheat breeding and genetics. So last couple of days, we have learned, I have also good opportunity to learn a lot of historical things about Mendelian inheritance. So I'm not going to talk too much about the basic stuff, but how we are going to utilize the Mendelian genetics as well as the modern tools to accelerate the genetic gains in wheat to mitigate the global hunger and as well as malnutrition. So just to brief about the importance of wheat. So wheat is one of the important staple crop, produced more than 780 million metric tons every year. And this provides about 20% protein and dietary energy for almost all a whole population around the world. So this is worth of more than $50 billion trade every year. So just to give some, a lot of school children's, kids are here, how the breeding has started. So just to give some background, the oiled emmer wheat is, originated about 10,000 years back. And then you can see a lot of, during this time, there is no breeding, the human just started cultivating crops. So and then it moved on to other crops like maize, and all goes back to history. And after the Mendelian inheritance, so a lot of things has improved, like cross breeding started in 1900s, and then hybrids, mutations, and tissue culture. Recently, now we are in a genomic selection approach. So just to give an example of green revolutions and the major impact of the RHT dwarfing genes in wheat. So the quantum yield jumps happen because of those RHT genes, which was introduced into the tall varieties. So you can see this was the original cross was made by Dr. Borlaug when he was in Mexico. So when Dr. Borlaug came to Mexico, there was no CIMIT. So that time he was outside CIMIT, funded by the US government, who was doing the breeding work. So this was the tall varieties, and this is the base, source for the Norin 10, which was crossed to the tall varieties. And then it became the, the green revolution varieties. It can be Sonalika, Kalensona, other varieties. So that led to green revolution in India, also other countries, Pakistan and other, Turkey and many countries. After that, there's slight incremental increase in terms of yield potential. And Dr. Rajaram, he took the breeding program in 70s, and then the full-fledged breeding program led to a lot of yield gains in the modern germplasm, utilizing some of the winter by spring process and also 1B, 1R translocation, which led to famous varieties like PBW343, we call Attila Simit, and there are Babax and other VD type materials. And I would like to emphasize few examples of some of the major genes which revolutionized the Simit wheat breeding program. Thereby, many varieties came and released in many countries. So this 1B, 1R translocation, you can see here, this is the fluorescent, this is 1R translocations. So this was one of the fantastic work which led to, a lot of genes. This was originally introduced for resistance, but which led to, you can see the root depth. So this has longer root, which means it can take more water from the subsurface, and also resistance to several diseases like leaf rust and stripe rust and other things. So at one point of time, at Simit germplasm, we have more than 60% of the material with 1B, 1R translocation. But this is the trend now. So recently we made some genetic analysis. So it's almost at the at the stage of extinction because of it linked to the some bad quality traits, as well as the UG99, the SR31, breakdown of SR31, which is the well-known, durable gene for stem rest, which was holding for many years. So that broke down in late 2000, early 2000. So also some of the other genes like translocations from the wild relatives, Aegilops ventricosa, we call VPN segment. This has been introduced from wild relatives into the cultivated wheat. But you can see now more than 95% of our material has this gene. So why this has happened, So basically this was a bit to introduce, I think, powdery mildew something, but it also linked to YR17. So still this gene is effective in many countries, and India, there are some, virulent races against this. And also recently the wheat blast, which was identified in Bangladesh. So this gene is really good source for wheat blast resistance. So that's why, and also in addition to that, the yield gains. So this particular translocation, it has more than 6% yield gain across different environments. So that's why this frequency of this particular gene is more prevalent in Simit germplasm. So here I just want to show the GWAS study on this particular segment here. It is in 2 areas, 2 areas chromosomes. You can see this particular gene can contribute more than 480 kg per hectare yield gain. So that's why the frequency of this particular gene is very high in our material. But also another area, Simit has done, a wonderful job in terms of rust resistance. So we have been, we have very good success stories breeding for adult plant type resistance or slow rusting resistant genes, not the race specific. So starting from LR34, these are like paleotrophic genes for other diseases as well, stem rest or yellow rust, as well as LR46, which is almost kind of fixed in our material. This is also linked to a phenotypic trait we call leaf tick necrosis. You can see here, we can easily able to identify whether this particular line or plant, they have this particular gene. So these are some of the, for the young breeders, if you have a particular like molecular marker, we should really go for that, not just look for fancy stuff, but also we should think of some molecular, phenotypic markers like this, which can enable to increase, frequency of during selection process. And also there are other genes, but in the concept of the minor gene based resistance is, if you have multiple genes, if you can pyramid into same background, so we can be able to get near immune type of resistance over in our germplasm. So this will avoid, the boom and bust cycle of rust. So that was the more problem in the, the Western countries. So they just focus on the race specific genes. So here also, just to highlight the leaf rust, I would say the leaf rust is not a problem anymore. If any of the breeders want to use the cement materials, because we have a good source of resistant lines. Same story for yellow rust. So, but sometimes these APR genes fail to cope up with the early epidemic of yellow rust. So that's why we need to combine with some major genes. Some of the major genes or race-specific genes like YR48, YR54, or YR60, they are still good for yellow rust. And also in the case of stem rust, we have made a lot of progress over the last 20 years after the breakdown of ISA 31. So many good resistant materials has been developed through our shuttle breeding scheme. So as Dr. Kasingh was mentioning about the accelerating the genetic gains. So now we have started using the speed breeding. It's nothing but rapid generation advance. We have small facility in CIMID. Using the special lights, we can able to get 3 to 4 cycles per year. Basically, we use this facility to introgress some known genes with molecular markers. So by backcross breeding. So this is, for instance, if you have our famous variety we call Borlaug 100, it's not so good for yellow rust. So we use this facility to quickly introduce another yellow rust genes or stem rust or any other disease resistant genes. This is functioning from 2020. So we have named this station after Dr. Sanjay Rajaram, who was passed away last year because of COVID. So our breeding focus is, the primary focus is yield and yield stability and durable disease resistance, and also the end use quality and nutritional quality and water use efficiency. So one of the success stories or the impact of cement material germplasm is because of the shuttle breeding scheme, which was introduced by Dr. Borlaug. So in many countries, wheat can be grown in one crop season. Here we have full crop seasons in Mexico. One is in the northwest of Mexico. Here we have the winter cycle in the central Mexico. In the highlands, we have summer season. So we have full cycles where we apply the selection pressure and advance the segregating populations. But after the UG99, so we also expanded our shuttle breeding with Kenya, from Mexico to Kenya shuttle breeding. This also broadened our material base, so which led to widely adopted germplasm with disease resistance and other essential traits. So what are the new approach to get the yield gains in our new materials? So how we are going to maximizing the probability of getting the rare transgressive segregants. So there are so many approaches we can able to do that. So we are modernizing our breeding program, starting from optimizing our crossing strategy and also the selection scheme, as well as the large number of stage one, first year stage one lines tested in different selection environments. Let me go through one by one. So selection method is really important. Based on our genetic principles, we decide what will be the selection method which will be used to advance for making better genetic gains. Here we use mostly selected bulk during our early generations, which is very cost effective. We bulk and we advance until F3 or F4. But when it goes to later generations, we go for expanded testing across different environments by creating or by simulating different target population environments, either it be South Asia or Africa. So we can able to create different kind of environments by regulating water, as well as planting early or late, as well as the planting method. For instance, wheat is planted in flat conditions, but we have all the trials in the raised bed, but also we have an environment under the flatbed system. So this will give kind of robust phenotypic data, which will be useful to identify the best performers, and we can distribute to the national program partners. in the different countries. Here I would like to show the yield gains over the last 5 years time. So you can see this green one is in 2014 and in the blue one is in 2019. So almost about 6 to 7% gain happened over the, this is based on the population mean, all the lines, on average of all the lines. So on average about 1% gain making, this is not so big, but still with the large genome and with a lot of challenges like climate change as well as diseases, this is still promising. But we have different, I think, approaches to increase the accelerate the genetic gains. What we call the predictive breeding. So it's mostly based on the genomics as well as using some new high throughput phenotyping techniques. We can able to accelerate the genetic gains. Here we have a small project with the University of Queensland. So trying to get the artificial intelligence based models to design the best process using all our genomic data. We can able to design our process. Once the processes are made, we can advance those segregating population under the speed breeding facilities. So then we can quickly deploy into the target population environments. This is really interesting. And one of the implication will be what will be the accuracy versus speed. We are moving very fast, but we are not sure some of those lines which are good or it's not robust data to make that decision. So as we all learned, the quantitative genetics, the population improvement, every cycle we make gains in the right tails, the red ones. So by doing so, this can be achieved, either it can be a single trait or a combination of traits. We can reduce the time between cycles, as I mentioned, through speed breeding, and we can select parents based on the first year yield trial lines. And we have now more than 4,000 to 5,000 lines in the first year stage testing. And also we can have more greater accuracy and selection intensity for the traits of interest. And also we need to consider if you want to make longer gains, so we need to have enough diversity in our the closed population. So this is the new breeding scheme we are applying. We call rapid bulk generation advancement, also the rapid cycle recurrent selections. I'll just quickly go through. So the traditional shuttle breeding will take 4 to 5 years to get the fixed lines, but in this case we will try to get within 3 years. And in this case, still we can able to get 2-year cycles. This is quite fast, but still we are in the preliminary stage. We need to compare the performance of the lines derived from this scheme versus the traditional shuttle breeding scheme. I don't want to go in detail. So this is the scheme, how we advance, how we made the process inside the screen house. We make all the process in the screen house. We advance F1, F2 in the screen house. Now and then we take the F3s to the field and get selected, and then we advance to the yield trial stage. And also we are trying the 3 generations per year field advancement. It is much low cost. We don't have to have special lights, no infrastructure. So what we do, we just plant 2 seasons in Obregon just by adjusting the time. Immediately after crossing in the highlands, by October, we plant the first F1s. We harvest the F1s by February. We plant in March, and we get the F2s within 3 months. So this is where we are advancing. This is all field-based advancement scheme. And also traditionally we are extracting lines from F4 or F5, even later generations. So now we have started extracting lines from F3s. So there are still a lot of debates, a lot of heterozygosity, and still alleles are getting fixed. But we are now moving toward this directions. And last year we had about 100,000 plots in Obregon, which were selected for agronomic type disease resistance. And out of 100,000, we came up with 18,000 lines, which is being grown now in summer season in the highlands, which are being genotyped. We will evaluate for diseases, and then immediately it will go to the yield trials in this December sowing. So this is so quick. recycling strategy we are now following. So this is the slide from one of our colleague, Famine. She is based in in Louisiana. You can see the large diversity of CIMIT material for the last. This has been genotyped. This includes the cohorts of lines from 4 to 5 years. Consist of 62,000 lines. This is one of the largest genotypic data have in the public domain for wheat breeding. Again, there are some QTL regions for grain yield. That's not so stable, but because of yield itself, it's a complex trade, so we can't expect stable QTLs. But perhaps there are some regions always is showing good high yield potential. And this is one of the interesting slides from her also. So there are how the favorable alleles getting fixed over the years. Here in the x-axis, you can see the year. There are different QTL regions from 1A, 1B, and 2A, 2AB regions. For example, the 2A, which is I was mentioning before, the 2A is here. The frequency has gone up from the almost 0 to 90%. So this is because of the philotropic effect of that particular gene. So this is also what I was mentioning, like the stage 1 of the yield trial, the first year yield trial, we grow in only one environment before. So now we have changed our trial design. So we just go for single rep augmented design. The big trial, you can see all these colors are checks, which are distributed nicely to able to adjust the spatial variation. So here we have 4 different environments to capture all the genotype by environment interaction. By considering these all the data from 4 environments, we select the lines as candidates, which will be advanced. So last part of my talk on the biofortification, because last 12 or 13 years I have been working on this project on improving the micronutrient, especially zinc and iron in wheat. So about 2 billion people are malnourished. If you see the base levels of zinc in major commercial varieties, either it can be India or Pakistan, in many countries about 25 to 28 ppm, and also same for iron. So the target we put like about 37 to 40 ppm, so which will be considered as biofortified. So there are a lot of dietary supplements. I mean, the food which is not affordable by resource poor farmers or consumers. So this is one way, best way of delivering the essential nutrients, micronutrients to the target population. Here I would like to show here in the left side, you can see the bright color shows the zinc localization of zinc in the mostly in the crease and the embryo, whereas in this normal varieties is not that promising zinc. So you can see the most of the zinc are going into this part. So that's why we encourage people to consume whole grain atta chapatis like that. So this is the pipeline. So for this particular project, we make about 400 to 500 cross, and half of them will be back cross because of we use unadapted material, high zinc lines. So we back cross with the elite lines because we use only one single back cross approach, and then we select for agronomic type in the early generations, and then we go for large plots, yield trial plots, and then we identify lines, and then we share with partners with high zinc. This is some of the examples from the gene bank all the way to farmers field within 7 to 8, 10 years. This is one of the success stories. We can able to deliver from the, you can see how wild they are and how they become varieties with extra additional traits. So through, I think, targeted breeding and looking for the large transgressive segregant. So that led to some of these varieties. And also we put a lot of effort on identifying some QTL regions because of this is phenotyping. Sometimes zinc is not that easy. So it requires, cost and resources. So we did identify some of the QTLs through biparental as well as association mapping studies also identified some of the QTL regions. And also the genomic prediction accuracies are quite good, based on the different statistical models. Using this is a cross validation model. We use the same set or take off of them as training and then predict off of them. So still the correlation was quite high for these traits. So these are some of the candidate regions for zinc, and also we have paleotraffic genes associated with these. For instance, in chromosome 1b, the 1000 kernel weight also linked to the zinc. So there are so many QTL regions. So now we have handful of CASP markers we are also using in the markerized pipeline. Recently, we published this paper, just came out. So we analyzed the international data from several partners, thanks to a lot of partners in India. So we analyzed this data for last 11 years. We could able to see across locations about 1.5% yield gain, as well as it is translated into about 109 kg per hectare gain with the Hatch PYT trial. Also, by maintaining the zinc, the minimum zinc levels of about 6 ppm extra additional in addition to the checks. So based on this success, we got a big grant from Gates Foundation a few years back towards mainstreaming high zinc in all the elite materials. So instead of doing the targeted breeding, if we can deploy these QTLs or genes into across all our elite germplasm, we don't have to have the separate pipeline. So you can see the heritability and all the variance components is quite high for zinc. So this is just kind of encouraging for us, to deal with this trait. So now we have made so many crosses based on the genotypic, estimated breeding values considering high yield and high zinc. So we are expecting within next 4 or 5 years we can able to sift the whole population, all our summit lines with zinc enriched. So this was happened because of the rapid, non-destructive analysis, which we call XRF. So we can able to measure thousands of samples. Every day we can able to measure 200, 300 samples with no cost, almost only the equipment cost. And also we have now optimizing some handheld devices for phenotyping even before harvesting. So we should not, exclude our, quality is very important. So we are improving quality standards of our summit material. You can see the low volume is one of the indicator of the quality profile. So you can see the large, large set of lines with prepared quality traits. And also we are increasing the kernel weight, 1000 kernel weight over the time. So this is good for many countries. They look for bold plum seed. Farmers like that kind of phenotype. And also you can see the bread making test also been done in Summit Quality Lab. So you can see the poor to excellent quality of different combinations. So we also consider these traits. And at the end, the climate change, we have converted our about 18 hectares of new land with the drip system, so that we can able to regulate the water supply, and we can get more uniform data in our Obregon field. And also I said these are different phenotyping studies we do. And at the end, wheat and heat is not mutually exclusive. So we put a lot of effort on the heat stress. We plant our normal sowing in November or early December, but we plant these trials in late February to expose under continuous heat. So this time we put almost 5,000 lines, the stage 1 lines under heat, to identify some of those heat tolerant lines with better yield stability. So after this whole breeding process, these are the nurseries, Smith nursery, we distribute to different partners. There are so many partners in India, they get these trials and they return the data. Basically, the data comes from this international testing. In fact, we use them in our processing. So this is best way of utilizing win-win situation. So the national programs, they can use release as varieties, or they can use our material in their breeding program, as well as we can use that data for our crossing purpose at Mexico. So this is some of the conclusions as future outlook. So yield, as you could see, the yield potential is continues to increase, but we need to expand it or modernize the breeding program. And the best way to do is packing of several traits, the yield potential, as well as the stress tolerance, nutrition quality, and the set of germplasms. So rust remains one of the important disease. We need to put a lot of effort. And new diseases like wheat blast also emerging because of the climate change. So we do have good resistant source, but we need to identify some non-2NS translocation sources. And we now standardized, optimized our breeding cycle from cross to cross 3 years cycle when compared to the 4 to 5 years, and optimizing the testing in different selection environments. And also we send our material early in the stage to the partners. They can have to get access to that. And we use large amount of genomic data for making our decision making on our crosses, the combinations when the optimal crossing plans. As well as we put a lot of effort on the mainstreaming zinc and iron on our breeding program. With that, I would like to thank some of our colleagues, Dr. Ravi P. Singh and Crespo, Pawan Singh, Alison is our director, and Simit Bisa team, Philomene, Uttam Kumar, Karthik is also here. And thanks to our funders from Gates Foundation and other agencies. Also, thanks to other governments, including India, to CGR funding. With that, thank you very much.
Friends, it has been a very nice presentation by Dr. Govindan from CIMIC. And I am sure definitely there may be some queries, maybe clarification, questions. Yes, Dr. B.B. Singh.
My question is that in SMIT program, do you aim to increase zinc and iron or maintain high level of zinc and iron in most of your elite lines, or you have specific varieties which are rich for zinc and iron?
Thank you, sir. So actually, like few years back, we are targeting only for few varieties, but from this new project, we are trying to mainstream zinc across our elite lines, which means large majority of our lines will be zinc biofortified in coming years.
Yes, October.
Dr. Balu, how much improvement you expect from introduction of NS translocation in your population? One thing. Another thing, how did you estimate that 480 kg gain was there by introducing a single gene?
Okay, that was a good question, sir. But now there are small models, we can able to identify the allelic effect of a particular gene by comparing, to reduce, take out the other factors. So there is certain way to do, but I'm not the one did the analysis, but I can connect the person who did the analysis. There are certain ways we can able to estimate the gain from particular gene. But the 2NS is quite promising for many traits, except that YR17, it's not that holding up anymore, or there are some virulence in different countries. Perhaps still the prevalence of 2NS is very high in our material. But also I figured out there are a lot of variation within 2NS segment size. For instance, in Brazil, they have small 2NS, and in other countries, they have different size of the segment. So that's a difference.
Any other? Yeah.
So, Velu,
yes.
Actually, I wanted to ask this question because Dr. Singh is here from medical science. I was wondering that since we are applying zinc sulfate most of the time in order to increase the uptake by the plant, and we are measuring in the kernels zinc as a ppm, maybe some sort of a method.
Unit.
So I want to know whether the active compound, zinc containing compound, what is that? Probably some coenzymes and or zinc containing proteins may be there. Secondly, how much do you think it is left when we are processing the wheat in terms of the flour or the chapati?
Yeah,
So this is
that's a good question actually. Let me take the second one first, that's easy to answer. When we make the whole meal atta, it's most of the zinc iron will be retained. So that's why, this is good strategy when we go for whole meal flour or whole grain flour atta. But in case of enzymes, actually I'm not,
Mic, please.
Yes, please. Yes.
We talk of iron in isolation. Most of the studies are done in isolation. When we talk of zinc, we talk of zinc. But actually, you will find that in the bioavailability, they can be counterproductive also.
Yes.
One may stop the absorption of that. Secondly, I'll give you one example which I did was, and it was a very, very interesting observation. Pan-India, iodine is required for IQ. So one of the first successful fortification, if you can say, this country, debated in the parliament is addition of iodine in the soil. How many of you think actually the mother gets the iodine? Because that is straight away related to the IQ of the child. Straight away. Iodine is a very sensitive marker for IQ. That is how it was introduced. Most of the parents put the iodine once the food is, while cooking the food. And iodine being a halogen, it gets evaporated. So literally, I think when we talk of 45, we also must know the customs in which we are cooking the food. Because you see, you are only measuring till that packet is open.
We don't know what happens after.
What happens after?
After in the pot.
So it was learning for me, and the only place in the country which was adding salt in pregnant mother after cooking was Andhra and places of Andhra. Every other person was adding it before. If you add it before or you keep it, it doesn't. My other request is, while food fortification is something which, even when I was in the ministry, talked about. What about those foods? Because I was hearing Sir's lecture that during the Green Revolution, we left over so many things because that was needed at that time. But those resilient varieties already enriched with micronutrients which nature has provided.
Right.
Do you consider that to be inferior to the fortification? Or do you think that both should be encouraged? Or do you think that that should be forgotten? Like a ragi, you come from south.
Right.
So ragi is already fortified. As a doctor, I write calcium. I do not write ragi. But ragi contains it. So vegan rice, if we fortify with calcium versus ragi, do you want to support both? Do you want to think only of the fortified, or have you thought about some advantage of the fortified food over the already fortified?
Yes. So I would say that, the fortification and biofortification, it should be complementary, So we should not stop fortification. That is really important. As you mentioned, iodine is really important. We are not getting from any other source. For also zinc, so we need to get every day or in our diet because it's not being stored in our body. So, but maybe in iron it can be stored. So I think we should take a both approach, either fortification or biofortification. But I would say that within biofortification we consider both iron and zinc, because if you increase one should not go down. So that's why we always talk about zinc and iron together, but we find always good correlation between these two traits in wheat at least. I think it's a case in other crops as well. Yeah, so.
Dr. Kasingh.
So thanks, Velu, for excellent presentation. I will just add a bit on what Professor Singh said on biofortification versus fortification. So we all know that, fortification is a short-term strategy, is a good strategy to address the problem of malnutrition, but as a long-term sustainable solution, it is biofortification that should be stressed upon. But since fortification has got a lot of, industries interest, that you take, for example, all broken rice, which otherwise sells at a very cheap price, make flour out of it, then knead it, add iron, zinc, folic acid, and then subject to extrusion machine. And by extrusion, you print the grains. We call it genomic imprinting. You can print the grains exactly of the same variety size in which you want to mix it. If it is basmati, you can print long grain. You can print. If it is samba masuri, you can print short grain. So you mix it. You can't differentiate whether it is fortified, how much is added, and then you add in a proportion of 80 and 20% fortified and 80%. So this way there is a huge utilization of all broken rice into a high value rice, converted to very high value rice. After all, government is supplying, but government is buying, paying the taxpayers money. And that's where industry is pushing a lot as far as fortification is concerned. And biofortification in the process is getting somewhere on the back front, where we are enabling plant to accumulate more of nutrition through their root system into the grain. It is proven fact by experimentation that bioavailability of the micronutrients which are provided through fortified food versus biofortified food, it is always high. Biofortified has got much better bioavailability as compared to fortified. Welcome, Dr. S.R. Rao, from former, Department of Biotechnology advisor, and he has been, in fact, addressing this issue through, a project that we were doing on golden rice to enrich rice with provitamin A. Now, coming to another question to you, Velu.
Yes.
And in relation to connecting with the presentation of Professor Arun Kasingh, he raised certain very pertinent points, saying that how we raise child, how child is born, whether it is cesarean, whether you have surrogacy, or you have in vitro fertilization, or you have natural birth. All these factors play a very important role in the growth and development, right? Coming to plant, now we have come out with split breeding, and we are targeting 8 generations of a crop, up to 6 to 8 generations of a crop in 1 year, which otherwise it is natural process, is 1 crop 1 year, and max 2 crops when we have off-season nursery. Now, when we go for 8 crops in a year, in an extremely controlled condition, where you are giving light of your choice, temperature of your choice, and everything of your choice completely changing, the development of these plants and the kind of epigenetic variation that is created or generated, the epigenome, how it will be inherited to the next generation, what will be the impact on the adaptation of these plants in the natural conditions. Because in breeding program, when we are selecting plant in F2 generation, F3 generation, F4 generation, there is a progressive build-up of homozygosity, exploiting genotype by environment interaction, right, which is an important component of breeding that we all understand. Now here, the genotype by environment interaction is completely done away.
Right.
All we are going to look for is in the finished product, we will have multi-location testing, and then we will try to identify something good. But during the progressive development of homozygosity, that component is completely missing. So what is your comment on that?
Yes, sir. Thank you, sir. That's a very good question. So that's why I mentioned, sir, we are not putting all our eggs in one basket. So this is kind of, piloting, and we are proof of concept. We're trying to see how speed breeding material, I mean, advancement versus the conventional shuttle breeding. So certainly there will be a trade-off in terms of agronomic performance, but the quantitative geneticists, they say we can offset that trade-off by increasing the speed. So we are playing this game and trying to see how we can able to get both a good agronomic type as well as the yield stability, etc. So we are just trying set up populations, but we still have our traditional shuttle breeding scheme. Yes.
Any other queries, question, clarification? Professor Singh, you have some? No. Mike, please.
Good afternoon, sir. Thank you for the opportunity. So when we talk about adding nutrition to our food, we just talk about fortification and biofortification, which means we are adding the things that are not present in the crop, or we are improving the already present things which are very low in that. Instead of that, we can also suggest other minor crops, like if when compared to wheat, wheat has a very good source of iron, but we can also consider buckwheat, which has more protein and also iron content than wheat. So why don't we consider that? Why are we running on adding things that we don't have in that?
Right. No, it's a good question as well. But I'm not against minor crops. I mean, millets.
So this is not only for you, but
Yeah.
in general.
In general,
when
yes.
we talk about anything, we talk about adding protein or any other things into rice, wheat, or maize, and not other things.
Yes, actually.
I would just like to add that on the call of Honorable Prime Minister, now 2023 is going to be celebrated as the International Year of Millet. Globally, the world has accepted it. Millets are extremely important. They are called nutrition, no more force season, and they are going to become part of our regular supply chain.
Thank you, sir.
There are no questions. I have one query. That you said that you have bulk up to 3 to 4 generation, and then you do selection. Then you also said that in your greenhouse, you grow your F2 plant population. So, I mean, per cross, what is the number of plants you have in F2?
So, well said. In the field-based selections in F2s, we would have more than 1,500 plants per population per cross. But in this greenhouse, because of space constraints, we may not be like 200 to 300 plants. So that's a drawback, I would say, yes.
Yeah, then we should not really go for such thing. I mean, if you have population, larger population, always.
The F2
Dr.
is
Singh was telling that if you have, one pair of gene and then you take 2 to the power 24,
yes.
Then
all the shuffling and
combination where you have to grow billions of plant population. So I think that was one point. Thank you very much for nice presentation. So we have come to the end of this session, and I would like to profusely thank all the 3 speakers. The first one was Dr. Arun Kasingh, who emphasized the role of epigenetics in the newborn babies, various effects, 4D effects and so forth. And that was really very interesting topic. And that has been, further on correlated with other presentation. Then coming to Dr. N.K. Singh, he has made presentation of the basic researches in rice, genome sequencing and how the information generated has been used for developing a number of rice varieties through the techniques which are available, how you can transfer through mass or backcross or recurrent selection kind of approaches. And this is definitely a very good effort in that direction. And I'm sure all the breeders would take note of this thing and use in their own program to develop varieties. Further, he has also done genome sequencing of some of the crops like jute, for example, he mentioned. And in mango, he has done work where India is interested, not other countries are interested. So that's a very plus point which he has taken up. And finally, coming to Venu Govindan, definitely SIMIT is a large organization. They have a facility of shuttle breeding program that really adds to their program in a big way. And then also they have networking, international collaboration. So all the material which is developed is tested across the location globally, and therefore whatever comes out of it is definitely, I mean, whether it is biotic or abiotic stresses, they are all characterized, and then only after that it is released. He has also shown the effect of zinc and iron, how if you balance the two together around 20 to 25 ppm, then this is okay. But and then further he showed that in some procedure they have gone up to 6 ppm higher in increasing the level. So I hope that iron also increased along with the zinc, not only. So that's a good effort. And this biofortification, definitely, as Dr. A.K. Singh said, in long term, it is going to be a paying proposition. So with those few remarks, I again profusely thank all the three speakers and the audience here for their patience hearing and the queries and clarifications they have really raised, which has further enriched to our knowledge. So thank you very much for your patience hearing and kind cooperation. Thank you very much.
Thank you, sir, for the informative session. May I now request our director, Dr. A.K. Singh, to felicitate chairman and speakers of the session with token of love.
Mendel.
Thank you, sir. Finally, may I now request Dr. K.K. Vinod, sir, for vote of thanks.
So first of all, I thank this opportunity to convene this session. And at first, I would like to thank our chairman, Dr. Arun Kasingh, for meticulously conducting this session and taking the session forward. And I would also like to thank all our the speakers, Dr. R.K. Singh and Dr. Velu and Dr. N.K. Singh for their fantastic thought-provoking talks. And I would also like to thank all the speakers, I mean all the audience who has participated online as well as offline on this session. And finally, I thank our director for felicitating the guests. Thank you very much. I have an announcement. Please join, all the delegates, please join in the food court for the lunch. And all the school participants may remain seated. Thank you. The next session will start at 2:30.
Just a minute.
Yeah.
Because we are running behind schedule and we have an additional speaker, Dr. Singh, will be speaking to students. So we are starting the student session right now because we are slightly running behind time. Dr. Singh has to catch a train. Dr. Arun Singh, he has kindly agreed to speak to students. So we will right now start the session, next session. Students, be seated. You can come front once the front rows are empty. Sir ka khana ho gaya? Ho gaya.
Okay.
Yeah, that.
Fine.
Give some time. Welcome and who will do? Huh? Director sir will be there, no? How are you, sir? Sir,
Just to introduce.
Water bottle. Water bottle. मुझे दे दो. शुरू कर वेलकम कर
किसका है
Very good afternoon to everyone.
After a wonderful morning session, I welcome you all to this special session 1, Genetics in Education. To chair the session, we have with us Dr. R.P. Sharma, President, 15th Genetics Congress Trust. Sir, I welcome you to grace the session. I also request Dr. S.R. Bhatt, sir, our co-chairman for this session, to kindly grace the dais. I would extend my welcome to experts of the session, Dr. Arun Kasingh, Dr. N.G. Prasad, who is joining us online, and Dr. S.R. Rao, sir. A very warm welcome to you, sir. I would also extend my welcome to the conveners of the session, Dr. Akshay Talukdar and Dr. V.S. Hegde. And I would request Dr. Jyoti Kumari, Dr. Vigneshyam, and Mr. Manoj Gowda to act as rapporteurs for the session. Now, I would request Dr. Akshay Talukdar, sir, to kindly introduce the experts of the session to the house.
Good afternoon. I welcome you all to this special session. This session is specially dedicated to the students and teachers. I welcome all of you to this conference, and there is a heartiest welcome from the 15th Genetics Congress Trust, because this session is basically organized by the 15th Genetics Congress Trust, and chairman is the chairman of the session also, Dr. R.P. Sharma sir. He is here. Another trustee, Dr. S.R. Bhatt sir, is also associated with that, our 15th Genetics Congress Trust. Other dignitaries in the dais. Now we are going to listen first. Professor A. Kasingh, Arun Kasingh. Today morning also he has made a very beautiful presentation, but we felt that he has a very good connection with the students. So that's why we requested him for a special waiting for us and to interact with the students. So before going to the formal introduction of our speaker, S.R. Rao sir, or other dignitaries, that we will start straightforward. We'll go to or request Professor A.K. Singh sir, because he has to catch a train. So student friends and my respected teachers, let me just 2 lines and let me tell about sir. He is a doctor by profession. He is currently a professor and head of neonatology at AIIMS Jodhpur. Neonatology is, this is your branch, means this is related to child only. And that's why we requested him that please give little time to our child. This is our future generation. So that was a good time for you to listen to him. It's a beautiful orator, I would say, and he can connect nature, science, and all education, everything put together in the same plate. You will enjoy. So I will not stand before you and sir. So straightforward, I'd request Professor Arun Kasingh sir, welcome him with a loud applause. This personal request.
So, first of all, thank you very much, sir, for providing me this opportunity to connect to the students. आप लोग अंग्रेजी में पसंद करेंगे या हिंदी में सुनना? Or it should be by choice is yours. Both language, by okay. So let me start that. जिंदगी का एक पक्ष होता है, a part of your life you spend in schools. When you are in school, some of you do great, some of you do not do so great. Some of you think that this child, parents are very, they come from a family, traditional family of people who are highly educated, and hence they have an advantage. Please raise your hands how many you feel that if your parents or you were born in a family, let's put it in simple way. Tendulkar's son, did he have an advantage to become a cricketer over others? And why was that advantage? Why was there an advantage? The right tools to use, okay, that's one. So in this discussion, we will not take up anything. We'll go with everything. Among the students, anyone, your opinion? Yes?
guidance. Tendulkar's wife was a doctor. Whatever I have read about Tendulkar was that there was no one in his life who could made him read a book, even a book on cricket. There's a shop in Mumbai which keeps all the things of cricketers. So he said, many a times I invited him, they put their signature, etc. on a bat, etc. and to come and read a book on cricket. He would not enter the place, otherwise I would push a book on him. On the other hand, he has a wife who was a pediatrician. Okay. So who is going to guide? Tendulkar or Mrs. Tendulkar? Because Mrs. Tendulkar is good at medical science. Tendulkar is a wizard person in the field of cricket. Yes. See, none of you in nature, excepting plants, you are all born with diversity. You are neither your mother's child nor your father's child. You are entirely anew. Mendel, whose statue you just saw it being presented, he could do cross-pollination. That means the same parents, the children, then marrying between the children. But usually, even if you marry, the genes in human beings are dimorphic. They are not monomorphic. That means you get from two. So nature wants diversity. So whose influence now would be? I would find it very difficult to be born in Tendulkar's family. On one side, the mother would be pushing for studies. The other side, the boy would feel, why do I need to study? My father never did it. He did pretty well. So now, are you still thinking the same? People thought of this. There was a time when people thought, why don't we get married Nobel laureates? So children will also become Nobel laureate. Did it happen? Yes. This was a huge research area where they wanted that people who are highly educated, if they get married, I think the best answer, whether it is a joke or etc., I do not know. But at least when I was a student, one of my teachers had commented this, that there was a beautiful lady and there was a beautiful male person who was not so beautiful to look at, but he was a person of great talent. So the lady said to him, what if we have a child with your brain and my beauty? And the answer to it, he said, it could happen the reverse. I would tell you, it doesn't happen that way. It doesn't happen that way. Though you can make him play cricket, but to make him Sachin may not happen. I grew up in Calcutta in an area where the best lawn tennis coaches were there. The child used to be taken from a very early age to play lawn tennis, but he didn't become Akhtar Ali. So his name was Jishan Ali. It doesn't happen that way. So what is it I'm pointing it out? All of you are sitting in this room have the potential to become what you want to become, irrespective of who your parents are. If you all think and be very clear with it, yes, there is an advantage if my mother is educated, because the environment she builds, it's not your genes, the environment she builds is stimulating me to learn. I'll give you another example, very famous economist, though nowadays it can be very controversial. But his achievement after 200 years also is unparalleled. His name was, in Germany, he was a Jew changed into a Christian, Karl Marx. The problem was different. Everyone reads about Karl Marx's achievements, etc., whether you like him or you don't like him, depending upon. But nobody asks, what was the impact of the parents and the teachers on Karl Marx? Raise your hands. How many of you have read? What was the interaction of the parent with Karl Marx? And when I started reading those letters, I understood Karl Marx was not a genetic answer. The answer lied with his teachers and with his parents. What was so special of Jewish parents, especially Jewish parents? So, one of you are studying in the school, you are in the hostel, you are not paying your fees, you are using that money for drugs, for addiction. One day the school principal writes to your parent, your child is lost. What will the parent do? What do parents do? They'll come and scold you. Karl Marx's father didn't do that. He took out his child from that university, put him into another university. There he got two teachers, Feuerbach and Hegel. He was a completely changed person. Completely changed person. A drug addict, a person who was not caring for anything, not interested, was a genius of his times. So the role of a teacher and the role of a parent is never to lose hope in any child. But that brings a very big question. हम सब जब parents होते हैं या teacher होते हैं, how do your parents talk to you? If you belong to a very elite family, let's say Mrs. Tendulkar talking, I'm just hypothetically making it up. I just know Mrs. Tendulkar because she's a pediatrician. I don't know Tendulkar, I've never met. I'm just hypothetically thinking कि उनका बेटा होगा and he is not listening. So what would the mother say? Your father is such a revered person. People him, he's like a godlike image. He's a heartthrob. The whole nation claps for him. You are his child. You have to work harder. What will the child feel? Did I write a letter to be born in this family? I didn't write any letter. Every day, if I have to listen to this, he'll say, forget me. Because in nature, genetics makes each one of you, irrespective of your parent, a genius in your own right. Each one of you. You may be bad at spellings, you may be bad at maths, you may be bad at this or that, but there would be something you would be great at. Find out that. Problem is, we are ever told, like this coat, you are wearing this coat, hence you must behave in this way. So I would rather take out this coat and be myself. There is a problem of this country, and you are the hope of this country. Our generation has let us down. Our generation, my generation has let us down. But today I am speaking to you. I can miss my train, but I do not want to miss the interactions with you. If any one of you sitting here listens and thinks, what is it that I'm talking? What I'm going to talk to you is to understand this country. When I was told by my parents, you belong to this caste, this, you belong to UP, you belong to this, I forgot that. What's my first name? Anyone? Now, even before that, Homo sapiens. There was a young child who is now a doctor. As a pediatrician, I would interact with him, child. So I will ask him, "Aapka naam kya hai?" And he would start, "I am a child on the planet of Earth in a continent called Asia, in a country called India." And then he would slowly come to Purulia, where I had met him. That's a beautiful way. You are all Homo sapien. And what do you have? You all want to look very bright, very beautiful. Let me tell you, nature has not made you the most beautiful animal. If any one of you believe in the power of Lakme and Fair Lovely, then write a wish list next time. You should be born as a peacock, not as a human being, because all of us have one color. A peacock has multiple areas of colors. The most beautiful woman no one would look at if a peacock has spread his and is dancing in the rains. You can go and listen. The most beautiful actor and actress, no one. I've seen this happening in Jaipur. The whole road was blocked. A peacock was standing and no one was complaining. Everyone was seeing it. Because understand the principles of physics of light. When the light rate falls, there are more colors than the peacock has it. But the peacock has a very small brain. You talk of an weight. All of you are weighed. Huh? मेरा वजन 30 किलो है, मेरा वजन 40 किलो है. 30 किलो वाला बेचारी या बेचारा यार उसके पेरेंट्स को बोलते हैं, आप क्या खिलाते हैं बच्चे को? वजन ही नहीं बढ़ता. All my life my father had to listen to this. I was always taken to a doctor. इसका वजन बढ़ा दो. Today as a pediatrician, I can recall that pain which happened to me. I didn't know what to tell to the doctor. जितनी भूख है उतनी तो खाएंगे. भाई आइसक्रीम सबको अच्छी लगती है, मुझे भी लगती है. मेरे सारे खाने बंद हो गए. डॉक्टर ने प्रोटीन, कार्बोहाइड्रेट, फैट दे दिया. हर रोज मुझे खाने पे उल्टी होती थी. But I had to take, because my father was under the community pressure and he related that pressure through the doctor to me. Don't have this pressure. You are not an elephant. Elephant weighs 60,000 kilos. रास्ते बनवाने हैं, हाथी को चलवा दो, रास्ते बन जाएंगे. 60,000 kilos, 6 tons. Neither you are a giraffe whose height is 22 feet. So what do you have? What has genes provided you as a Homo sapien? The height of the brain, the beauty of the brain, and the color of the brain. Why is the skin darkened? Anyone? Have you ever questioned? Why do I require fair and lovely to become fair? If you were born in the equatorial region, because of the high UV radiation, nature had to provide you an extra cover, an extra sunscreen, so that you do not suffer from skin cancer. Not a single dark-skinned person suffers from skin cancer. That's the extra protection which nature provided you with. You lost that. You didn't think, you didn't say thank you to nature. You said, and this doesn't happen only in India. Highly talented person, one of the best dancers of what you call, I hardly see them, who died quite young. Can you name? Michael Jackson. You know what happened? Michael Jackson's father said, your music is not enough. Your skin color is important. And he bleached his skin. Can you imagine? This is the power of an unrealistic thing which has no scientific basis. The African blacks were provided because there's excessive sun rays, which will cause them skin cancer. If I transfer the darkest person to Iceland, 4th generation, 5th generation, you would become as fair as a penguin. People in the marriage market, when they say, I want to marry a fair person, I say, go and marry a penguin. There can't be a fairer person. When you listen to a poetry, when you listen to some good music, do you really see the face of that person? Because that is brain's creation. So from today onwards, believe in brains. And when you are an Indian, you are neither a North Indian or a South Indian, etc. First is you are a Homo sapien. Secondly, your genes and your parents have got nothing to do with you. You are the flower which will choose its own color, not the color chosen by your parents. Have faith in you. Keep on questioning everything. You'll be surprised that people could contribute to physics, to chemistry, to botany at the same time because they felt they were dealing with nature which had no walls. So when we say, ye inke bacche hain, I always say, don't introduce me with that. I am a Homo sapien, that's good enough. When you become someone's child, you cease to understand that you are the child of this planet. You could question everything. There's no supremacy. That is what science is all about. When C.V. Raman was asking why the sea is blue, his teacher told him, why are you wasting your time? He persisted with it. C.V. Raman writes, when I got the Nobel Prize, I wept because the music which was played was not my own country's anthem. My country didn't have a flag, it didn't have an anthem. All his life, C.V. Raman wanted a young scientist to be born. Have you heard his last speech before he died? I wish I could make you hear. He was paralyzed. There was no TV during those days. Sir is from Bangalore, so he would know. His lines were, and I quote it exactly the way he said. He died just a few days later after this. He said, my life has been an utter failure. My life has been an utter failure. I thought I would do true science, but all I have left is a bunch of people who only copy the West. My life has been an utter failure. I thought I would do true science, but all I have left is a bunch of followers of the West. At this rate, there can be no science in India. There can be no real discoveries. He stops. C.V. Raman was a person, interestingly, who understood that arts and science, they merge when knowledge becomes free. I'll tell you a very interesting. He had a classmate of him who was after him because he was good in studies. And after the school, both used to walk together, discussing about science, mathematics, etc. But while going home, at one point, C.V. Raman would stop and would ask his friend, "Ye veena kaun baja raha hai?" And his friend would say, "What is that here?" Next day again, he would say. And every time his friend used to get irritated because his sister was playing the veena. And he didn't want C.V. Raman to get distracted. Incidentally, C. Raman married that girl and she became Mrs. C.V. Raman. After he married her, he did a beautiful research paper. You know what? He asked himself, I had never seen her. Why did I stop? Why was it that I could not think anything else? And there's a beautiful paper published in which he said that the pitch and the tone of that music instrument can really affect your brain's area in such a way that you'll stop thinking everything. And that was the reason why. He found a physics answer to something. So, पहली बात ये है, आप में से जिनको नींद आ रही है, वो खाली एक ही चीज सुन लीजिए, सोने का बहुत वक्त मिलेगा। एक ये जान लीजिए, आज से आपका परिचय कोई टाइटल नहीं है, आप होमोसेपियन है, जीन्स ने आपको यही दिया है। 99.9% genes of the American child and the African child is the same. All of you, your names may be different, your parents' salary may be different, you may be coming through various vehicles, but let me tell you, all of you are inherently the same. And what do you have as your wealth? 100 billion wires in your brain. How much is 100 billion? 100 times the population of this country. That's the richest gift nature has provided you. If it is possible, use them. When you think of a problem, you are using those wires. But when you are thinking, don't try to mug up something. That's not using the wires. Just take a plain piece of paper, start thinking. Why I have come to this world? Why I have been a Homo sapien? What is my advantage? This is the biggest gift nature has provided, and no one can undermine in this world. 100 billion wires. Usually, we hardly use when we die, 20 to 30%. That's what people say. When I go to a graveyard, I do not weep for the person who has departed. I weep for the biggest gift of humanity is now going to be consumed in flames, and nothing has come out of it. A country of 1.4 billion means 1.4 billion brains. One of you could become C.V. Raman Tagore. What was C.V. Raman pointing out? You just don't have to excel in class. You have to excel in your mind. You have to think. You have to get your answers. It may start with playing with a plant. My last thing is that all of you who are addicted to smartphones, for heaven's sake, with folded hand, one day you will realize it is taking away your 100 billion neurons. That's a commercial thing. When you tell a story to someone, your brain is getting fired, your imagination is getting there. Whether it is the structure of the carbon atom, where the person went to sleep and found a snake taking his tail to the mouth. So many of these are examples when dreaming. So the grandmother stories were actually helping you. Color of the skin, color of your smartphone, all this is too trivial a matter for a person with 100 billion neurons. Now, I would like to listen to you. And the second thing is cultures, religion. The entire genome sequencing of India has been done. What does it show? Irrespective of the caste, irrespective of the creed, irrespective of the religion, 78.1% of this population has Indo-European genes. They are the same genes. That 0.01% difference, that I am taking. That 78%, you have the genes of people around the world. I ask my students, on a piece of paper, write, if you had the choice to decide where would you be born, what would you write? And they write, which country? Which country? If I give you the choice, which country? Be honest. No. And that's perfectly okay. Our generation has not made you proud. Yeah. The maximum would write US. And you know why? Because USA is not made by one group of people. I have lived there. It is a diversity. Diversity is your strength, not your weakness. If your food is diverse, if your classmates are from various parts, if I had to open an international institute, which was the most important international institute of the world, now you can talk of India. सबसे पुराना विश्वविद्यालय 1000 years before Nalanda. 1000 years before Nalanda. Takshila. Why was Takshila? Where is it? If you travel, that's what I was telling in the morning, how to make this country called Hind, Jai Hind jo kehte. So these are the two bricks. This is north, this is a mountain. This is known as which mountain from north to west? Hind Kush. And from north to east, Himalaya. So this two top, this is the area covered by two mountains, Hind Kush and Himalaya, and two seas. This is the Arabian Sea, and this is the Bay of Bengal. And where the two seas meet, that's the Indian Ocean. So this geographical area, Takshila, is somewhere in the Hind Kush near the top. It is a window to so many civilizations. I teach at AIMS. What's the strength of AIMS? People come from all over the country, whether it's Karnataka, whether it is Bengal, whether it is. That diversity is the strength of my institution. The day that diversity is done, if the Takshila was an international university, people would come from China, from Central Asia. Chanakya was a teacher there. The first medical university was also there. So try to understand that diversity doesn't take away. Nowadays, there's a fight. In Bengal, there should not be anyone outside. In Assam, there should not be any non-Assam. All this fight is because we have forgotten we are Homo sapiens. In the words of Tagore, knowledge should be free from all domestic walls. And he said, the more there is a mixture, the better it is. Bengal did good because they are Armenians, French, all came there. They gave something, they took something. So from today onwards, whenever you meet a classmate, whether he has two hands or one hand, whether he has two legs or one leg, whether he has two eyes or one eye, that's not important. What is important is the brain, for we are all the brain. Thank you very much.
So student friends and my dear participants, so you might have heard, sir, as I told you, what is the beauty of his presentation. So he's leaving us because of his time. So let's give him a big round of applause. Thank you so much, sir. Thank you so much. And wish you a happy and safe journey back.
ਕੀ ਹੋ ਗਿਆ ਇਹਨੂੰ 11 ਤੋਂ ਇਹ ਕਹਿੰਦੇ ਪਏ ਕਿਆ ਮੈਂ ਬਣਾਣਾ ਆਪਣੇ
Now I request our president, sir, to conduct the proceeding of the session. Thanks.
Thank you, Dr. Talukdar. Good afternoon to all of you. This is a special session especially organized by the Trust, Genetics, International Genetics Congress Trust for the students. I will have to, because we are running short of time, I will just take 2-3 minutes to tell you a little bit about the trust, and then we go to the lectures, the 2 lectures which are organized. One will be by Dr. Prasad, which will be online, and another lecture is by Dr. Rao.
ਉਹ ਹੁਣ ਬੁਲਾਉਂਦੇ ਆ ਨਵਾਂ ਅਗਲੇ ਵਾਲੇ ਨੂੰ ਤੂੰ ਤਾਂ ਵੇਖਨੇ
Well, there is a body internationally known as International Genetic Federation. They organize International Genetics Congress. It started, the first Congress was 1899. This Congress is organized every 5 years. The 15th Genetics Congress was organized in India, and credit for this goes to Professor M.S. Swaminathan, the father of the revolution, which all of you might have read in your textbooks. In 1978, he went especially and requested the Federation to have the 1983 International Genetics Congress in Delhi. At the time, those were the days, and many of the children would certainly know indirectly. Our country didn't have that kind of much impression in the international world. They were thinking it's a snack charmers country. What is there developed? Dr. Swaminathan went especially with 2 basic themes. One, that was the time when India has already harvested the fruits of Green Revolution. We were almost self-sufficient in food. And for the picture that we are starving or we are poor, that was slowly getting eroded. And the second point which he emphasized to the international body was that India is a country with its social conditions. It still maintains a huge amount of genetic variability, human genetic variability. Well, for many of you, your knowledge, J.B.S. Haldane, a very famous British scientist, he chose and opted for Indian citizenship, primarily because he wanted to spend the rest of the time of his life on studying the human variability. Dr. Singh has emphasized on variability, how important the variability is. Variability in plants, that's what we study in this institute, variability for human beings, etc., which you see around, no 2 persons are alike. Your genes, 90% of your genes might be similar, but there are some genes which makes you different from each other. And that's Swaminathan to convince the international body that to have next Genetics Congress in India, and that's how in 1983 the Congress was held in India. It was a fabulous success. More than 2,000 people from across the world participated in the Congress. There were about 6 Nobel laureates who participated in that Congress. Dr. Swaminathan and Dr. Chopra, both of Dr. Chopra was Secretary General of the Congress. Dr. Swaminathan was the chairman of the Genetic Congress, which was held in 1983. Very successfully organized. Part of the money which we saved from that Congress, Dr. Swaminathan was of the view that we must utilize this money for propagating and spreading the message of genetics. And that's how in 1985, Genetics Congress Trust was born. We, from the beginning of our inception, we have been addressing various issues, genetics in relation to plant breeding, genetics in relation to many other things, but also genetics in society. Over these years, we thought that it would be much better if you want to take the message to the public. The school teachers and school students are the best media. And for that, we had organized teacher training program over these years. Many of the teachers, maybe there might be some who have participated in it. More than 200 school teachers from NCR region participated in that hand-on experience on different experimental protocols which you read in NCRT books. So that's the kind of message we have been spreading through the Genetics Congress Trust. Since the time is running short, I will request Dr. Bhatt to take it over and request Dr. Prasad and Dr. Rao for their lectures. And thank you very much. I am with you here.
Dr. Prasad, there is a difficulty in contacting him. We will start with Dr. Rao's lecture first, and then we will see if we can establish contact with Dr. Prasad.
The slide change because Dr. Prasad, who is to be online, is from Mohali, and it's taken a little time to streamline it. Meanwhile, we have Dr. S.R. Rao with us, and I'm requesting, I'm giving you the brief background of Dr. S.R. Rao. Dr. S.R. Rao fortunately did a PhD from this institute. He was a visiting scientist in Tota University, Japan, and Wake Experimental Station, Adelaide, Government of Australia. He joined the Department of Biotechnology, Ministry of Science and Technology, Government of India in 1989, and progressed to become senior advisor to Government of India. Currently, he is Vice President of Sri Balaji Vidyapeeth University in Pondicherry, promoting cutting-edge research innovations and interdisciplinary development. He has rich experience in dealing with R&D promotions in agriculture, medical and animal biotechnology, planning, budget and technical coordination, biotechnology regulations, capacity building, university education, teaching and training, establishing modalities and negotiations in assessing major international facilities. He served as Officer of Special Duty during 2005 to 7 to Ministry of Science and Technology and implemented projects and programs of the interface between science and society. He is specialized in core and cross-sectoral policy issues of biotechnology development. He is founder and editorial board member of Asian Biotechnology Development Review and has published 36 research papers in national and international journals, 2 books on animal vaccines and diagnostics, and others on biotechnology in Asian countries. Most of his vast experience which Dr. Rao has with him, he's going to talk to us on an issue which is so important for all of you, the kind of opportunities biology can provide in your life. And therefore, I will request Dr. Rao to kindly enlighten us.
I will go, as per your request, I go bilingual, okay? So, yeah. So first of all, I thank the organizers for calling me to this important function, particularly meeting the students. It's a great pleasure to welcome all of you to this session. In fact, I will take you through a journey of some people. Although Dr. Singh, previous speaker, has said several things to enthuse you in genetics, biology, and others. But on ground, we face several situations. It was so post-independence, it is so now. So how the journey was with biology and what are the current career opportunities to be more practical. So on the other side, you have got a lot of pictures which just shows that the science has progressed from one leap to another leap and has gone to several heights. So, yes. So this was I'll do a conventional presentation, but I'll tell you in between a lot of stories. This is a slide which I borrowed from our earlier president, Abdul Kalam. He used to use this in many meetings as a first slide. He says that before the Industrial Revolution, even the Stone Age people were having science. A lot of things were discovered during the Stone Age. Even after that, we had a Mohenjo-daro Harappa civilization. And then when the Industrial Revolution came, a lot of equipment manufacturing took place. In fact, now, since about 100 years, we have entered into a revolution of computer education, information technology, communication technology, as well as biology. The biology and information technology now are competing neck to neck. Finally, they ended into a race and they found that they are together now. I'll show you how it is. So there is no distinction between IT and bio. Finally, it all gets merged into one place. That's how the journey goes. What I'll do in the beginning is, I will tell you how the Mendel's concept has taken us to current level of science. The entire biology traveled for the last only 100 years. I will tell you, and then I will tell you journey in the post-independence, what was in opportunities, how a person traveled into diverse fields with a biology, with an example, is very, very practical, a simple individual, and then we'll go to the opportunities. This is one of the mind-boggling innovation in my view, the invention of microscope, which you all might have studied. But the one which is on the right, left, or on your right, it is called folderscope. Recently, I'll show you all the slides are open for you. You can get, no need to take pictures. Okay, I'll leave these slides. Anybody want to use? See, this folderscope was used basically to many students in NCR region made this folderscope. Department of Biotechnology sponsored. And then what a wonder was, first time the world of microbiology was discovered. This is a microbiology which made all the difference. This is a PPT this is a picture from a recent outlay of what all microbes we know. So there's at least some 100,000 microbes, even in the extreme atmospheres like hot springs and then cold deserts are all documented now. They are producing wonderful compounds for the future of biology. Now I'll come to the theme of this seminar. From Mendel, how we traveled to genome editing. The journey is a very astonishing. It is all happened just in 1.5 decades. Whereas when Leeuwenhoek invented the microscope, the lens which he had, when he passed the light, all the 5-7 colors were visible. That is called chromatic lens. To prepare a achromatic lens, which does not pass only the white light, it took 300 years. Whereas the revolution, which I showed in the first graph, happened only 1.5 decades. This revolution made the biology opportunities. Plenty of them. I'll show you how they are. Now, after Mendel in 1933, there came the first description of genes. Then Watson and Crick came into DNA, you all know. Then we came to a plasmid sequence. Then we came sequencing platforms. How do we sequence the DNA? It is called Sanger's platform. See how within a short period of 70 years, Mendel's hypothesis progressed to levels of providing the evidence, scientific evidence, that there exist genes and the genes are the background of DNA is the background. So I used to always wonder whether it was a hypothesis. But now we have a scientific evidence that genes exist through the Sanger sequence. Then the first thought about sequencing of human genome came in 1984. And this thought went into further. And then finally, Human Genome Project was launched. And in 2002, this project has given into a full sequence of humans. And then it has motivated sequencing of several organisms, bacteria, mouse, and also the worms, Drosophila. And then finally, these are all incidents, 2003, the Tony Blair and also Clinton announced to the world the human genome sequence. And after that, what happened? Several companies started developing technology of sequencing. And that made the human genome, which has got 3 billion letters, 20,000 genes, and 3,000 have defects, we can call mutations. So these mutations were recognized, some of them are very lethal to the human existence. Although Dr. Singh previously spoke, genes are all good, but there are some genes who are very bad. They can be genetic disorders. I do have some genetic autoimmune disease. Somebody discovered, then they said, no medicine will work for you. It is a genetic disorder. So a lot of people, India has documented something like 3,000 genetic disorders. It's a huge book of ICMR. So there are recognition that some bad genes do pass on from generation to generations. Now, even you see I was wondering how many of them are in maize. Maize is one where I got my PhD. You see the colors, so many, such a beautiful amount of maize variation. Have you ever seen this? You see, I'll give you this slide, you use it. It will be very interesting. I always wonder how the nature has created. Now, after that, the institutions came. Actually, China was in the forefront. They started a Beijing Genome Center. Every day they sequence hundreds of genomes. They have hundreds of machines. And I was very lucky to visit every floor of it, which is not common for anybody to visit. At least minimum 300 people work in this on a shift basis to understand which sequence is responsible for which trait and which character. So then there was an important person called Richard M. Myers, who actually was one of the stalwarts in human genome program. On his name, they have built one sequencing platform in Alabama. It's called Hudson Alpha Institute, and where I was visited. And they are now sequencing most of the infants' sequences on a daily basis to find out and document all those genetic disorders. Today, actually, when human genome was sequenced, it costed something like $3 billion or 150,000 crores. But today, the latest quotation the industry gives is 15,000 rupees for full genome sequencing and with all the analysis. And they will give you a chip if you have an ability to screen them. But as of now, with all this equipmentation, 100,000 highly diverse whole genome sequences are available. You just imagine in 1967, now what is it? It is 2002. So all this happened in 1.5 decades. So fast. The field is moving so fast, uncomparable it is. So today, in fact, we are all storing the data in cloud. It's called big data now. Several terabytes of sequence have been stored. Even if you don't do anything else as a scientist, if you understand this data and publish, you are a great guy. So now this science also has led to this genome availability, all those technologies, equipment made available to develop other technology, proteomics, metabolomics, computation biology, system biology. Why I'm showing you this is, the biology is not one term now. Biology has so many disciplines: system biology, synthetic biology, stem cell biology, animal biology, nanosciences, advanced materials. Now the last one is artificial intelligence with biology. So the whole big data that I am showing, the artificial intelligence is used to decipher very fine information. So that is how the growth has happened. And then finally, recently, in some 7 years ago, there is one scientist, he's a Chinese scientist. He found that in curd, some bacteria actually resist the viruses which kill them. Then he went deeper into this, and he found that these bacteria produce nucleases, enzymes, which modify the DNA. Then he invented genome editing. This is called CRISPR-Cas nucleosis, and I will not go into detail. So these enzymes can substitute. In place of C, you can have T. For example, you hear sickle cell anemia as a disease. That is only different by one nucleotide, one letter. If you replace the letter, it is gone, it's so gone. Then you have insertion, then you have a deletion. So any of these three things can be done very easily today. So that gene of Mendel, the genome, we can now manipulate the genome any way we want. Without bringing additional gene, we can bring the required variation. So what can we do with this? Correct infertility, dominant mutations, recessive mutations. So all these things, for example, sickle cell anemia, muscular dystrophy, and etc. These are some examples. Now what is happening? There are schools which are misusing it also for muscle building, for building the microbes in the stomach, for manipulating our own genes for a better features. So all these things are going. In US, at least 300 garage laboratories, which are called do-it-yourself. So then gene therapy came. Now today you can correct the genetic disorders, which I was telling, due to bad mutations. All these new technologies have come for medicine. This is what biology done in 1.5 years of journey, starting from Mendel to correcting our own genetic disorders. But post-independence, what was the opportunity? The British left a bad legacy of creating slavery, slave students, clerks, accountants, and some, of course, good institutions. Like this institute was called Imperial Agriculture Research Institute, Imperial Veterinary Research Institute, like that. But otherwise, they all use these institutions for their own advantage. It was first time Radhakrishnan Commission recommended creation of University Grant Commission. At that time in independence, after independence, there were only 2.5 lakh students all over studying in 20 universities and 496 colleges. So what was the pathway for biology? The pathway for biology, you do 10th standard, and then you do either go to biology or you go to PCM or PCB. Either you choose biology or use mathematics. In some places, PCMB is also there. Very rarely that is also there. But at that time, when you have a biology, you go to MBBS and MD, you do. And then now you can do DM also. You can do PhD also. Similarly, the other one is MSc Agriculture and then MVSc. And the last one is traditional BSc, MSc, PhD, and postdoctoral. So this is the pathway available. Postdoctoral, either in India and abroad, was a very common feature. And in several disciplines, you can do this. This was the limited opportunity, almost up to 40, 45 years after independence. Most of the people who are sitting, maybe 60 years, 50 years, have gone through this blockage of opportunities. Not beyond that. They don't know anything else. So I'll share one person, Mr. X, how he traveled. He was born in one place in small districts of South, and this is the man, Mr. X. And the Mr. X studied in a very remote school. Even today, that school is like that. Then he studied in a college. At that time, it was an intermediate education. And then he happened to be in the 10th standard. He was the number 1 in the board exam from that small town. Then this is the place where he studied all his life. Then he came to a town where he did his BSc biology, taking agriculture as specialist, because I'm taking agriculture as this is agriculture institute. From there he came to this institute. Okay, from this institute, where he went to Japan. And from Japan he went to Michigan, and Michigan to Cornell. In this journey, what he has done, he has left his subject. Whatever he studied in agriculture, whatever he studied in 12th, those days even, the opportunities were such abroad, he shifted to biochemistry. He became a fatty acid molecule synthesis, fatty acid mixes, and he became a fatty acid chemist. From there, he joined Unilever's. In Unilever's, he used to do for some time the fatty acid synthesis, mixing of edible oils, etc. From there, he came to Unilever, Hindustan Levers, which is a branch of that one. And from there, again, he was back to Delhi. Okay, he started his journey from Delhi, and then he was from Unilever's. He came to Krishi Bhavan. There was a technology mission on oilseeds, and he started his life in the technology mission on oilseeds. The oilseed technology, one of the missions, was held by Korean, who is Korean. He is the one who did the milk revolution from NDDV. Okay, he was responsible also for the brand Dhara oil. So he was in the mission. From there, this man went to biotechnology department. And from biotechnology, after that, he came to a completely different endeavor in a medical school. So you just see, during this journey, where all he went. He was PCB and post-independence. He did all the way, and now he is in medical science. In the pathway, he gained experience, not only in agriculture, veterinary, and medical, but all the new areas of that opened up. I showed you how the biology came. So the opportunities came as the challenges were taken, risks were taken. But for you are very lucky. 1990 onwards, there was economic liberalization. Manmohan Singh and the Narasimha Rao opened the world to the World Trade Organization and also to the, it just opened up everything. Education, trade, transport, goods, manufacture, food, everything. Foreign exchange. So over time, till now, this policy was used from 1990 onwards. So these are the different reforms they did. Now you see, that time it was only 20 universities. Now we have 1,000 universities, 54 central universities. This shows how much opportunity we have. 416 state universities, 125 deemed universities, 361 private universities, 159 institutions like AIMS, IIMS, IITs, ISERs, and so many so forth. So today you have plenty of opportunities till there is a momentum to increase this into double number. Almost every district will have one multidisciplinary college as per the current plan. So now the same theme will take. What happened? Now today, same again PCB is there, PCMB is there, that is you take the same diagram I am showing again. This opportunity remained the same. The only difference is MBBS MD, if you don't pass NEET properly, So 1990 onwards, there was also one good or bad phenomenon. In MBBS, in previous days, every college you have to write an entrance. But here, one entrance for all. So wherever if you get a high rank, I also wanted to become a doctor, but I never became. So the thing is that when you get a high rank, you will get a good place. It will be only 50,000, 200,000 rupees, you can finish the entire medicine. Otherwise, you may have to pay up to 40 lakh rupees. So MBBS MD has become very expensive. Unless you have in your father, mother has a pocket of some crores, then you can study. At least 1 crore or 1.5 crore. But BSc Agriculture, MSc Agriculture, MVSc are still better. They are affordable. Even private institutions are starting now, like SRM in South, VIT in South, have already started agriculture colleges. And a lot of people from other universities are also joining. Now coming to the traditional pathway of BSc, MSc and PhD. Now the disciplines are plenty. You have huge opportunities. And as you grow towards the postdoctoral, I told you all those different sectors of biology you can take up in research. And a lot of biology people are now the only ones who are getting the Nobel Prize. Okay. Now, suppose I did not get all this because it's still a competitive field. The other one is B Pharma. B Pharma is not a joke. It has a lot of opportunities. You can do diploma. You can also do B Pharma. At least there are 300 colleges, and then some of them are top B Pharma colleges. They have M Pharma also. Recently, I found in the medical colleges, medical universities, what you call allied health sciences. If you still are interested in the health sciences, you can see the range of courses that they offer at the BSc level. In fact, two of them, I tell you, occupation therapy and paleotherapy, that is taking care of the old people, and also the radiotherapy, radiologist post. They are paid so much after the BSc. It's the most lucrative job. So you don't have to be really unhappy that you didn't get an MBBS or anything. This is also a very recent, decent course. Particularly, you see the optometry. All those people who prescribe you, the specs are only from allied health service. They are not doctors. They are basically from allied health services. So you have got these 19 to 20 opportunities after the graduation. You don't have to unnecessarily worry about, I didn't get NEET, my father did not have money, all those things. So then you have, besides BSc Agriculture, you have BSc, Home Science, Nutrition. There is one Avinash Lingam Institute of Nutrition in Coimbatore. Every year they churn out 100 to 200 students. You see any hospital, abroad or here, you can find their people as a dietitians. Very great opportunity there. Similarly, now you have food science and technology. In Rohtak, the Ministry of Food Processing has started. There also you can join as a food processing mission. All food that you eat today is all packed. So you have 100 opportunities in food science and technology. Again, there are 12 subjects at diploma level. You can also go through. It is not enough that, you can also go for management and communication. Oh, I'm a biologist, people will prefer or not. Nothing like that. Today, there are many universities which take you, even if the biology background, for BBA, and then followed by MBA for hospital management, general management, sales and distribution, journalism. You are also equally eligible for BTech computer science. In many universities, they rather prefer biologists because the future is dealing with DNA, sequences, artificial intelligence and the biology, and big data and biology. So bioinformatics, not the old bioinformatics, but the new bioinformatics has plenty of opportunities all over the world. Animation, forensic science, medical technology, dietetics, electronics. So while Dr. Singh and before me told me several things about the philosophy of life, the genes, But what I am dealing is that practicality of life. Because unless you know all these opportunities, you can choose from plate now. If you see not a simple menu, it's a huge menu. For me itself, it was very difficult to compile all this and show you. Since I am now in university, it was very easy to talk to people. Particularly, there are about 500 medicine students are there. At least 150 allied health services people are there. 200-300 nursing people are there. So BSc nursing is also another one, where people go abroad every year. The nurses are the key for many hospitals, and they maintain the hospital wards, and they are so important in running of the patient welfare. Now, it's not enough. You don't have to be bothered about these type of jobs. When you have in these areas, irrespective of whether it's basic biology or B pharma, whatever it is, you can take up pharma industry. In India, pharma industry is 4.2 billion. Particularly during the COVID, it became double almost. Now it is $130 billion. It is growth is biology-based bioeconomy. It includes several areas: agriculture, veterinary, diagnostics, all put together. India is $70.2 billion worth. Now recently it has grown into $80 billion. It is not biotechnology. It is a biology-based industry. Now the entire alcohol manufacturing industry requires a lot of biology-based engineers. Now you see, this is how it is. We started with 35 billion. Now we are $80 billion now, 2021. Similarly, there is another opportunity opened up by recent on. You can have your own company. See, you are enjoying to ordering Amazon, Snapdeal. Some of these ones are only startups. They just started distributing products. Today, they are all big companies. Startup funds are plenty. Every department has money. So even with a BSc, I'll tell you one example. Long ago, 6 years ago, 3 people rang me from Bangalore. Said, sir, you are in a so-and-so department. Can you help us to use some money? What their idea was, they will make the ECG in a small mobile, and then they have got a green, red, and what we call indicators. So you can go to the village, also use it. And then it was funded, and they made this mobile with ECG, and that ECG machine was sold many African countries by Pfizer, and Merck also bought it. These boys today have a company called ECGrom in Bangalore. They make something like 2 to 3 crores of money every year. These are just BSc boys, 2 BSc biology boys and 1 BTech boy. So now today startups are also in many areas, agriculture. So this is another opportunity before you. Through biology, particularly in biology, the startup companies are increasing almost 10% per year. The money is also available up to about 60 to 70 crores. So you should not regret having taken biology at any time after 12th, in case you end up with a biology. Then I was telling you in the beginning that there is nothing like biology now. Biology is converged into several areas. It is now what we call security and defense. COVID times, it was realized that we needed a lot of people for biosurveillance. We also needed a lot of people who are sequence alliance, how to judge what is coming from outside. Now there are huge global programs on security and biosurveillance, telecommunications, automotive, healthcare. So everywhere it is getting merged. Biology is one of the integral parts of this convergence. So there is departments which are offering BSc convergence technologies, where all these technologies are together taught. Some of the ISERs like ISER Pune, first year they don't teach you anything. They teach you everything. Second year only you can make a choice whether you can go to PCM or PCB like that. So all these concepts are coming. Taking the clue, the country has launched national education policy. According to that, every district will have one multidisciplinary college on the style of ISIS. So the lot of money has been allocated. PM is particularly keen that this happens. And this mindset of, I'm a biologist, I can't do technology, I am an engineer, I can't do biology, all this completely should vanish. This is what the national education policy is going to have. So the message is very clear that the road is there. You can, as much as you can travel, you can go on traveling. And if you have any questions, please. So all this you know already, or some of it you know, and some of it you don't know. बोलिए ना कोई तो बोलो. Yes.
So you talked about a career path in bioinformatics. Can you please explain a little bit more on that?
See, bioinformatics has 4 dimensions. One is that it is used to understand the structures of proteins and the different small and large molecules, particularly large molecules. For example, the protein structure for anti-malarial drug. From using a protein structure, see, there are a lot of machines which are used, X-ray crystallography, diffraction, X-ray diffraction, then synchrotron facilities are also available. All this data is analyzed on the computer through a software for bioinformatics, through bioinformatic analysis. The second important one is that today what I am telling is the big data, that is sequences, 100,000 genome sequences are available. So these sequences are being understood, whether what type of sequences are associated with what type of disease, what type of character and traits. This is called association. All this association, but these sequences have to be put together and understand. So the Genome Center I was telling, Beijing Genome Center, it employs 300 bioinformatics only to do analysis of this particular sequence is ATGC, but it is into volumes and volumes. So that has to be analyzed. The third area is drug design. Actually, the third area is lot of work has been done, whether you can do drug design. For example, you have a medicinal plant. You have got hundreds of ginger, they say. There are 1,000 metabolites are there. So if you have a metabolite structures, you can design what type of organism will get affected from this through the bioinformatic analysis. They are called technologies called docking and all this. But unfortunately, the drug design through bioinformatics has yielded so far only 2 molecules. But the whole approach now, because a lot of antibiotics are becoming resistant, so there is a huge amount of new bioinformatics for discovering antibiotics and to combat antimicrobial resistance. So you have a BSc technology, bioinformatics in some places, diploma in bioinformatics with biology, BSc biology with diploma in bioinformatics. Good combination. Then you have MSc Genomics in Madurai Kamaraj University, where you can combine with bioinformatics. Madurai Kamaraj University, one of the top places for bioinformatics. So there are opportunities, particularly in the India has got now, we are having a meeting next week with 27 genomic companies. We have 27 genome sequencing companies. So all the genome sequencing companies, 100% employ, 50% of their employees are only bioinformatic guys. Similarly, all those pharma companies, biopharma companies, who do vaccines, who do diagnostic kits, 50% of their employees are only on the computer. That is bioinformatics. Okay. Yeah, Earth Biogenome project I didn't show you. Now there is Earth Biogenome project. I think they want to sequence several thousand of these genomes, and which is going to be also available. Which will be, who will do that analysis? It's a bioinformatics. Any other question? How many people want to do MBBS only? Still? One. It's good. It's good. Yeah, of course, you should have an ambition, because now there are number of seats also are increased. It's not from my time to now. This time, the number of seats have increased. See, there are people, if you go to the foreign services, there are at least you count some 100 people who are from the MBBS and MD stream. If you go to IAS, there are many of them. Not necessary that you restrict you as a doctor. So even after MBBS, you go to many other places. But mere MBBS today, because I see where I work, of no use. They will get maximum 50,000 rupees salary, just like IT fellows. MD, unless your clinician skill is there, you will flourish. Or else you should be able to talk like our Dr. Singh. Okay, neonatologist says, okay, small children, chalega. तो लेकिन अगर आपको कुछ बनना है इवन मेडिसिन में, आजकल मेडिसिन भी इतना कॉम्पिटिटिव है कि my daughter is doing DM neurology. That is because he want to survive after that also. So, but yet the amount of the doctor to patient ratio, WHO wants 1,000. For every 1,000, they want one doctor. So that ratio, India want to implement under the Sustainable Developmental Goals. It's a big thing. So under that scheme, the PM want to open a college every district and then more or less close the private colleges. The next plan is that. So decrease the fees, they will close themselves. So there are opportunities in medicine also, unlike in the past. So anybody else? Medicine? Who want? Yeah, yes. I'll give all the slides to some madam can collect.
So there was a part, pharmacogenomics. So
Yes.
can you explain that?
Pharmacogenomics. Actually, in my journey, what happened, we used to fund programs. There is one psychiatrist in RML hospital. There is one biologist in Delhi University by name Dr. Thelma. So these two people came together and wanted to understand whether certain drugs given to schizophrenic patients work or not. That's where I understood also pharmacogenomics. Then they found by genetic analysis that one of the drugs that belongs to a company, Pfizer, is not suitable to our population. It doesn't do anything good, but it doesn't do harm also. But it's very expensive. So they wanted to write a paper against the drug. Then the Department of Biotechnology, government, negotiated with Pfizer, and Pfizer withdrew that. Today, pharmacogenomics is more intensive because the genomic knowledge is huge. So we know which group belongs to whom. So it led to finally personalized medicine. Now today is only personalized medicine, which is earlier called pharmacogenomics. Medicine for a person.
Thank you. Thank you, Dr. Rao, for exposure. I think the subject boundaries are demolishing. And therefore, earlier it was not available in India, but I think our new education policy provides now to move and branch out at different stages. And therefore, your interest in life will go on changing, and there are opportunities for you to diversify and take the subject of your interest in future. So it's a very important development, and be prepared for it. I think I will request now Dr. Bhatt to introduce the second speaker, Dr. Prasad, who is online from Mohali.
I would like to introduce Dr. Prasad. Dr. Prasad is a BSc and MSc in geology from Bangalore University. He did PhD in evolutionary and organism biology from Janskar, Bangalore. He is currently professor at IISER Mohali. Due to some medical emergency, he is not able to be present here. He has agreed to do the presentation from Mohali. I don't know, we are unable to contact because there is no connectivity. He's online. Dr. Prasad, without taking time, I would like you to start the lecture. This is a lecture that we would like to initiate. The students always have some or the other requirement to do some independent work, project work. And there has been a lot of requests to the trust to do project work. I have requested Dr. Prasad to talk about what are the ideas in biology so that one can do a project. Perhaps the idea is to do projects without sophisticated equipment, using your things available all around. I request Dr. Prasad to give his lecture, and subsequently we will develop on this. We will have the projects implemented also, ideas, taking your suggestions and input. We will develop projects, and ultimately my idea is that one day in the near future, from these projects, we would like to publish proper scientific papers in the journals. Dr. Prasad, please.
Thank you, Dr. Bhatt. Can you hear me? Yeah, okay. First of all, I should thank the organizers for inviting me. Also, my apologies that I couldn't be there. The whole idea was that I would have more of an interactive session with the students. So being able to see the students directly and talk to them would have been very useful and very helpful, but we will try and do whatever the best that we can. Now, as you can see, I'm in a classroom. I'm a teacher, so that's my job. When I talked to Dr. Bhatt, the idea was that we would try and encourage students to develop their ideas, to come up with projects that can be implemented. We'll try and help them to do those projects and probably publish them and record them at least in a proper way. That is the whole idea. And that's what we want to do. But one has to understand that for anyone, a scientist or a student or anyone to be able to do a project, it requires some internal drive, some understanding, and many a times a little bit of resources. So I've been thinking about what is the best thing that one can do if one has to do certain projects. In my own belief is that if you have to do projects, first of all, you have to be very comfortable with asking questions and answering them yourself. You have to be able to play around with things and figure out answers for yourself. And that is not something that one can do easily, again, because it comes back to whether we have the resources to do such experimentation. So what I'm planning to do today is spend most of the talk in trying to introduce you to certain resources that each one of you can use to have fun with genetics. Ask your own questions, answer your own questions, and generally learn about genetics and have fun with genetics. At the end of it, I want to introduce, especially your teachers. I see many students and teachers in the audience. I want to introduce your teachers to resources that the Indian Academy of Sciences that has come up with, which can be utilized by your teachers in your schools to help you develop projects. Of course, we are always there. At the end, I will leave contact details which can be used to further this dialogue, and as Dr. Bhatt said, come up with more ideas for projects and implement them. Okay. So basically, a lot of ideas about resources about having fun with genetics. That's really what I want to do. Now, when you think about projects, one of course is the research projects. The other is projects to learn. Projects to learn are projects that you create for yourself, work on them at your own spare time, and have fun with them. So that is the focus of at least the most of this talk. So why do all this and learn genetics? I have learned over time teaching students that when you teach genetics and when you learn genetics, it becomes almost rote like any other subject. You're given different names for different phenomena. You're given different studies that historically people did. You're given the results, and therefore you learn about it. Half the fun goes away. I mean, it starts from the very fact about once you ask students, what is genetics? The most common answer that you get nowadays is the study of genes, which is true. But that's not how it started. Genetics, in its most original form, was the study of heredity and variation. It answered a very simple question that was very obvious to everybody, starting from the very, very ancient humans, that progeny look like their parents, but not entirely. The question is why? You look like your parents, but not entirely. You looking like your parents is heredity, not entirely part is the variation. Answering this is essentially, or was essentially, the point of genetics. To put it another way, to begin with, genetics was a science of phenotype, not of genes. It's not to say that it's not about genes. I'm saying that to begin with, it was the science of the phenotype. You look at something, you look at what its progeny looks like, and you infer something about heredity and variation. That was the whole idea. And that is the idea, again, that I want to push when I'm trying to teach you how to have fun with some of these resources that I'm going to show you for genetics. Now, again, one last point before we actually get into having fun, which is genetics comes in various flavors. Of course, the one that you have learned very commonly in your schools is that of Mendelian genetics. Some non-Mendelian genetics, but Mendelian genetics is essentially what you have learned. Early 20th century, there was a great advancement to Mendelian genetics, extending it to entire populations, and that became known as population genetics. Quantitative genetics followed. You also have evolutionary genetics, and of course, you have more genomics nowadays. So genetics comes in various different flavors. So what I'm going to try and do is I'm also going to try and show you some resources where you can have fun and learn these genetics concepts, have your own small projects, do them quickly, and get into the habit of running projects so that you can do other projects at a later stage. Right? So essentially, Mendelian genetics, population genetics, evolutionary genetics, those are the kinds of resources that I would like to show you. Now, these resources, of course, can be live organisms. Working with live organisms is always exciting, fun. You get to do a lot of things. It takes a lot of time. It also takes some resources. For example, the most famous organism that a lot of us work with is Drosophila melanogaster, the fruit fly. But growing the fruit fly, maintaining it, doing crosses with it, needs some basic setup. You need to maintain it at least in some containers. You need to give it some food. When you have to work with it, you need to have certain basic things like some way of anesthetizing it, either using ice or carbon dioxide or some other way. Some resources are required. So working with real organisms is fun, but requires some effort. I'll give you about those resources slightly later on. The other resource that has become very, very useful, and its usefulness has been sort of certified over the last 2 years of the pandemic, are online resources, softwares. One can work with these resources and run small projects. That is also possible. And I'm sure a lot of you have access to phones, smartphones, And many of the softwares and resources that I'm going to show you can be run on those. Also, you can run these online projects using these. Certainly, schools which have access to computers can help you run these much more easily. But you can run these on small phones also. I have tried these, which is why I've chosen some of. Okay, so let's start with having some fun with some very basic Mendelian genetics projects. Okay, very, very basic Mendelian genetics, right? We all have learned. One of the earliest experiments that he ran, or the most famous in that sense, was what was called the monohybrid cross, right? You take two pure breeding organisms, let's say something that has a red flower, and cross it with something that has a white flower. I'm making this all up, but the whole basic idea I'm sure is clear. You get, of course, red flowers, and then you take this and you self it, and you end up with something like a red flowered plant and a white flowered plant, and these ended up in some ratio. And I'm sure you learned this, and then you were told that 3:1 is a very characteristic ratio of a monohybrid cross. Right? In reality, it turns out that these are a little more complicated. I've always had this question, you know, how, as a student, I've always had this question. I'm sure many of you also had this question. How true is this? I mean, do I end up with this? Always happens like this? Can I do this? Of course you can do this. You can do this if you had real organisms. What if I did not have real organisms? Can I do it quickly, fastly? Make up my own projects about genetic crosses using something else. Now I'm going to try and share screen. I don't know whether I can do that. Yeah, I can. Thank you. Here is a very simple online resource. Right? It's called sciencecourseware.org, and there is something called a fly lab. This is literally an online fly lab where you can have small projects that you can run on your own. Again, remember, the goal is to have fun, learn genetics so that you can do other projects at a later date. Here is, you know, Drosophila, a male and a female. Let's try and figure out, okay, let's say I want to do a cross. There are many, many different characters that I can choose. Let's say I chose the first one, bristles. Bristles are these hair-like things that you find on the body of Drosophila. Okay. Let's say I chose some bristle. Let's say spineless bristle. Or something more obvious, like various kinds of antennae, or even more obvious, some kind of eye colors. Let's say sepia eye colors. So I've chosen, just by clicking on this, now I have a fly that has sepia eye color, and that happens to be a female. Okay. Now that has to be mated with a male of a certain kind. Now let's take a male that is completely wild type, right? It has all wild type phenotype. Let's do that. See, this is a typical monohybrid cross. Both of them differ in only one character, which is the eye color. Let's say I'm doing the parental cross. Here is the result of your cross. So these are the progeny, the F1 population. Unlike in plants where you can actually self them, you cannot self Drosophila because it's a sexually dimorphic, it's a sexually reproducing population, right? So you have a female and the male. The best that you can do is you can have symmating, right? That's possible. More importantly, the point I made about genetics being a phenotypic science, all that you know from this cross are the phenotypes of the parents and the progeny. And the phenotype that you're interested in this case, is the eye color. So this is sepia, red, red, red. So this is the F1s. So we can do a F1 cross F1 and look at what happens in F2. Here it is. The red eye, red eye, red eye female, red eye male, sepia-eyed female, sepia-eyed male. And they occur in certain numbers. Right? Now, if you look at these numbers, they're not really 3:1, right? 394, 376, 118, 129. If you actually calculated a ratio, they're never going to be 3:1. So how do I handle this? Is this therefore a monohybrid cross? Is this not a monohybrid cross? Well, I could do an analysis. I could ignore the sex of the flies. I could include a test of the hypothesis. You handle the data statistically. It is said that one of the greatest reasons why Mendel succeeded the way he succeeded was the mathematical quantification of his results. He just did not note, white flower, red flower, etc. He also noted how many of those. And that mathematical quantification and the statistical analysis becomes very important. Now I could come up with the hypothesis that I want to test. Okay, so hypothesis I'm saying is 0.75 and 0.25. This is, I hope you recognize, is another way of just putting a 3:1, right? Now I can test my hypothesis. The question I'm asking is, right, these are the observed numbers of the wild type phenotype and the mutant phenotype. Are they in this ratio of 3:1? I can test my hypothesis. If I test my hypothesis, there is something called a probability. The higher the probability, then you are saying that yes, there is really no difference between what you are observing and what you would expect. Not just this. So this is one type of cross. You could design any kind of fly, as many different varieties that you want, and you could test this. Why am I showing you this? This gives you certain basic ideas about genetics that go missing when we actually learn genetics in class, which is the quantitative nature of genetics and the inherent variability that is there in this ratio that you see. In any future projects that you will do, quantifying things, handling those numbers using statistics is very, very important. So such software will help you run these experiments in a very short time, fairly easily, and will help you learn about how these are analyzed. Now, one can ask the question, if I ran the same experiment again, will the software give me the same numbers? So is it something like that is fixed? So if you take this phenotype and cross it, the software will always give you the same number? No. The software will actually give you different numbers different times, but you can always test whether in each of those runs your observed and expected ratios were different or not. So what we just saw was something very, very simple. I can make it very complicated, right? For example, we could take eye color, right? And we could also take something else, right? Let's say some other bristle. So you have a fly that is variant in 2 body characters. And then I can mate it with another fly, which is completely wild type. This would typically be your dihybrid cross. Right? I can ask what is the phenotype of the progeny and note down what are the numbers. In this case, you see that both the progeny are wild type and are equal in numbers. And I can do F2 cross. And of course, I have different phenotypes in the progeny, and they come in different numbers. Right? If I see carefully, this seems to be wild type. This seems to be having spineless bristles, but red eye. These have sepia, but wild type bristles. These have both sepia and spineless bristles. So you have 4 different phenotypes in different numbers. Now, again, I can ask the question, does this show a certain distribution? Right? I can ask whether this follows a 9-3-3-1 distribution. You could do that. That is possible. Right? And this software allows you to make more and more complicated things. I'm sure you have heard of things like mapping, recombination mapping, mapping distances, linkage mapping, all these. Even those can be addressed using software like this. Right? This is one software that I have extensively used to allow students to have fun with genetics. You can use this, you can run it on a small a phone that you have at home, and you can have a lot of fun with this. What kind of projects can one come up with this? Is this a serious project? As I said, it is serious for you because you are learning, using that project to learn something about genetics. You can make hypotheses about the inheritance of various characters and test out those, and that is your project. Right? So you can do that. So you will get lots of such resources for Mendelian genetics. What about something slightly more? I don't know how many of you have heard this story. Early in the 20th century, 1900s, early 1900s, many people were looking at traits in humans that were inherited in a Mendelian way. And one of those people who was looking at human traits that were inherited in a Mendelian way was this very famous scientist. I hope you have heard his name. His name was Punnett. Most of you would have heard his name Punnett in the context of the Punnett square through which you are taught genetics, right? Which his name is associated with that, but he did a lot of other very interesting things. And one of the things that he discovered using pedigree analysis in humans is something about a condition in humans in which the fingers become slightly shortened. It's called brachydactyly. So what he figured out by analysis was if you took an individual who had brachydactyly and you had an individual who had normal fingers, their children tended to have brachydactyly. So he said, after analyzing a lot of such pedigrees, he said that, maybe brachydactyly is a dominant character, whereas having normal fingers, normal sized fingers, is a recessive character. Again, normal, dominant and recessive is something that I'm sure you have heard. When he was giving a talk about this, I'm told that somebody asked a question, or at least he misunderstood a question when he was giving a talk in the Royal Society of Doctors or Royal Society of Medicine. And the question was to the effect of that, look, if brachydactyly is dominant and normal is recessive, then if you did a cross like this, in the F2, you will get 3 brachydactyly individuals to 1 normal individual. That is the phenotypic distribution. So 3 brachydactyly to 1 normal. Now, if that is true, then is it possible that after several thousands of generations, in the human population, you will have 75% individuals with brachydactyly and 25% with not? This was apparently the question that was asked to him. At least that is how he understood it. People argue that maybe that's not how the question was asked. And he started thinking about it, and he did not really have an answer. The reason being, the approach that we are taking here in the entire Mendelian genetics is that of family genetics. Parents, their progeny, follow their phenotype, follow the numbers, etc. But the question that was being asked to him was about an entire population. In the population of humans, will 75% become brachydactylous and 25% become normal fingers? So it was a huge jump. The person who helped him make that jump was a colleague of his in his university who was not in his own department, but he knew him because they played cricket together and they were on certain committees together. And the name of this person was G.H. Hardy. And we Indians know him for a different reason, as the mentor of a very famous mathematician, right, Ramanujan. The same Hardy helped him solve this conundrum, and in doing so, he established a whole new field of population genetics. And I'm sure you are taught some population genetics, or you will be taught population genetics in your 12th standard, something about the Hardy-Weinberg law, etc. And the jump from Mendelian genetics to population genetics is not simple. So therefore, again, it is interesting, it is important to be able to learn it hands-on by asking your own questions, making your own hypotheses, and answering them. So the next resource that I'm going to try and show you would be a resource that will help you run little projects on population genetics. Okay. So let me share screen again. I hope you can see it. There is this little window, right? Okay. So this is a little software called Allele A1. Again, important to note, all these are free softwares. You can directly download them. You can play with them. Some of them are available online, so on. And this is, none of these are made by me or anyone related to me. These are again made by various people around the world and put online so that students can have fun running little projects. So when you're talking about population genetics, you're talking about populations and allele frequencies and all that. And I'm sure many of you who are at least in the 11th, 12th standard have already learned the statement of the Hardy-Weinberg equilibrium, which says that, okay, if you have large random mating population with no mutation, migration, selection, etc., the allele frequencies and the genotype frequencies will remain a constant. Right? I also mugged it up. And I'm sure many of you have also been taught a certain mathematical formulation of it. And when I was taught it, I was always interested. And I was always a little confused as to why am I being taught a plus b whole square at this age. I did not really understand it till I started running it myself as a little project and understood it more and more. So what this helps you do is it helps you, this software at least helps you look at how allele frequencies change or do not change over time in a population. So this is a default setting, right? You have a certain frequency of an allele. Here is an allele A1, which we are interested in. And there are certain default settings that are shown here, right? Basically, it is saying that there is no mutation happening. There is no migration happening. There is no selection happening. There's a large population size, so on. So if that is the case, and if the allele frequency to begin with was 0.5, what happens to this allele frequency over generation? So what you have on this axis is the allele frequency. What you have here is the number of generations. So we can run it and see. See, if you run it, nothing changes. The allele frequency remains the same. Now I can change the allele frequency. Maybe if it is 0.5, it won't change. What happens if it is 0.2? Let's run it. Well, nothing happens, right? It remains the same. What if it is 0.9? Nothing happens, right? So irrespective of what your initial frequency was, if you have certain assumptions met in population genetics, your allele frequency will not change. Well, what if certain things are not met? What if I change what are called fitnesses? Suppose I say that, okay, fitness of 1 is high and the others are not so high. Let's say this is 0.9 and let's say this is 0.8. Okay, let's run it and see what happens in this case. You see? I can run it again. So the moment I have differences in fitness, then the allele frequency is changing. The genotype that has the highest frequency and therefore the allele with the highest frequency increases in number. So this is all that this, at least in this run, I am trying to show. You could do many, many things, very interesting things. For example, I have one here. Let me make it, okay, 0.7. Maybe I'll make it 0.7.
Dr. Prasad, can
Right.
you hear me? Dr. Prasad,
Yeah, yeah,
can you hear me?
yeah, I can hear you.
Sorry for interruption. We are running behind schedule.
I will wrap up in the next 5 minutes. Is that okay?
Yeah, I think.
Yeah, I'll do that.
There's another session.
Yeah.
Sorry for this inconvenience because of
No
the
problem.
deadline.
No problem. So we could do this, and there are many such little projects that you could run for yourself and learn. There is one which I'm just going to show, and this is going to, some of this is actually used for running hands-on projects, even as research projects, even we do it. There is this thing called the Avida Ed. This gives you the genetics of, just think of it as bacterial genetics. So you have a circular genome, and you can use this, again, a software. You can run it on your phone for learning many things in genetics and run small projects. Please, important to remember, each of these things that I've shown you allows you to run small projects and learn genetics. Okay, and I'm showing you software so that these things can be done very quickly. I also promised that I will show you something about actual hands-on animal experiments. And this is again more for your teachers. Let me try and bring it up. Yeah. Now, this is a resource that has been published by the Indian Academy of Sciences. This is an e-book. Anybody can freely download it. Please note down, it's the Indian Academy of Sciences, Bangalore. If you go to their website and look for e-books, you will find this book. It is free for anyone. And it gives you a large number of little experiments that you can run, a large number of small projects that you can run in your own schools with very little facility that you might have. Okay, there are about 85 or so various experiments that one can do. There are other small experiments that we have done with students in our own institution. For example, there is something called propyl thiouracil, an equivalent of PTC. It's as simple as tasting it and getting your entire family to taste it. And note down, what was the taste? What did you feel? And with just this one simple question, you can unravel a whole host of very interesting genetics. It can become a very interesting project. The other kind of project, again, that we have run in our own institution, just by collecting data, not doing any experiments, very little resource required, is collecting the heights of everybody in a family. So all our students go out and collect heights of themselves, their parents, their grandparents, uncles, aunts, nephews, nieces, etc. You can use just this data and learn a lot about another branch of genetics that I talked about called quantitative genetics. Okay. All these, of course, have to be a little more nuanced than the software that I showed you, and that's where I'm sure your teachers can help you. Any of you who are interested in running hands-on projects, of course, you can contact Dr. Bhatt, and through him, me. We'll be happy to help you out in designing and running projects. And hopefully you will have a lot of fun learning about genetics while learning those projects or running those projects. Okay. So thank you all, and thank you, Dr. Bhatt, and the organizers for having me. And sorry for not being able to travel there. Thank you.
Thank you, thank you very much. I'm sorry we have to interrupt him, Dr. Prasad, but we are really running short of time. But the message from you is very clear. The teachers, if they are in the students, they can certainly contact Dr. Prasad and have online demonstration of various genetic experiments in their classroom. That's the message. Thank you, Dr. Prasad. Thank you very much, and thank you, Dr. Bhatt, Dr. Rao, for your presentation. I think both the presentation, the way we are organized for the students, basically, I think the purpose is served. But if it was a little more time, we could have been a little more relaxed and could have gone into greater detail. What we have been doing through Genetics Congress, just 2 minutes, I will take only 2 minutes, and then we will have some prize distribution for the students. We had requested them. One was that we are organizing a regular teachers training program on the subject matter which given in NCERT book. We take the teachers to the best labs available in Delhi and get the experiments done in 2 days, Fridays and Saturdays. Then to the students, we are going to the schools and providing them lectures, etc. In addition to that, our colleagues, Dr. Firoz and Dr. Bhatt from this institute have developed material in maze. You can take those maze scopes and do the counting of the segregation of different characters and then demonstrate to the student in the class itself. And those scopes we are trying to multiply and give it to the teachers to be used in their school. These are the kind of things which we are doing, and I hope that these efforts from the Genetics Congress is going to help to understand genetics and genetic principles in greater detail. And thank you very much. And I request now Dr. Bhatt to put the next project distribution.
Thank you. We come to the end of this session. I'm sorry we had to rush through the project, the program, due to shortage of time and some uncertainty about Dr. Prasad joining us because of some medical reasons. I really thank Dr. S.R. Rao as well as Dr. Prasad for sparing time to be with us. Most importantly, because of again truncating the whole thing, we did not properly welcome the students. This is the first time, in fact, after the long period of COVID, that we had the opportunity of inviting students, school students, and perhaps, as far as I know, this is the first time in a scientific conference we have carved out a special section for school teachers and students. I hope in future National Symposium, we will maintain it as a tradition to have interaction with the local schools and teachers as an integral part of any symposium. Because if we have to attract people into various areas of branches of science, interaction with the students is essential and indispensable. So this is one thing that I would like to say, and I thank you profusely to all the teachers and students who have come forward to our invitations. In fact, we had many teachers and students coming because of the space and other constraints. We are restricted, and we also asked the students as a incentive as well as a deterrent that they have to send us some cartoons, pictures, poetry, whatever about biology and Mendel, and we have received more than 50 posters which are displayed perhaps in the poster section, which you can see, and we have judged them, and we are announcing 5 students who have won the award. The first prize goes to Divya Hajra, class 12 student from Salwan's Girls Senior Secondary School, New Delhi. Yeah, if you are there, you can stand up. I'm just announcing the award now. The award will be given in the next session in the valedictory. At least those students who have won the award can stay back if possible, because I know that they will have to go back because they came to school in the morning and their parents must be waiting. The second prize is to Khadija Joya Kwamar, class 12B, Carmel Convent School, Chanakpuri. The third prize is to Sanya Dhingra, class 12, GD Goenka Public School, model town. Then the consolation prize is to Hebsiba Rezi, class 12B, Kerala School, Vikaspuri. Because we cannot award to everybody, and I should say that the efforts they have put in some of these is enormous. Their skills at drawing or composing with poetry, because we started with the poetry, you remember, Anuvanshiki ki Pratham Sopan. So we have also poetry. In fact, I thought that we should ask them to recite, but there is no time. Because of that, we are not able to do it. But thank you very much for your participation. We would like to help you in developing this project of idea of projects in the schools, for which we will again organize separate sessions from the Genetics Trust Club Trust in the future to work on this. With these words, I thank the speakers and the organizers for giving us this opportunity. Thank you very much.
You want to say something?
No, no.
Thank you, sir. I would now request our organizing chairman, Dr. A.K. Singh, to kindly come onto the dais and do the honor of felicitating the chair, co-chair, and experts of the session.
So, dear students, before you go, Dr. Rao mentioned about one Mr. X. Can you guess who the Mr. X is? It's Dr. Rao.
The students and teachers, we request you to stay outside. We would like to have a group photograph. Just 5 minutes. We are part and parcel. We also have to be Dr. Prasad, are you there? No.
No, it is good. Okay. Okay, I think with this, thank you very much. We closed the session. We are really late, but it was beyond our control. We started late and we tried to finish it within reasonable time. Thank you very much for the projects. Thank you.
So 2 minutes आएंगे हाँ
Let us now break for tea and we'll reassemble after 15 minutes.
Welcome back, everyone, and very good evening. Moving on to the second special session, Plant Breeders' Right, I'm delightful to begin the session by inviting our Honorable Chairman, Dr. K.V. Prabhu, and Co-Chairman, Dr. D.K. Yadava, to grace the dais. I would like to welcome the conveners of the session, Dr. Sujata Vasudev and Dr. Shailesh Tripathi. I would also like to welcome the reporters of the session, Dr. Ranjit K. Ellut, Dr. Ramaprasad, and Abhijit. May I now request our director, Dr. A.K. Singh, to welcome the chairman, co-chairman with a bouquet of flowers. May I now request Dr. Shailesh Tripathi to introduce the chairman.
Good afternoon to all of you. It's my pleasant duty to introduce the chairman and co-chairman of this session. I welcome you, sir. Dr. K.V. Prabhu, sir, is known to this audience. He was educated at Banaras Hindu University. He did his graduation and post-graduation from BHU during 1975 to 81. He completed his PhD from Indian Agricultural Research Institute between 1982 to 86. He was the chairperson of Production of Plant Varieties and Farmers' Right Authority, Government of India, from 2018. He started his... Sir. He started his career from IRI. He was scientist at the regional station Shimla from 1986 to 91, senior scientist from 1998 to 2006, He was officer in charge of the National Phytotron Facility between 2002 and 2006. He headed the division of genetics from 2006 to 13 and finally rose to the position of Joint Director Research IRI from 2013 to 18. He has many awards and honors to his credit. ICR Jawaharlal Nehru Award in 1987, BP Pal Award in 2003, VS Mathur Memorial Award in 2004, Borlaug Award in 2012, Rafi Ahmed Kidwai Award in 2012, Nanaji Deshmukh Award for Team Research in 2018, and Atal Ratna Award in 2018. He is Fellow of National Academy of Agriculture Sciences and NASI. His research areas include plant breeding, genetics, and marker-assisted selection. Now, I would also welcome the co-chairman of the session, Dr. D.K. Yadav sir. He is also known to this audience. He is currently holding the position of Assistant Director General Seeds in the Crop Science Division ICR. Prior to that, he was Head Division of Seed Science and Technology IRI from 2014. He was Principal Scientist in the Division of Genetics from 2009 to 2017. And he started his career as Assistant Professor in Plant Breeding and Genetics from Rajasthan Agricultural University, Agricultural Research Station, Ganganagar. He has many awards and honors to his credit. He won the A.B. Joshi Award in 2022, Rafi Ahmed Kidwai Award in 2017, NAS Recognition Award in 2017 and 18, BP Pal Memorial Award in 2012, and many other awards. And he is fellow of many societies: Indian Society of Genetics and Plant Breeding, Indian Society of Forage Research, Indian Society of Oilseed Research, and he has developed many varieties of rapeseed and mustard, and which include the 00 varieties. So now I request Dr. Prabhu sir to take over the session.
Thank you, Shailesh. Most respected Professor R.B. Singh, Director of the Institute, Dr. Ashok, my co-chairman of this session, Dr. D.K. Yadav, and my very close friend of IRA, and one of the closest friends right from my early career here, Dr. S.R. Rao, Dr. V.B. Singh, and many other distinguished colleagues here who have stayed back despite the late evening, and the students. I'm going to target the presentation more for the students of plant breeding or people who are involved in developing materials in horticulture sciences. I hope this is what I requested the organizers to attract for this particular session, because it's good for you to know what is it that you have as a plant breeder, whether it is crop species or horticultural species, that you are in for in international perspective and competitive trade. You have to follow in any case if you become a breeder in your profession. Since you are training to be a plant breeder or crop improvement person, you would like to know what are that you are in for, which cannot be avoided as the times of intellectual property possession and exploration of how best you can get your intellect converted into business has to be done with the plant varieties. You have to do it. So there is no option as plant breeding perspective go in, and as the new Seed Act is likely to be on the anvil soon, and then it might get effective. In that case, you have to know the compulsion which are required for you to know that how important it is to be associated with the ownership of a variety, so that whenever there is any claim, counterclaim, and compensation demand, accordingly you have to act. So you should know what is it that we have in the Act of Protection of Plant Varieties in India, and how it is necessary for each of us to be keeping that in perspective. So I'd like to go ahead with I'm sorry for the poor visibility. This is a statement of Johann Mendel. Okay, Johann Mendel is the name of Mendel as he was born. It had, I'm not able to read it well.
it had become impossible to continue such strenuous exertions. It was incumbent to enter a profession in which would be spared perpetual anxiety about means of livelihood. Private circumstances determined the choice of profession. This is what Mendel wrote in 1880, son of a struggling farmer, and he became a monk only to be able to feed himself and not to struggle to have his livelihood and food met at the age of 22. This is what it was. So you can imagine how connected the science of genetics, which he was to unravel, is to overcome the problem because of which he became a monk. He himself has written it in his autobiography. This was recorded in 1943 by Hugo Iltis and published. This is a reference for that. If any of you want to read it, you must go through it. It's a wonderful account of Mendel, other than the genetics which we have seen and the controversies related to what he had with Nageli and other company, which you have heard in the discourse here. Friends, it was a big surprise to me to realize till 1958, there was not one definition which was internationally accepted for plant variety. Okay, this is something this is why I wanted to begin with this. And finally, what is accepted by international community as a variety is this: an assemblage of cultivated individual, which is distinguished by any character, morphological, physiological, cytological, chemical, or other, significant for the purpose of agriculture, forestry, or horticulture, and which, when reproduced sexually or asexually, retains its distinguishing feature. This is what is technically a plant variety. So this was made possible only in 1958, although India had started developing variety. First variety in India was released in 1908 by Dr. Howard in wheat, and that is when the first variety was released, but definition of variety was very flexible. And since we are going to talk about PVP, plant variety protection as an intellectual property, you must also know what are the other intellectual properties, because some of them are coexisting and can be concomitantly held on a particular product. So first is the Trademarks Act and Patent Act, which most of you may be hearing about, because patent is something that is looked up as something great achievement by any individual. So you might be aware of patent. But there are other acts too. One is copyright, which you might have seen as a symbol of C in your textbooks, which are not supposed to photocopy or things like that. Then the Designs Act, the Geographical Indications Act, very important for plant breeding perspective or plant community perspective. Those which are maintained, nourished by farmers or communities. It's a community right, not individual right, community right. It cannot be owned by one individual, the GI. Some of them are the famous GI from IARA is Basmati rice. The community of Basmati farmers hold it. Of course, you may know many other GIs if you have heard of. Darjeeling tea is one, Assam tea is another, many. The next is the Semiconductor Integrated Circuits Layout Design. This is in terms of layouts and circuits, and Protection of Plant Varieties in Farmers Act and Information Technology Act. These are the legislations in India which have empowered an individual or a community to claim its intellectual right on something they have either invented, discovered, or maintained, or conserved. So this is what the system is. And PVP is the only act among all the other acts which are away from the Ministry of Commerce. All others are maintained by the Ministry of Commerce. Only this is away from Commerce Ministry, and this is an Agriculture Ministry. And this is the only act or only intellectual property perspective which is done on the basis of evaluation we do and not on the claim made by the inventor. Okay, all other rights on intellectual properties are based on comparing with the existing information over which an inventor says mine is different. So you simply compare by the claim and available information. These are the only two things done in all other rights. Whereas in the PVP, we actually do research, identify which are to be used as comparators, and go closest to the one which is being claimed as a new variety by a breeder, and then see whether it's actually new or not by generating data for 2 years in 2 locations, just like any other coordinated trial business. So this is the only intellectual property which is awarded only after trialing is done. So that's one other difference. That's why it is different. It is away from the other intellectual property related regulators. And this is one authority which is also kept in Agriculture Ministry because it's technical. The rules of the game are technically managed by understanding the science of plant breeding, because the claims are interpreted by whether it's a single cross or a double cross or a composition or a synthetic population or a land race. The claims are made like that. And whether if it is that, somebody will say it's a back cross, somebody will say it's essentially derived variety. Whether the data perspective is matching with the claim made, and whether there's a historical record to say the claim is correct or not. Things of this kind have to be seen with an understanding of what is the meaning of an essentially derived variety or back cross progeny or things like this, which is why it's required to be a plant breeder to understand the interpretation of the act as provided. So that these are the exceptions to make it very unique act. And there's another important factor which all of us should cherish with pride. Among all the 196 countries which trade globally, of which 179 as on today are signatories to the different regulations or different agreements and different international conventions which exist as signatories, including the International Treaty on Plant Genetic Resources for Agriculture, which is done by FAO. We are the only country, including all developed countries put together. India is the only country which has complete compliance to all the three commitments which all of them have signed but not followed. So India is an exceptional country in that perspective, which has signed ITPGRFA, which has signed WTO, which has signed the TRIPS, and has been compliant to all the three, while all of the other 178 countries who have signed all the three, out of the 196 countries, the other who are enabled to sign before 2030, by that time most of them will have to sign. If they don't sign, they will not be WTO members, which means they will not be able to trade in the global trade community, even if it is related to varieties. I will tell you what is the advantage or what is the requirement and how plant variety protection related right and legislation for it is most important among all the IPRs to be member of WTO, which means to be a member to trade with your product without any hassles. You have to have Plant Variety Protection Act in your country. The one which is globally used, because this existed before the WTO came in and before the plant variety right related legislations were enacted in most countries. This was a conglomeration of all European countries to reward their plant breeders or the plant variety producers, claim on their rights of the variety being their own, and when somebody uses it, they get a benefit out of it, either as seed or as a material for further breeding, etc. So they were the ones who did it first. So WTO adopted that as the base to TRIPS adopted that, because the TRIPS only WTO also came in. So historicals are very finely tuned here with the World War II. That's how the sequence of events happened to get up to 1994 when the TRIPS and WTO got in place. So this UPOV is the name which is given. When you read it in English, it doesn't translate, but if you read it in French, it will translate. So it's a French terminology which makes it as an international union for protection of new varieties of plants. This is an importance. It is new varieties of plants. So you may ask, what is new? New varieties of plants. Why the term new should be there? Why not only varieties? So that's another important factor in intellectual property. Timing, existence of the material in trade before you stake a claim that it is yours. This is the dicey situation. You cannot make a claim if it is not there. An idea cannot be patented, protected, copyrighted. It has to be there. People have to see it. How you did it and what is in there is the one that you want to hold as your intellect. But it has to be there. So it means it has to be there, but it has to be there not so old that it becomes public domain. Everybody knows about it. Something of common knowledge it becomes. Then it cannot be patented. So there has to be some deadline after which you cannot make a claim. So plant variety protection internationally is only new varieties protection, which also immediately tells, one of my friends was here showing me the pictures of mango, which he has in his orchard, which is maintained by 6 generations. Those are irrelevant as far as UPA is concerned. The age-old systems of maintaining the old varieties which have been there, the langdas of mango, or all the old Alfonsos, all those cannot be protected today, and that is public domain. Anybody can make use of it, anybody can make money out of it by selling the saplings and trees and those things. That is what is international community. Had India agreed to it, had India joined this, when WTO got framed, that was the easiest job to do. And UPOV is existing. Their offer was either you join UPOV or have an act sui generis, that is on your own, generate it on your own as required by the country, and then accordingly join it. So India decided to do this. Let me go ahead. This is another important factor which all of you should understand. What are plant breeders' rights? This is a passport for development. I had another slide where I had noted in 1700 BC. Sir, this is something you may also like to know, Professor Arun Kasingh, sir. In 1700 BC, it is recorded in international history of the world. It is mentioned in India, if anybody used a particular rice material for propagation purposes, he would be hanged to death. So intellectual property on a plant variety of rice, which if propagated with the permission by somebody else, a particular dynasty saw that that person or the people who did it were killed. This is noted in the intellectual property related historicals that as existed in India in 1700 BC. You can imagine, I tried to look into the historical behind it. I didn't find any document other than that document alone. The slide was there. I don't know how I got missed it, but that is what it is. Which means in India, an understanding like we stake a claim on zero and now Pythagoras, all of it, even Newton's law is now being claimed as originating from India. Now we can also claim intellectual property right claim with a criminalization related penalty existed as far behind as 1700 BC. So that is something of importance, and this is recorded in WTO and WIPO. It is recorded there as an existing literature. So that means something which is very important for us to realize, that possession of variety as possessive of ownership of a person or a community existed in that period also, very close to civilization period. So plant breeders' right is a passport for development of a nation. The topic which I have used here is the IPR on varieties is key to societal value enhancement that is coming from here. Whether it is agriculture or not agriculture, that is a mainstay occupation of a country. Like a country may have only less than 2% population dealing with agriculture there. And agriculture is the power drive of vehicle business success in country other than the OPEC nations. In every nation, whether agriculture is the mainstay or no, this is a requirement that they can be there as developed country or any country if they have plant breeders' right. Development of new varieties, production of distribution, sale of new varieties is successful agriculture, for food and trade of a country. That is of course true, because you cannot say produce more than what you require to trade if you don't have good varieties to produce that. And agricultural success is the foundation of industrial base for any country. You want to check it out? Do this. Every developed nation, including those with less than 2% population in agriculture, with 100% private funding. Okay, those 2% are there with 100% private company participation only, as the technologies or products for use or inputs. Has public invested high-tech advanced research in and education setups in agriculture, and plant breeding and genetics as the mainstay there. They don't have agriculture as the main occupation of the public. and they don't deal with products which are used by farmers. I mean the government, the public sector does not, and yet they invest hugely. You must have heard, all of you who are in PhD or post-graduation would know which are the best institutions of agriculture research. All the developed countries, there is no agriculture as a mainstay other than Australia, for an example. But every other country, which is to which country most of you would like to go and do your postdoctoral or doctoral studies, are with advanced research systems in the public system, but the products and the agriculture itself is dealt with private system. Why do they do it? Because they know that is the basis for their country to be developed. Today, Sri Lanka's crisis is fundamentally generated by agriculture failure. Why it failed? Because of a policy, there's a different matter, but it is fundamentally agriculture failure in 1 year. It's only 1 year's failure created the crisis and the chaos we are seeing in Sri Lanka today. So that's how important agriculture is. And why did TRIPS agreement on agriculture? It's becoming very difficult to. TRIPS agreement on agriculture at Marrakesh connect plant breeding right and WTO membership. There's a history behind it. 1944, after the World War II, United Nations found most countries are unable to trade their commodities because there is either allies or other group, and they didn't want to accept some other product, which you are seeing even today. And then there were only about 43 countries who were members, and they were finding that people are not trading or giving goods which they require because of this embargo which was there in existence. So decided to have an establishment of World Bank and International Monetary Fund and ITO, International Trade Organization, put together to facilitate funding those countries which are suffered because of World War, so that they produce their products, not just agriculture, any products which can be traded, which are the raw materials are available, but finished products are not being possible for other countries to have access to, which they don't have, but they can afford to buy, but they don't have. So in order to facilitate this, they decided to have these establishments. In 1947, the World Bank and the ITO created GATT. Okay, GATT is the one, that is the General Agreement on Tariff and Trade, was the one which got converted into WTO. So eventually, after about 40 years, it got converted into WTO after another agreement, which was TRIPS agreement. That is the trade related aspects of intellectual property rights. So that means WTO intellectual property right are directly linked for any country to become a successful country. Successful country, developed country is only based on the basis of the financial resources they are able to generate, and that is by trading, and therefore it's all linked. The IPR, trade, successful country are linked together as one continuum, and TRIPS is the basis for that. That happened. And since from the plant side, since plant plays the role of ball of a game, of a football or something, and farmers are the players, and the ground, of course, is the field, like the field of football, the creation of varieties become the most critical factor. If the ball is not there, nothing can be kicked around and nothing can be scored. No goals can be scored. So plant varieties become the game ruler for a game creator for agriculture. That is variety. Minus variety, all other things put together, you cannot do anything. That's what happened even in Sri Lanka. There were no varieties which were able to have early ground coverage because they had stopped spreading weedicides, herbicides. Weeds occupied more than the crop varieties because crop varieties did not have that ability to spread wide and cover and prevent the weeds from coming up naturally. So weeds took over and farmers could not get yield as good as they were getting earlier without any herbicide application, things of this kind. So varieties suitable and varieties required for a particular practice, particular system is the key for successful agriculture. Legislation on plant breeders' right via TRIP compliance is the only gateway to WTO of any country. If the point is, if any country did not enact plant breeders' right in their country, this is what the TRIPS Article 27 says, then they will not be retained as members of WTO, depending on their economic status. Some countries were asked to be members of the members of WTO by passing the enacting. Some had already enacted, some to enact and have a regulatory system for plant breeders' right. In 1995, they were all ultra-developed countries. Countries like India were given option up to 2000 December 31 to enact or join UPOV. Choice was only these two. Either India joined UPOV or India enacted. These were the choice. That's one part. So if you don't do it after 2000, we will not be WTO members. I'm not talking about seed or agriculture. You will not be WTO member for any other trade. You cannot send your IT technologies. You cannot send your steel. You cannot do any marketing at all globally if you don't have plant breeders' right or mechanism to give rights to the plant breeders in their country. So it is that important. Okay, whether one is dealing with agriculture or not, having plant breeders' right on varieties is the key for being retained as WTO member. I'm using the word retained because most countries who did not have these enacted laws or systems of plant variety protection were given option to do it by some period, some years. So India had up to 2000. Many countries which are underdeveloped or very poor countries, they have still a time up to 2030. So therefore, not all countries are at the moment having the plant variety protection laws, but most have. More than 140 countries have. There are a few which have still not joined, but they're all given time up to 2030, most countries. And if they don't become by 2030, unless WTO's TRIPS Act is amended, they will not be continued as WTO members. There will be sanctions against them for trade. So that is that crucial. So there is absolutely no misunderstanding about the importance of plant variety rights, because the moment it happened, many countries had typical jumps of their production, their agriculture production, and their total growth in agriculture, and the countries, say GDP and those things, improved significantly. These are examples which are there in the case of wheat from France. The moment they started giving plant breeders right, new varieties started coming up, and there it is compulsory to have a new seed bought every time. Farmer is not able to save his seed and use his seed, so they have to buy new seed. So there was easy option for new breeders to come up and claim that their variety is better than the existing variety, and they could make money. And of course, along with the higher production, higher productivity, everything helped enhanced. Same thing in the case of US, where they started with corn hybrids, the first products. So that is how they started improving. And this is what it is. Then this is important for us to understand. This has been done in a journal called Canadian Journal of Agricultural Economics, where they did a very good study on how Canada has developed as a big country in canola trade. Most of you who cook are now aware of canola oil. Our co-chairman here is a counter canola man for Indian Brassica gentiana. Canola is Brassica napus. So he is the one who has produced a variety which is protected by us for canola gentiana. Okay, it's not canola. He has called it Indola. Okay, all right. This is a 6-year study between 1995 and 2005 data. They did it for 6 years based on the data of 1995 to 2005 by surveying over 10 million ton crushed oil area. Okay, and they find with clarity that adoption of variety protected under plant breeders' right was significantly higher and in priority demand by farmers, and that is the reason that Manitoba and Canada became successful in trading with canola, and now globally that is the biggest money spending point. Another thing that happened with this investment of plant breeders' right is investment in R&D in agriculture in those countries. That's what I showed you. Ask you to check up with all developed countries. When the plant breeders' right was given, the governments were induced to invest money in research, like ICRs, because they saw the country's progress was visible by returning that investment in agriculture. You might have seen, agricultural economists speaking, investment in agriculture is returned the fastest in terms of return made to the investment made, in terms of magnified ratio of investment versus product. That's what happens best in agriculture. That is what is the reason why after the PBRs were given, those countries started investing greatly in the investment on R&D. Not about variety of plants, but R&D in agriculture. That means other technologies, plant protection technologies, production technologies, many other processing technologies, investment happened. So there is an overall growth in agriculture happening because of plant breeders' right. This is UPOV's data, okay, not mine. And this is another case with Korea, where again, they've governmental investment multiplied by this after they joined UPOV and started giving plant breeders' right. Okay? That's what it is. So UPOV concludes that by diversifying, awarding plant breeders' right as a facilitation for competitive breeding point, it would increase the diversification of breeding activities by breeder, because they have to be one up on the other. They have to be better than existing variety. They have to have something added to the variety. So they have to do better than previous variety. So add a trait or add value to a trait, things of this kind, but being done genetically. So that is how the varieties got developed, and new varieties were getting developed. And PBR also introduced foreign varieties. Friends, I would like to tell you, among the varieties which we have registered here in our PPFRA, we have more than 50% of the companies who have registered with us are of foreign origin. So they know that in a country where there is a protection of plant breeders' right given, along with farmers' right as we have given in India, it is still possible for them to get the returns of their money, and they are investing most. Some companies have invested most in India compared to any other country they have existed, even in the country of origin, because there is no market as large as India in agriculture. No other seed market is there which is as big as India in any commodity which is in trade. So therefore they know the value of it, and that's what happens. That's what UPOV says. Exposure to foreign varieties will happen if you have rights in your country, because new foreign varieties will start coming in. You might have seen in the market when you're buying now apples or grapes, where are they coming from? They are coming from the materials which are come from abroad, and they have registered with us, and they are able to trade with them. And there are several cases which we have been hearing. And today morning, there was a court case in High Court where we had to contend with 5 very senior lawyers who take more than 10 lakh per hearing. 5 senior lawyers were there today against an order which I took out, and the High Court honored, in one hearing itself, they honored what we have said is correct, and the party was asked, the order was not dismissing the order which we gave. The order was retained and accepted. So that is the kind of priority today in agriculture investment. If a company, seed company, is investing on 5 senior advocates at almost 30 to 50 lakh rupees for one hearing, you can imagine the kind of money that's at stake, even in plant variety commerce. So this is what I told you internationally. Let's see what India has done. Did India join UPOV? Because I told you by 31st December 2000, India was to join UPOV. Did India join UPOV? And if not, why didn't we join UPOV? Because early in 2010, after our enactment in 2001, this is after 10 years, an Australian has written, Indian government decision to accede to UPOV. So this should make you ask a question. I'm not saying India has joined UPOV, but this person is saying India's decision to accede to UPOV severely compromises the farmers' rights and provisions of PPFR Act. I mean, contradicting the act is what she's saying. India at one point is having its own act, and then it is also acceding to UPOV. How is it going to happen? This is not correct, and this is defeating the purpose of Indian government, is what this review says. Why was PPFRA established under sui generis system? That's because sui generis system gives the optimal case, and this I noted from WIPO's 2015 report, which says they have accepted it, and this is what we have followed. India has followed this. This gives you an answer. What is the meaning of UPOV, the claim on UPOV, and then not being part of UPOV. An optimal sui generis PVP system would have to take into consideration the type of domestic seed industry, that is Indian seed industry for India, and use of farm saved seed, a very exclusive factor among all other countries. No other country, including Bangladesh, Sri Lanka, Thailand, Philippines, none has been given this option to give their farmers the ability to use farm-safe seed without giving anything in return to the variety breeder whose seed they are saving. Okay? First, they are not allowed to save, and if they are allowed to save, there is a cost to pay. The breeding strength, the national breeders' aims the next 5 to 10 years. These are the things which have to be seen before jumping onto the bandwagon of UPOV or having your own act. Atal Bihari Vajpayee government was the government when this happened, and they took courage. And I should say it's courage, because many other, even Indian Act, which was drafted, UPOV, the TRIP says UPOV has to be consulted and their opinion has to be taken before enacting. That is also a signed agreement by India when India was a signatory, one of the formative signatory, 1994. So it required that India sends its draft of its act to UPOV, and UPOV gives its opinion, and that has to be built into the act. And Indian, say, legislation took courage, which now happens to be unique courage. No other countries have shown it, that we will not accept the objection they had. And UPOV had objected to the same point: no saving seed and no sharing seed. In your act, both are there. You have to delete it. That is what they wrote to Indian government. But Parliament of India went ahead, because we are not doing anything against the TRIPS agreement. We are consulting, but we are not agreeing with you. We have consulted you, but we are not agreeing with you, and that the Parliament has approved. That is what it was. We had a wonderful chairman of the parliamentary committee, and he was very brave to go on with it and brought in all those checks and balances to see individual plant breeders' rights are not really hurt, which I will disclose after some time. And then how we can coexist with the farmers' right, defying the UPOV's diktat. See, India tried. You must also know, in a democratic country, it is not easy to get an act passed. 99 December 31st, that is 2000 January 1st was the last date before which we had to enact. 2000 December 31st was the last date before which we had to enact. And nothing was being placed, as you can see here. On February 16, Dolly Chakrabarti, who happens to be an alumnus of IARA, seed science. Raghu may know her. She was there when we joined PhD. She had joined MSc. Sorry, Rao is Raghu to me. Deputy Secretary Seeds, Ministry of Agriculture, visited UPOV to explain India's proposal for legislative bill, which was unlikely to complete the parliamentary procedure by 24 April 1999. Okay, by 24 April 1999, we will not be able to do it. That was the meeting. On 25th, there was a meeting in WTO to review, and we were not able to do it. This is what Dolly Chakrabarti went there to tell the UPOV. You know, on April 12, UPOV writes to the Additional Secretary, Minister of Agriculture, that non-compliance of one article of 1978 of Indian draft PPFR bill seeking deletion and exchanging selling rights to farmers protected variety. Okay, so they objected to the point of delete. They wanted us to delete the exchanging, that is sharing of seed, and saving of seed and selling of seed. They wanted us to delete this. Farmers are should not be allowed to sell the seed. They should not be allowed to exchange the seed. They should not be able to allow. In 1978, there is a fraction allowed to save on the own farm. Whereas in the 1991 UPOV, which is existing today, we have no option. That is what this says. On April 22, Vice Secretary General UPOV, who is the chairman of UPOV, just like Vice Chairman Planning Commission or Niti Aayog, is the CEO of that organization. Similarly, UPOV, because the chairman of UPOV is also chairman of WIPO, All Intellectual Property Acts International Organization chairman is also chairman of UPOV. So therefore, the Secretary General or Vice Chairman is the one who deals with it. So he met N.K. Singh in Geneva. Mr. N.K. Singh was Cabinet Secretary, went to Geneva on April 22. 25th April was the meeting. On 22nd April, he also goes there. to tell them and explain to them our situation by receiving the Secretary General UPOV to give us some leverage so that we either become member or enact in the parliament. Becoming a member was a problem because of this contention. So Indian government had this very slippery situation of accepting not to give the rights or give the rights of the farmers and don't accept UPOV. Both were there contradicting each other, and we had to do something before December 2000. On September 17, UPOV informed High Commission India that 1978 law will be finished if we don't accede to UPOV before 1999 December. That is what it is. On December 17, 1999, see how close the dates are. Just about 13-14 days, 2 weeks before the deadline, same Dolly Chakravarti gives a copy of the Protection of Plant Varieties Farmers' Rights Bill 1999 that it has been placed on the tables of Lok Sabha on December 14. This letter is sent to UPOV to say that we are on the way to enact, so therefore accept it. And the Joint Committee chaired by Sahib Singh Verma, the one who was very critical in reviewing, most of us gave inputs to him and he was very vigilant and he was so active in accepting the scientific laws and scientific meaning of the provisions of the Act and incorporate many. And that's how he stood by not accepting the 1978 related stipulation which UPOV said, delete so and so, so and so. We did not delete. That thanks to him. India was kept as an observer. Then UPOV agreed to keep India as an observer. So they have maintained that status of India even today. So therefore they believe we are going to accede to UPOV even today. Because on that date, Dolly Chakravarti wrote on 17th December 1999 that we have put our act on bill and therefore we are entitled to retain the UPOV possibility also, because most acts, UPOV says, when you pass the act and make the act in tune with the UPOV. So therefore you become member technically of UPOV. That's what it is. In 2018, from 2000 when we got the act through and 2018, 17 years later, UPOV is clearly informed by India that she will not accede to UPOV till UPOV amends its 1991 Act, admitting farmers' rights in simple compliance to IT-PGRFA. India, we have shown to UPOV that you have actually defied, non-complied with the IT-PGRFA yourself, being full. The entire EU is member of IT-PGRFA, who are members of UPOV, who started UPOV. And they have themselves not complied with IT-PGRFA, which is equally powerful act, powerful agreement, and they are wanting us to get into UPOV, defying the UPOV IT-PGRFA's clarity on farmers' rights. And we are the only country which has brought in. All the farmers' rights are IT-PGRFA rights. Nothing different. 100% IT-PGRFA rights these are. So that's what we have done. So we used that in 2000. I did that in UPOV. I went there and strongly said, we will not accede to UPOV 1991 unless you amend and you comply with IT-PGRFA or answer to United Nations why you have not complied with IT-PGRFA. This is what I did, and that is how we made it clear. And India consolidated its stand in say 2020, when our Honorable Prime Minister goes to Bangkok to sign a possible RCEP membership. RCEP is an advanced trade system among some countries, 17 countries. India would have been the 18th country, where there would be no custom or any other barrage of trade in terms of goods exchange, goods traded, no taxes, nothing among them, and they can unblock trade with any other country. And the members of these 17 are China, Japan, Australia, New Zealand, and all South Asian countries, Thailand, Malaysia, and many countries. India should have joined. Our Honorable Prime Minister went there. There were negotiations. For 4 days, there were negotiations. I was in touch with the Commerce Minister who was there representing India. And the condition, one of the conditions that put was India joins UPOV 1991. There were 4 conditions. One condition was India should join 1991 UPOV to be member of RCEP. Look at the global business as it happens. These are all, you may not understand the implications, but this is how trade happens, global agreements happen. They wanted to dangle that carrot of RCEP to us and make us join UPOV 1991. And if India joined UPOV 1991, that's what I wrote. If India joined UPOV 1991, there would be no farmers' rights. And India cannot afford to have no farmers' rights of saving seed and using seed ability for the farmer and selling to the neighbor farmer, because even today, with 100% capacity of production of seed by all seed companies and public systems, we are not able to cover more than 40% of area. ADG Seeds is sitting here. If every farmer replaced new seed by buying fresh seed, which would be a requirement if UPOV 1991 has to be followed, there would be no way more than 40% area can be planted. It will be worse than Sri Lanka situation. If 60% of land remains vacant, you can imagine who would give us the food required. No way. If UPOV 1991 is exceeded. So I wrote that. This is what it was. And then it was decided that our Honorable Prime Minister will come back without sign. And he did it. He went to Bangkok, but did not sign. Because if he signed, one of the 4 conditions would be joining UPOV 1991. There were other conditions related to Kyoto Protocol and those things. But this was one very, say, significant, critical change that would have to be brought into India after we became RCEP member, which the RCEP has been implemented from 1st January 2022. So India did not join. By 2022, there would be no seed available other than purchased seed. This would have been the condition. Okay. I think I'm going too slow. Let me go fast now and finish. The story is like this, that India happens to be the only country which has stood the full power of strength of its farmers by them to see that their conservation abilities, their ability to observe changes, record their materials, and India is the only country which has actually done justice to the farmers in every respect. Old varieties have been accepted. For even today, we register old varieties which are not older than 15 years, of course, but old varieties tradable. We register. But UPOV does not allow any variety less than 1 year old. DK, you can tell me if that is the condition imposed, where will be the new varieties? First of all, there would be no coverage because the total seed produced and traded will not cover more than 40% of total land cultivated. And if that has to be only varieties which are less than 12 months old in trade, where is the question of availability of seed in India? It would have been disastrous. So the government has fully supported us in taking this stand. I leave this because I have extensive point. Some things which are important, let me deal with it. The Euroseed is the very large organization of all European seed. Big companies, other than a few such as Monsanto, are based in Bayer, etc. They're all based in Europe. Okay, Syngentas, Bayer, they're all Europe. That circumstance. The plant breeders' rights, they say, 1991 is the basis for successful trade in agriculture, and that's what they say. And there is continuous flow, and therefore this is must. This is a must, they say, and they have to do it. And their opinion is PBR's benefit is both to the society, research community, and farmers. So therefore, don't hesitate in becoming the 1991 member. That's what is their recommendation. But I just want to tell you the preamble of ITPGRFA, that is International Treaty on Plant Genetic Resources for Agriculture, is that the farmer should be enabled to save, use, exchange, and sell farm saved seed and other propagating material. This is what is our fundamental right of farmer. We have given that in our act as such, just to tell you that. Okay. This is an important part. I hope you will be able to understand. What is the sui generis point which we introduced? The purpose is not to lose sight of centuries of farmers' traditional involvement in seed systems. So we wanted to use sui generis system by introducing this part. Therefore, land races are to be saved. Otherwise, land races would not be saved. You can imagine, just to tell you an example, if we did not have PPFR in India and did not save the land races, then any company can take any land race from India and we cannot object to it. Because that's public domain. Anything public domain is accessible to anybody. That would have been the. Then the option of enabling non-monopolistic. That's what we introduced by having multiple licensing system. UPOV does not have any access to licensing system. Ashok, there's another point which is important. UPOV 1991 does not have any option for licensing, which is only two parties decision. There is no legal provision for licensing. In our case, licensees are also registered with us, so that lay liability is also fixed. Whereas in UPOV, even that is not there. That means the breeder owns the entire right on the material and it is his discretion to deal with it as he wishes. No compulsory licensing which we have. If he decides to charge a variety by certain price, if you're willing to buy it, otherwise I don't care. That is not there in India. If the price is beyond certain buying ability of farmer, which one can assign, any farmer or anybody, any seed producer can come apply to the chairperson and say that this variety has been registered by you and the cost the company is charging is this. We cannot produce seed at this cost because we cannot sell it and farmers are not able to buy it. So therefore, kindly release the licensing compulsorily to me. If that is evaluated by us in perspective and found true, we have that power to do it. Compulsorily license it to any other agency. Even that is not there. Even a trade arrangement of this kind is not there. So that is totally in one direction. PBR of plant variety and plant breeder is the supreme most in UPOV, not the user of the variety. This is something important, and in a country like us, where the area is so big, it is not possible for individual breeder to say sell variety, produce variety and do it. It is not possible at all. So therefore, it is just not relevant to India. So that's why we use TRIPS, and we have. And if you look at the number of varieties being applied to us, applications being made to us, and the number of breeders who are given right to, it is comparable to any other country, any other developed country. The Netherlands Deputy Prime Minister, she came to discuss with me, convincing the Agriculture Minister, our Agriculture Minister, to somehow join UPOV by saying that, see, our varieties will not be made available to your farmers. We have such wonderful varieties in horticulture, such as grapes and all, roses, grapes and materials, you will not be able to produce in India because we will not come here. But she was wrong. We had several of more than 780 horticultural varieties are licensed with us. I showed her that data. You're wrong with your information. Somebody has not informed you correctly. Because of the volume of trade, which is bigger than the entire Europe put together, even in a small state like Karnataka and Maharashtra, they are there. They are producing, they are selling. So it is not correct that by not joining UPOV, we will not have access to the varieties. Our farmers are doing it, and our companies are also giving it to the farmers. So and companies licensed by you, and the variety is registered with us. So I showed that, and this is how it was. That was just before the Bangkok meeting. So that kind of negotiations they engage into, trying to do everything possible at international. That's how the international. You must have heard current foreign minister, who has been very straight and direct in his interactions. If people are interested in writing UPSC, you would have heard, you would have seen his statements, read his statements on how India is directing its control over Europe and other developed countries and able to stand up. So this is one of those areas in which we are able to stand up. Let me leave this. I don't have time, Ashok, no? Okay, all right. Okay, the important point which about farmers variety, which is important for many of the breeders here to know. It is not that any variety the farmer comes up and gives to us is given as farmer's variety. It has to be only traditional variety or selections in traditional varieties. They cannot take Dr. D.K. Yadav's variety or Ashok's basmati variety and do something in that and come back to us and claim it is a farmer's variety. While we have given the farmer identity of breeder, which no other country has given, we are given that. Farmer is also a breeder, but he will have to breed only in his field as he grows his crop, select in the traditional variety. And if he engages in any activity which is incorporating the breeding principles of crossing and selecting in the crosses, then it ceases to be farmer's variety. It is like any other plant breeder like D.K. Yadav or Prabhu or Ashok is. He'll be treated like that only, which means he has to compete with every other perspective like any other applicant competes. Otherwise, he has absolutely nothing to prevent him from applying as on today. If it is traditional variety, free of cost, no trial cost, nothing. And trial is done only to see if it is testified as uniform, because farmer's variety need not be uniform. That is what is tested in the case of farmer's variety. So not everything is farmer's variety. If he steals any material from the university or IRI and then brings it up as farmer's variety, it will not be. Many breeders did this also, sir, who after their retirement brought some varieties in the name of farmer's varieties, in the name of some farmer. So we have put an end to all those because obviously one can know whether it's traditional or non-traditional. It is very easy to know. With 1 year trial itself, one will know whether the variety is traditional or non-traditional. So this is how it is. So that is totally prevented now. We have legalized those. The guidelines have been issued and it's very clear now. So this just wanted to tell you, no apprehensions about such things. So these are the rights for the plant breeders, which are globally there. And these are the rights of the farmers to save, to use, to sow, resow, exchange, share, sell. The last four are the ones which are internationally contentious, and we are the only ones doing it. Of course, leave this. I'm leaving this. Many things I wanted to have a discussion on, but it's not happening now. For a short time. I will like to explain this part to the audience here. When the farmer also is likely to violate farmer's right. Any farmer who comes with you, comes up to you and says it is farmer's variety, it is not that he has full freedom to do anything. He has to be also within the law boundary. That is what it is. He has to sell it only unbranded form. If he sells it in branded form, he is violating. Branded means packing, labeling, naming. He adopts any practice that can be described as processing. If he adopts any cleaning process or roguing process while production of it, or isolation, things of this kind like any of the seed producer does, then that is also not to be categorized as farmer's variety. He is violating his right to sell. The product is beyond what is described as farm produce. So anything which is other than farm produce, like the normal rice, normal wheat he would grow, or normal vegetable he would grow, anything other than that he is doing, like nursery or extra nutrition provided, then planted at distance, space planting. If any of these techniques are adopted, somebody complains, that will be honored and that will be denied as right. Then if he claims illegitimately obtained seed from somebody and then grows. Any of you have heard of the Pepsi potato case, which we took out an order on? Okay. That was one case of this kind. Then he sells the seed with variety denomination using the farm produce if it is not true to type. The Basmati 1, for example, 1121, it is being sold as Basmati 1121, it is not so. Just because he says, I got it from so-and-so farmer, he is not entitled to say it that his is 1121. Even if he doesn't sell it, if he says it, claims it, even that is violation. So there are many violations which the farmers are also to be aware of. So that is something important for the plant breeders also to realize that giving farmers right is not an impediment, is not a problem for us. It is something that we have to live with. I'll just take two examples of other countries and then stop my lecture because really crossing time. Otherwise, I just wanted to say This is one example of how plant breeders' right with farmers' right can ensure mega returns to right holder plant breeder. This story of SD 2967 by Indian Agricultural Research Institute, we shared it with 16,000 farmers by giving them 2 kg each before we registered. Okay, and then subsequently in 2008 we shared. In 2010 we licensed to 39 companies who came forward based on the result of those 16,000 farmers. So that is how this variety started going up to more than 10 million hectares, more than 5 years. There is no other variety, friends, in any country which has covered this area put together, or multiple countries in a given year. Okay, like Canada, US put together or some variety, or entire European community put together some variety of any company, maize hybrid of Monsanto, whatever. None. There is no coverage. Only some IRI varieties of Dr. Kush had covered 4.7 million hectares. That is the maximum estimate. Nothing crossing 10 billion. And this variety did because the farmers' rights had it. And this is a world record. And the next variety was licensed to 220 companies. So this is something most important. And I want to read this. This is the most unsung success story from India. I want to make it myself tell this because I'm involved in these varieties, but it is important for us to realize this part. When IRA made 24,000 farmers use their farmers' rights on plant variety to become live ambassadors or super spread of wheat variety or to world record area of more than 5 years. Okay, there is no other comparison. The farmers who saved, shared, and sold their seed using their farmers' right of the 2 varieties were actually unpaid promoters who were just like the demo farmers of prospective selling of companies. So they did this job for us. This advance saving, sharing, and selling by the farmers did not dent the licensees. See, the licensees already knew the farmers had this material. Still, they came to us. So which means it is not an impediment. It is not a limit because these companies knew that they don't have to spend in promoting the variety. And look at the phenomenal 75 lakh quintals of SD 2967 being traded between 2014 and 2020. 75 lakh quintals of all categories, certified, registered, all category of seed, self-sown seed, transferred, everything put together, because that is the coverage of the variety. This multi-mega volume of seed trade of a single variety, 2967, is of course the most ignored fact in our own country. I'm unabashed in telling this. While this is a huge world record and that has gone as the largest ever officially unrecognized event in the history of seed trade in India. The estimated seed value per year is 2,500 crores per year. Seed value only. And the estimated grain trade value is 80,000 crores per year. DG ICR tells about 19,000 crores of Pusa 1121 or 30,000 crores of Basmati varieties of IRA. That is because export data is there. This data is not there. Neither 4.7 million hectares of World Food Prize winner Dr. Kush is there. There is no data. It is only estimate. Here also it is by the seed production and the seed licensing and the quantity of seed sold and market arrival. This is the estimate, 80,000 crores. To this, you have to add anywhere between 7,000 to 8,000 crores more for the forage or the straw sale of the variety. So single variety is giving more than 100,000 crores money traded in a year. There is no other commodity, no other trade, whether it is Maruti cars or Mercedes-Benz cars or Rolls-Royce in India, which has this equivalence of trading by one commodity. Okay, and this has happened after registration of plant breeders' right, licensing and trading. Many of the licenses are traders with us, and this is also an impact in India. I showed you Kenyan, Korean impact. Here is an impact of this. In 2014, SD 2967 had less than 5% area in UP. In 2019, data from the state governments and other availabilities, more than around 60% of the area of wheat is with this variety. In Bihar, it was nil in 2014, it is 50% now. And SD3 second variety is nil in the 2014 area and around 30%. And because of which UP's production has crossed over the previous one by 50% and that of Bihar by 41%. Productivity, not production, productivity increase. Average production per hectare is increased by this. So this is the gain by holding on to right and making licensed seed producers to distribute seed. The point to note here is licensing and enabling licensed producers to distribute and sell, which would not have been possible by IRA selling it or ICR institution selling it or any other national seed agency or anybody in National Seed Corporation or anybody selling it. It was only because of licensing and having large number of licenses. This is what it is. I think I will stop here. Otherwise, it will go on forever. Okay. So finally, I would like to state this: creation of new plant variety and enhancing its value through plant variety production for improved livelihood of farmer, life standards of consumers, and profitable investment to breeders is a true tribute to Gregor Johann Mendel. He became Gregor Johann Mendel after he joined the monastery. Otherwise, his name was Johann Mendel only. So when he was born in 1822, he was Johann Mendel. In 1843, he became Gregor Johann Mendel. So this is for his capacity and his genuineness, ingenuity in enabling science to create new plant variety. Thank you very much. I had very interesting points to tell you, but the time is not ripe. Time is very ripe, but time is not available for me to discuss that. But you would have known many points. How UPOV intervened in Malaysian regulation, Philippines regulation, Bangladesh regulation, by all these small things. I'll just tell one small thing, Ashok, if you permit, so that the point is well realized. Sir, this is important from UPOV versus non-UPOV. If you are a farm holder, you have 5 people in your family. If you became 1991 UPOV member, then you can produce the, suppose it is rice. You can cook the rice and eat it among 5 of you, while the farm is in the name of X. There are 5 other members in the family, so all 5 can eat without any problem. But if you want to share 1 kg rice cooked with your neighbor, then you would have to take permission from the breeder for the harvested product, processed product. Is it possible in India? But this is the reality. This is the truth. Thank you. Yeah, please ask questions, silly or otherwise. I want at least plant-breeding students to ask, please. Yeah. Please go ahead.
Sir, I want to know about trait license in India, whether the trait like herbicide tolerance, disease tolerance can be licensed. If yes, what is the procedure?
See, as far the licensing part for ownership of the variety is concerned, it is not trait based for the licensing part. But if the variety is having a particular trait unique to it, the breeder can claim that this trait is unique in this variety, and this is the mechanism or this is the methodology to record it. Please use it for my distinctness of this material and give me license. So application has to be made that way, that it has herbicide tolerance, and this is measured by this methodology. Even if it's similar to other varieties, my variety is different for this trait. So therefore, register this and protect this. Then we protect the variety, not the trait, but because of the trait. We mentioned the certificate, it is distinct variety for this trait. What is protected is the variety, but for the trait, not the trait. The trait is not protected. The difference between the two is, if anybody else uses the trait, then the right holder can ask if the trait was to be protected. Here, if somebody has to use the trait, he has to use the variety. And for that purpose, if it is only single use, there is a researcher's right, he can use. If it is something else, like say hybrid, where this becomes a parent, then we will award benefit share to the holder of the variety for that, although for the trait, but not based on the trait alone. I hope I'm able to clarify. Okay.
In that case, so if a farmer cooperative is utilizing the particular protected variety for their breeding program, so whether they have to take a license from the breeder or from the PFR?
See, if whether the farmers cooperative or anybody, any company, if they have to use a protected variety in their breeding program, there is absolutely nothing required to be taken as a permission, whether it's UPOV or non-UPOV. It's the same. In India also, there is absolutely no requirement to take any permission. But if that is a parent of a hybrid, if that's going to be used as a one parent of the hybrid, then you need to take permission from the breeder, which means the breeder will negotiate. Okay, you produce, but give me this much after you sell the hybrid. So,
okay, sir, I have 2 more questions. May I?
Please.
Sir, I have heard you before some few months before. So you had said that the time limit for registering the extant variety is going to be completed.
Yeah.
So if someone comes up with registering an extant variety, which is a variety in common knowledge, so I have also read a statement that it will be case by case basis. If it is necessary, then the PPFR will admit it.
Yeah. No, it is not. Okay, no, it's fine. It is not now or then. It is what it is. If it is a farmer's variety, then it is amenable for protection of 10 years after it is notified. The extant variety statement is for a period after a species is notified in the Gazette of India that will start protecting varieties or registering varieties in this crop species. That happens, say, in 2022 of a particular species. So the extant varieties are admitted for 6 more years till 2026, even if they existed long time before 2022. Okay, that is 15 years before 2022, because 15 years is the protection period. So if the varieties existed till 2007, okay, in market, available information that the variety existed after 2007, they will be protected for 6 more years up to 2028. Varieties of 2007 can come. Okay. In the case of farmers, if it is farmers variety, then that is 10 years period. That is 2032. Farmers are small farmers in small area. So many varieties, but in small area. So therefore, we give more time for them to come up. 10 more years. If it is a community variety, such as landrace, or those which are there for decades, no time limit. They could be 20 years old, 30 years old, 40 years old, anytime. They will be registered. That's what is the case by case meaning. Okay. Landrace, the community ownership materials, which are there for many decades, they can be registered with us anytime. Okay, during the 15 years of its sale. That's.
Sir, my last question is whether PPFR has any protocol to collect the details of how a variety is developed. What is the cost?
Yeah.
What is the cost? How much is spent for developing a variety?
How much is not our business. But what is the methodology used? What is the process used for breeding? And that is then, in fact, in one of the judgments I have made, I've done it on the basis of a claim which could not be verified by the breeder against whom the claim was about the process he claimed in the application, but the data and his breeding record could not prove that this could have been. I mean, the methodology he proved meant it would be a segregating progeny. Okay, the methodology which he gave.
There is no protocol for finding the cost for developing a variety.
No, cost has nothing to do with this.
Thank you, sir.
Yeah. You want me to stop, Ashok? Yeah.
Sir, good afternoon.
Yeah.
Sir, I have one basic question. Suppose one hybrid is there in the public domain, and a farmer is continues to cultivate that hybrid, and now that hybrid was bred for a particular oligogenic trait, and for which both the inbreds are improved. Now, suppose the farmers continues selfing and derived his own inbreed line for the blessing of recombinations and segregations, and derived his own hybrid. So this kind of hybrids whether violate the plant breeders' right or not?
Definitely not. Most plant breeders do this only. Most large companies, big companies do this reverse breeding by looking at the hybrid, then selfing them and creating their own inbreds, and that's what they do. So there is that is fully compliant, because you are not copying the parent of a hybrid, you are creating your own inbred through normal segregation process, which is not 1 year process as such, and you don't repeatedly use it. You're only selfing it, and the progeny is different from the parent material which you sell. So therefore, there's absolutely no problem whether you're a farmer or a breeder. Absolutely no problem.
So farmer can claim this.
Yes,
kind of
yes.
varieties?
Anybody. Why farmer? Anybody can claim. Yeah. Yeah. You have a question, Ashok, please. Yeah, please. Yeah. I did not finish the full presentation. There would have been then good number of questions. Yeah, please. Please.
Good evening, sir.
Yeah. Good evening.
Sir, actually, in 2018, I bought one hybrid seed for 40,000 rupees per kg. Chile hybrid 5531. And the next year, another Chile hybrids, two hybrids, 5531 and HPH 1052. I forgot the exact number. Chile, Badigi Chile, I bought it for 40,000 in 2018. In 2019, I myself paid 1,10,000 rupees per 1 kg. And 2020, now I was in Delhi, I couldn't able to place order before 6 months, even though I will able to pay 1,50,000 per kg of the seeds. The price increase you can see from 40,000 direct 1,10,000. Now, even though I can able to, I will ask, even I will pay 1,50,000, but they are telling that the seeds are already sold before 6 months of sowing. You have power to give either the power to register a variety, give power to sell or not sell. You don't have a control in which price they will sell. You don't have any control about the...
No, we have control. It is not that we don't have. India only has the control on the price. No other country, UPOP does not have control on the price. See, apart from India only has power, one must understand in no other IPR, the price ceiling is controlled by the regulator because there is competition. If there is no competition, this is what happens with the cars you're buying today. You have cars which are 50 crores, you have cars which are 10 crores. Why? Because they are being sold. They also go on the 4 wheels only. So, okay, same way in the varieties also, because this variety has some advantage, the company is able to sell it at 2 lakhs, it is able to do it. But now, if the farmers who are using that seed at 2 lakh rupees, if they are making 10 lakh rupees, which they were not able to make in a variety which they bought at 40,000 rupees, there is no harm in investing for the seed at 2 lakh rupees to make 8 lakh rupee profit, which was not possible 40,000 rupees investment. If those comparisons are made available, then we will not interfere. If the comparison says even with 40,000 farmer can make 8 crores, 8 lakhs, and even with this he is not able to make more than 8 lakhs, then it is a cost which is prohibitive cost, which means people are not able to buy it as they desire because of this, while they are not able to make big profit. So in that case, we will use the power of compulsory licensing to give it to other people and restrict the price for no more than this and cancel the right of the breeder. But for this, you have to give lots of evidences, and we have to hear it like a court. You have to give claims, giving proof, then they have to come with answer why they have to charge this much, and then only a decision will be taken.
And sir, another question, like for annual crops, the production period was 15 years. For perennial crops, production period is 18 years. What that small number of year difference, 3 years, make a big difference. When even, when to remember.
See, I'm not the author of that story. The parliamentarians did it. I'm only keeping it.
Whether you have to keep same or I have to
No.
keep it.
No, I in my very first month after joining, I put up a proposal to the government to make 25 and 30. This 15 to 25 and 18 to 30. The government did not agree. It is for them. It's for the government to go to the parliament and the parliamentarians to decide. It's an amendment. So that is not in our control.
Sir, another question.
Good, my purpose is served. Yeah, it's okay. That's what I wanted actually.
In our last year, our senior question paper, one question came. Like, if breeder has made any mistake while filling the performer or application, if made any mistakes, the Registrar General only has a power to make changes in that performer. If chairman don't have the power to make any changes, because the options are given chairman, Registrar General, and I'm confused.
No, there is nothing like this. It is nothing of this kind. The registrar has on his own can make changes in the application. This is before registration, okay? Before registration, it is only between registrar and the applicant. Registrar General, both are same as far as the work is concerned, okay? Before registration, any change can be made between the applicant and the registrar, between them. Of course, it is through basic official formatting is there, and that is on record, and that is done. Everything is judicial paper.
But chairman don't have power.
The chairman is not there on the scene when the application is made for registration. Post registration, of course, the chairman gives the direction to registrar. On that basis, the registrar can be asked to give it to the chairperson. So that's a different thing. But this is not a question you have to bother about in terms of understanding the law. Okay, don't worry about it. Yeah.
Good evening, sir.
Good evening.
Sir, suppose
your director also wants a question. He has raised his hand some 10 times. Can we give him option? Then you can ask. Yeah, please. Otherwise, you may forget. Yeah. Okay.
So suppose there is an extant variety which is notified under Seeds Act in 2010, but the species is notified under PPVFR in 2015 for protection. So that variety is not yet protected now. So can it be protected? If not, it will be in public domain. Or if it is protected, for how much period it will be protected?
Very, very good question. Since it is 2010, it can be protected immediately, but from 2010 only. Okay, 2015 has no relevance. But 2015 relevance is only for the 6 years barrage we are given. If it has crossed the 6 years we are given, in some crops we are given more. If in that crop we are given only 6 years as the say catch-up period for extant variety, that will be 2021. So if before 2021, and the protection will be only from 2010, 15 years more. That's all.
So it will be not protected.
No, it is not protectable. Yeah.
2025 it can be protected.
Provided it is within that say window for accepting extant variety.
But it was in PFR Act, it was given 3 years after the notification of a species.
No, in the Act we are not given 3 years. It is done variety by variety by us when we notify. At that time we give. It is not in the Act.
If it is a farmer's variety, as you said, it will be 10 more.
10 more. Yes.
What about the case of GM crop protections?
We have no distinction for the GM or non-GM.
The GM crops will be protected.
Will be protected if the process for development of GM crop is available in the country for that particular crop, for the GM trait. We have absolutely nothing to come in between.
So BT cotton is protected in India.
When almost 98% of the varieties in sale are protected with us.
Thank you.
Except few which are not, because they are not in market, so they are not protected. Otherwise, all are protected.
Thank you, Chairman sir, for very enlightening presentation. And my question to you is in 3 different roles: as a chairman, as a speaker, and also a chairperson of the party. It also involves the co-chair, Dr. Khadar,
Or
in his capacity as co-chair as well as ADG seats from ICR. The first thing is that, we are, and don't take me wrong on that when I'm asking this question, that I'm not for farmers' rights. But because of farmers' rights being in place, the investment by the private sector, well-meaning companies in R&D for OP varieties. And I must say that rice and wheat together, if we put 121 million ton rice and 110 million tons of wheat, of total 309 million tons production, food grain production, roughly, almost 250 million tons is coming from rice and wheat. The investment in these two crops is almost zero from private sector, except in rice we have some hybrid area, which is hardly 6% of the total 44 million hectares. Private sector, well-meaning companies, they are not going to invest in research and development in OP varieties because of farmer rights. The government funding for research support is declining day by day. Sometimes there is 20% cut, 30% cut, and the institutions are struggling for supporting their infrastructure, salary, pension. Now, under this situation, I would like to see, I would like to have your response as to how do we move in order to attract investment for OP varieties from private sector, if farmers' rights is there in the text. That's number one. Number two question is that involves both of you. If you have a protected variety with PPVFRA, which is not released and notified, whether we can go for commercialization. This involves me as a breeder, also as a director of the institute. And it doesn't invite any problem from the three holders.
Yeah, I had a very good slide on this, Ashok, about the second question. In fact, I took the example in that of Vishwas genome edited variety. I took that example here because of lack of time. I could not say. We should answer your typical question because it is not notified. It is not even tested in All India coordinated trial. But what I'm saying is you can come up to us and get it protected. As of today, because it does not carry any foreign gene, foreign genetic material, which will give me documentary evidence of it within the guideline, which says IBSC has to do it. You give me that, I will get it protected. And you don't have any other restriction in doing any kind of sale from that material anywhere, including exporting it without any, say, approval of NBA or any other agency. No exim, nothing. Okay, no problem. Then the second part, you're partly correct in saying there is zero R&D other than rice, which is wrong. Other than these major crops, where OPs are also there with hybrids, you have this query. In other crops, because of lack of breeding materials in, say, wheat, to compete with public sector, they have to have extraordinarily good material in wheat to compete with it. That is the limiting factor, not seed production, because licensee seed producers, despite farmers' rights, are doing it. That's what the companies will do. There is no difference between their selling seed as licensee or as original producer when it comes to marketing seed. It is the same. The variety has to be in demand, and since they will be the only owners of the variety, if the variety is good, they will sell. Licensees competing among themselves are selling single variety even today with huge profits is an eye-opener for them to have real good varieties. The problem is not having real good varieties in wheat, other competing material. In all other cases, whether it is mustard, whether it is ragi, whether it is pearl millet, even OPs are being invested by companies and being sold. Even in wheat, for example, Syngenta has invested hugely in R&D in India today. And they are doing it, and it is not limiting factor. And of course, it is in the act. You have no option. And we try to educate. I spent a lot of time with the companies to tell them it is your myth not to invest. It is your practical decision which you think is correct decision. But if you actually experiment with it by having OP material, licensing it, no farmer will go to use his own seed if you provide him the seed in time at a place where he can go and buy when he wants to sow. He will first buy the new seed. That's a fact. You do it, try it. Many companies have done it now, and they are licensing. Mayuko has also come forward with some investment in wheat now. And in other crops, in all vegetables, for example, all the registered varieties are, most of them are non-hybrids, which are registered with us. So it is happening. Volume.
Question is, there are some investors in pockets of excellence, but we need many investors. The volume of investment in R&D is completely dwindling. That is the worry. Not that there are not investors. So that volume is affecting general cycle.
No, this is nothing to do with plant variety protection. Of the 400 companies, no, wait. Of the 400 companies which are in trade today in India, only 49 or 47 have real R&D for new variety development. Okay, no, that's it. There's nothing to do with PVP. That is their basic organization. They don't have plant breeders to do the job, first of all, and basic genetic material to start breeding programs. They don't have. So that is a limiting factor, not PVP or no PVP or OP or hybrid. That is not the limiting factor at all. Where they know they can spend money, they can produce seed and provide it to the farmer, they will invest, and they are investing in vegetables. There are many who have occupied Uttarakhand villages with OP varieties protected with us, and they are making 100% trade in such as carrots or many are there which are OPs only. So they are doing it. ऐसा नहीं है कि नहीं, ये गलत information है जो rampant है because of people who give that information are not R&D experts. The companies who give that are not R&D companies. R&D companies are there who are not revealing the details of their R&D most of the time. And no, he might smile, that is true, because he is not able to get licensee for his rice. Yeah. But it is in the act, and unless amended, we cannot do anything about it. And that is now a unique selling point for India. So I don't think there's any dilution possible. But looking at the market size,
depending on these two crops, you would also agree that in wheat it is 95%, not even 99% is public sector bred varieties, nothing from private.
Yeah, 100%. Yeah,
100% only. So if this remains the situation and the funding dries up in future, there would be kind of problem with, because companies are so interested if they have farmers' rights not in place. Otherwise, the seed sale, recurring sale in hybrid, why?
No. It is not right, Ashok.
Well, you see, there are two contradictions that you are saying. One side you are saying 60% seed is being saved by the farmers and used for planting next crop. That means those 60% are not coming for purchase, right? So if we have a farmer's right in place, and then a farmer is forced to come back, he will go and buy. That will 60% will become 100%. In hybrids, we have 100% replacement. There it is not happening. Why it is not growing? In spite of all efforts.
Wait. See, the 60% is there because they don't get the seed. The 60% are 90% of the 60% self-seed farmers are
25% carryover in seed in each crop, which is not sold and it comes back to
that is because of wrong timing. Yeah, there's a basis. You have to analyze it properly. If it is not available when it rains, no, you cannot eliminate these points. You have to actually dissect the variety, each variety by variety, situation by situation. There is no generic answer here. And whichever in the vegetables example, what I've given you, they are mostly OP materials and they are all protected and they are marketing it. There is absolutely no say crisis about that. Then the same logic won't hold good. So it is, there's no, and let the production come down, Ashok. Investment will be there from the government. Yeah, we should not get to Sri Lanka. Yeah. Yeah.
Sir, I have a question regarding the breeding point of breeders point. Suppose some company comes to PPVFRA for filing an application and say that they have developed a new variety and submit the application. Then after going through the applications, you carry out the best test, invite if any person from the public have any objection regarding his filing the application. I don't know about the procedure, what procedure you will. You find that there is no conflict of interest has been shown by any of the person in the public. You go ahead, you award the right to the breeder, and when it is known by the farmers, I as a breeder comes to know that okay, this seems to be just like my material. I carry out, so now the DNA fingerprinting, it was very difficult earlier from the segregating materials, or say early fixed material, or say F6 or F7 fixed material, if somebody would have stolen. But now the DNA fingerprinting probably can come to rescue. I come to know that it's really my material. I approach to you and say that it's my material. You say that when we invited the objection, you did not raise the objection because I was not knowing about the character of that variety. So in that case, what will be the way to resolve the problem?
Very good. This is a very good question, but not unsolvable. Neither it is something which is difficult for anybody. As on today, if you are a breeder wanting to save your right, have your right on it, you must have full information about the characters which are required for dust characterization about the variety. You have to have it. You cannot say I didn't do it, so therefore I did not know. You have to have it. And when we publish for the variety, before we register, we give full access to the entire data on both the locations to anybody who wants to look at it. It's online available. So you have to have somebody in the IP cell.
Sir, I may be wrong, and I don't want to offend, but I have published a dust bulletin.
You have?
I have published a dust bulletin on barley. I know that what kind of information it needs to have. I must have an SSO to all the release variety, that the release variety are carrying which traits, and my variety or every stroke in my kitty is actually.
No. You're confusing the issue, Rajbir. You're talking about a variety which we have now found is being registered by somebody else, which is your variety according to you. If it is your variety, you will not be having, if you're an active breeder, you will not be having 100 varieties. You will have few which are tradable varieties, which you think are going to do well. It will be 1, 2, 3, 4, no more than 4 in a given season, given year, given crossing program. Not more than that. You should know about those 4 varieties, full information. And you should be vigilant to every month, first of every month we publish. Please hold on. Absorb what I'm saying, then react. Then please access the information available on the variety being claimed for registration. Look at the entire data, compare with your data. Even if you find 10, 15 characters varying, I'm giving very large number from your variety, still you suspect for some crucial characters it is something similar to you. Please send an application. You only have to spend some, I think 2,000 rupees or 4,000 rupees. Apply for it. This is a format. Then rest of the thing you leave it to us.
One lesson I have learned now, that continuously we have to search
Yes.
for your website.
Yes.
If anybody has filed the
Yeah, of course.
Second question, I just want to,
That every producer does it.
When you were showing the slide, it just came to mind that you showed that there was a large number of applications invited. In one of the slides, you showed the number of applications received by PPVFRA for registration. there was a period, if I have read the slide correct, from 2012 to 2016. The number of applicants were going into the thousands, and so was the number of genotypes and varieties. But immediately after that, it has drastically gone down. Whether there is a loss in interest of the breeders, or new materials are coming it's becoming very difficult to develop the new material, or there are some hindrance on the basis of some procedural difficulties.
नहीं बजट वजट कुछ भी प्रॉब्लम नहीं है. It is let me answer. There's no problem. That 2012 to 2018 period which you are mentioning, where you see a say sigmoidal log phase, it was not done as per the requirement of the Act. It was applications made for admission to schools of unborn children or without children. I think that should suffice. And I insisted you come with the child, then we'll register. That's the difference. Yeah. That's it? Good.
Sir, okay. Sir, can I? Sir, good evening. Yes, sir.
Where? Yeah.
Sir, is ex post facto resolution of disputes possible? If someone has not raised an objection, and it is published?
No. Benefit claim is possible.
So
after 6 months after registration.
Okay.
But if there is a claim which is after registration on ownership of the material, it is not there. It has already happened in a very famous case of Ravi maize. It has already happened where the material of one company did not come for application earlier, and they had to face it. Because there is no evidence available. Rajbir, in all these cases, you said simply that it is my variety. But there will be no evidence possible that you can give to say that this was in your possession before that other material which came for application was in the possession of that company. There is no evidence which can be certified evidence to say you actually had it. Yeah. Good. Okay. Chikappa, yeah.
The ex post facto resolution is not possible, as you already answered, sir. Suppose being a breeder, if I have all the records of my breeding book and also all of the traits, which is back date from the date the journal is published. In that case, is it possible in court, sir?
If I am Director General of your council, I will dismiss you for not doing justice to the investment made in developing a variety by taking it to the logical conclusion. You were lethargic because there is a system available and you did not use the system, and now claiming that your system should be accessible to you. So you have defaulted. So I will challenge you for not serving the country as required. No, that is in light vein, but then the fact is that you have to be vigilant. This is claiming I have done this. So you have to do everything to say, everything possible to say, nobody else says it. So that's your responsibility. हाँ बिल्कुल बिल्कुल ओनर को प्रूव करना है कि ये मेरा yes yes no you are right बिल्कुल ओनर को प्रूव करना है तभी तो राइट मिलेगा ना भाई.
Sir, thank you very much. And I think very
vibrant, brilliant, and live lecture you have as usual. So thank you very much. And Dr. Prabhu, actually a very typical geneticist, plant breeder, and then biotechnologist, and now dealing with the IPR issues. And as per his habit, up to until he doesn't go to the depth of the thing, so he will never sit, and he will go on discussing. And generally, Director Saab and Dr. Prabhu, myself, we have been in many platforms or in many issues. We are working on one or the other items. So most of the times they will be arguing. And as I was telling, Director is smiling. So when Dr. A.K. Singh is not convinced, so he will be smiling. But Dr. Prabhu will not agree that what he will try to say, he will not agree. But when he is not agreeing, Dr. A.K. Singh is not convinced, so he will be going on smiling. So that's why I was referring that. So, sir, regarding the lecture, I think you started from the Mendel's genetics, and then how the varieties, defining varieties, how the IP system came, and different type of IPs you explained very nicely, and the various protection organizations which are globally happening and how they came into existence, and what are the advantages, what are the disadvantages.
The important point is variety protection is the license for WTO membership.
Yes, sir. Yeah. So he has very, I think, elaborately explained about the history, how about the TRIPS, WTO, UPO, and the ITPGFRA, and what are the various advantages and disadvantages along with them, and the how PPFRA, the act came into existence, all the background which he has told. So that has been a very, I think, typical action from the government of India. And now after that also he has been discussing that in between also there were some pressures and everything. So he could manage that very nicely. And our farmers' rights as well as the plant operator rights, they have been, I think, saved, and the country is in very safe hands in terms of the protection of the various plant varieties. Regarding Dr. Prabhu's involvement in the various activities, since 2003, I have been with him very closely. I have been associated with him. So starting from the before that, what we generally think that this PPFRA Act, when it was being constituted, likewise the Biodiversity Act, and then later on the new seed bill, which we are dragging since 2004, and now it is again with the parliamentary committee. And the GI issue, which we have mentioned only one GI for basmati rice. So, and transgenics, whether the moratorium is there, and the case which we are fighting in the court, ICR as well as GAC and DBT, and the genome editing guidelines, and the getting exemption of SDN1, SDN2. So these are some of the very, I think, significant and landmark contributions from Dr. Prabhu, other than the varieties which he has just mentioned about 2967, 3086. So really, sir, your involvement every time, and I specifically, generally, I learn so many things from both of you, yourself as well as Dr. A.K. Singh, because almost every week at a very longer time on very typical issues we have been discussing, and both of you are the guiding force for me, and always helping. Regarding, sir, registration, Dr. Rajveer has asked one question. So I think the decline in the number, because earlier one point Dr. Prabhu told about without the child we are going to have the admission. The other issue was because this was new, and all the extant varieties which were there, that was very easy to register them. So those, because of that, the number was high, and everybody was, the institutions as well as the PPFRA were encouraging that more and more number of varieties should come. That is true, sir. So that may be one number. And regarding the commercialization and the registration of varieties, I think all of us, we should come forward. The advantages which we are seeing when there are conflicts and pilferage of material. So definitely that is required. And Dr. A.K. Singh's questions regarding the unreleased material which has been registered, I think during the new seed bill formulation, Dr. Prabhu was very firm belief that the registered varieties will not be coming to the seed act, and definitely that has been put in the act also. So definitely they will be registered varieties with PPFRA. They will be automatically registered in the national register once the new seed act come. But now if we want to do, many companies, all the companies are doing, and the commercial guidelines of commercialization which we are developing, we can put some component there, as well as our institutes are now, I think they will have to go for the licensing as per the seed control order, because many issues are coming. Earlier, Dr. G.P. Singh last year faced difficulty. This time our regional station Karnal is facing difficulty. People are putting RTI. So if we want to go the registered varieties and notified varieties, so definitely we'll have to go for the licensing for the seed from the state where we are going to trade that notified as per the seed bill. So that will have to take care. And regarding, sir, I have been one of the member of the extent variety in that release committee with Dr. H.S. Gupta sir. So many times we are interacting with the institutions, universities. So generally the maintenance part after registration, that is a big issue for not people coming forward. And in many cases, the institutions or the AACRP, they are paying the fees, but the universities, they are generally not encouraging their scientists for registering the varieties. And moreover, when the commercial value is not there and the variety is registered, as we are seeing in case of mustard also, maintaining 17 varieties, that is actually 4 or 5 are in the commercialization, that is also a big issue. So for that also, I think during your regime, some that solution should be there so that more and more material can come to you. So with these words, sir, I once again thank you very much for a very nice lecture, and thanks to the organizers, and thanks to all our seniors and our colleagues for being in this lecture. Thank you very much, sir.
Thank you, sir. May I now request our director, Dr. A.K. Singh, to felicitate chairman and co-chairman with token of love. Thank you, sir. May I now request Dr. Shailesh Tripathi for oath of thanks.
I sincerely thank Dr. K.V. Prabhu sir for chairing this session and delivering the informative lecture. I also extend my thanks to Dr. D.K. Yadav sir. I thank all the dignitaries present of the dais, and thanks to one and all for present in this auditorium. Thank you.
Dr. You also join us.
Is there? No problem. Pick up it now. No problem. No problem. Just keep it. Wherever it's not there,
Distinguished chief guests, invited guests, galaxy of intellectuals, delegates online and offline, faculty, students, ladies and gentlemen, a very warm good evening to one and all. On behalf of organizers and on my own behalf, I am delighted to offer a gracious welcome to you all to the concluding session of the successful 3-day symposium. After enriching ourselves with illuminating and thought-provoking lectures during the scientific sessions, some refreshing our previous knowledge of Mendelian concepts, some propelling us to think and rethink, and all motivating us and inspiring us and rebooting us by showing a way forward. Finally, we are at the valedictory session. So it gives me immense pleasure to begin the session by inviting the distinguished dignitaries. Chief guest of the session, Professor R.B. Singh. Invited guests, Dr. K.V. Prabhu, Dr. S.R. Rao, Dr. D.K. Yadav, Dr. Rasbir Yadav, Dr. A.K. Singh, and Dr. Vinod. I would now request Dr. Gopalakrishnan to present the recommendations of the symposium.
Thank you, Harita. Respected Honorable Chairman and dignitaries on the dais and off the dais. First of all, I would like to make a disclaimer that it is very difficult to summarize a 3-day proceedings on with 5 minutes or 10 minutes. More so, it is very difficult when the speakers or leaders or stalwarts whom you epitomize as role models. And I will be very brief. We will have the detailed proceeding posted in the website. The 3-day symposium, Tending Mendel's Garden for a Perpetual and Bountiful Harvest, organized jointly by Genetics Club, the 15th Genetics Congress Trust, and ISGPB, was inaugurated under the leadership, under the chief guest, under the chairmanship of your kind self, Dr. R.B. Singh, and the inaugural address was delivered by Dr. Trilochan Mahapatra, Secretary DARE and DG ICAR, who appreciated the commendable efforts made by the agricultural researchers in enhancing productivity and addressing hunger and poverty in India and elsewhere. He emphasized on the need for building partnership and collaborative approach, improvement in quality teaching, and the inheritance of disruptive thinking and innovations, and the ways to create and discover novel alleles for prosperity, and also leadership in competitive research in emerging areas of research, including epigenetics, and also taking care of the nature by nurturing it. We had 4 sessions with which were followed by 2 special sessions, one on genetics in education for school children, organized by the 15th Genetics Congress Trust, another on plant breeders' right for postgraduate students of genetics and plant breeding by PPVFRA and PPFRA. The first session, we had 4 speakers, Dr. B.D. Singh, Dr. V.P. Sahi, and Dr. Ebenezer Hyde. In short, the recommendations emerging out of this session is encouraging more languages in the medium of communication of genetics through local languages. Creating more tools to make science more practicable by the students, and also interacting with students, explaining the career options in biology, thereby promoting science among the students. the special session 2 on plant breeders' right, chaired by Dr. K.V. Prabhu and co-chaired by Dr. D.K. Yadava. Dr. K.V. Prabhu made an insightful presentation on IP in crop improvement. This is not taught in any of the courses in anywhere across the country or anywhere, and possibly with there may be some directions to include in the syllabus so that the upcoming breeders are able to realize the potential of IP in crop improvement. At last, I'll like to thank all the conveners, co-conveners, rapporteurs of all the sessions. I will not take any names for the want of time. All the sessions began with welcome by conveners and felicitation of speakers, chairman and co-chairman, and vote of thanks by the co-conveners. Thank you, the rapporteurs, for helping me to summarize the proceedings. The detailed proceedings will be available on the website. Thank you very much to one another. Thank you.
Thank you, sir, for briefing the proceedings. Moving on to the most important event of the valedictory session, that is award presentation. May I now request Dr. Shailesh Tripathi, sir, to kindly come on to the dais and announce the awardees.
So there were 4 sessions for the poster display, and in session 1 we have Manisha Saini. Manisha Saini, is she present here?
Yes.
नहीं ठीक है.
Next is Ashwin Kumar Katral. Next is Harshita BS. Harshita BS. In the session 2, we have Ajay Kumar. Ajay Kumar. Abhijit. Abhijit is there. Abhijit is not there, I think. Next we have Preeti Sagar Negi. Preeti Sagar Negi. So Abhijit has come. Just has come. Abhijit. After Abhijit, we have Preeti Sagar Negi. Preeti Sagar is not there, I think. We have Pritesh Kumari. Pritesh Kumari. Next, we have Vikas VK. Pritesh Kumari. Vikas VK. After Vikas, we have Akash A. After Akash, you are Akash. Rintu Jha next, you go. Rintu Jha. Now in the session 4, we have Shivani Nagar. Shivani Nagar. So after Shivani, we have Gopinath. Gopinath. Gopinath hai ka? Nahi hai. And last we have Har Gopal Datta. नहीं गोपीनाथ नहीं है. Har Gopal Datta, please come. Okay. Now next category was for the students, school students. We have 4 awards. First prize goes to Divya Hazra, Salwan Girls Secondary Senior School, New Delhi. Khadija Zoya Kumar. She is there, no? Sanya Dhingra. Hepsiba. Huh? So this was all, sir. Thank you.
Thank you, sir. Thank you, sir. I would now request the chairman for his address.
बेटा देखना ठीक है, तुम देखना ठीक है बेटा, ये it's okay. Very good evening to you all, ladies and gentlemen, the dignitaries on the dais, the director of the Institute, Professor A.K. Singh, Dr. Vinod Prabhu, Rao Sahab, I'm so delighted to see you, sir, you are here, Rajbir, and of course, DK, and Vinod again, great distinguished colleagues, friends, dear students. I must congratulate for organizing this very successful historical event. All the presentations were really outstanding. I would urge the Honorable Director to kindly request all the presenters to let their full papers be nicely prepared and with due editing, kindly bring out the full proceedings of this, which will be again a historical landmark that you will be able to create, which will be available for all the colleagues for all times to consult and look at. Gopal, you did a very good job in summarizing. Thank you so much. And first of all, as a matter of fact, today again, I visited the Division of Genetics. And when I saw the statue of Mendel, I felt so honored and I felt so proud of the institution that all over the world this is being celebrated. And the way IRI has done it, we have brought a great honor to this institution, to this country, and to the world of science. As a matter of fact, that picture of the saint who happened to emerge as an outstanding scientist will always be inspiring all the students and the faculty who will every time pass through that place. And that inspirational statue will be something which will maintain the legacy of the outstanding contribution that this institution for the humanity as a whole has made. And I think this is a great achievement. The originator of science of genetics, of course, we saw his statue and I talked about him. But equally important, friends, when you look back little, just few 100 yards from here towards the National Agriculture Science Complex, you have the picture, you have a statue of Borlaug. Now here is the statue in front of Genetics Division of Gregor Mendel, and there is the statue of Dr. Borlaug. Can you think of a better combination of creating these historical statues? One is the creator of the science, one is the one discoverer, and the other is the deliverer, discoverer and deliverer of science. Both statues are here to serve the humanity. I think this is a great thing, and please connect yourself always with these statues, what they stand for and what it is standing there and what we are supposed to really look into that. The entire concept then gets harmonized in this process. The recent global thrust on the 17 Sustainable Development Goals, that is called Agenda 2030, the United Nations Agri-Food Summit, which was held last year, and the whole world is watching towards the outcome of the different world negotiations which are going on today under the very difficult conditions of the world and the globe and the planet Earth that we are seeing, the human conflict at some places, the COVID suffering at other places, and still the continuation of the underprivileged and high proportion of hungry and poor people in the world. Therefore, with the ever shrinking resources on one side, with the ever increasing pressure on the limited resources, the more from less for more, MLM, becomes so relevant today. And to achieve that, you can't do it unless you get the help of the best of the science. And the mother of the science for doing this is nothing but genetics. We know that the classical genetics, the kind of work which has been done in most of the institutions that we see here, either cytogenetics or plant breeding, all of these embrace the outstanding achievements and some of the examples which were presented by the various population genetics works and molecular breeding and so on and so forth, genomics, everything, all they taken together. I would only say that all that is relevant today in terms of what we have been reading in terms of history whether the origin of the isoalleles, the allelism, pseudoalleles, multiple alleles, polymorphisms, SNP, polygenic expressions, the phenotyping, genotyping, you name it, genetic gain, selections, and so on. All these, the QTL-based genome-led selections of new varieties, germplasm characterization, even the farmers' varieties can be characterized and so on and so forth with that process. These all speak volumes for the best of the science to lead the world today. And that you are doing in the name of the saint who really discovered this and taking inspiration from that is again extremely important. I thought I will mention to you because there are some, I saw in the award winning posters also, some of the ladies and girls doing so well. I can't miss out in mentioning. I remember Barbara McClintock and the work that she did on maize and the presentations which were made about many of the crops, including maize. The ACDS systems, as you all know, activated dissociation and heterochromatization of certain of the chromosomes to a very large extent. That is also silencing of genes and so on and so forth. The transposon concept, all that which Barbara brought out. At that time, she was not recognized for Nobel Peace Prize. But then, following her good work, Jacob and Manod, when they brought out the regulator operator gene systems, operon concept, transposon concepts, they were awarded the Nobel Peace Prize. And after their Peace Prize was announced, they cited Barbara's work. And I remember I wrote to Barbara because I worked with Barbara in her lab together. And she said, after 10 years of that, she was given the award of Nobel Prize. In other words, friends, a good work which had led to a historical development never is too late to be recognized, will be recognized. And this is where I thought I want to exalt all the scientists and students who are present here to please remember that the work that you are doing today or tomorrow, if that quality will bring you to the world which must believe in one health, one world, and all the work will be requiring today the interdisciplinary cooperation where there is no scientific boundary. And this is where even when you look at the concept of genome interaction with the environment, the nature and nurture both work together, come together. We talked about various other things. I will not go in all those details. I will leave it. I will give Gopal to you. You can take advantage of this to really include it in the summary. I will only try to mention one thing which Ravi K. Singh this morning was able to highlight. Again, it brought out very clearly, very strongly, the epigenetic control of trait expression, such as the control which is getting increasingly much more important. You remember he showed a child with the brain development, and he said right from the conception up to the delivery of the child and the early care of the child, and the way the child recognizes the left and right touch. Please remember, this is a very humane approach which we talked today. And I'm so impressed that genetics is the one which will give you the solutions. How in the early stages of the development, the brain development cannot be allowed to suffer, and there cannot be epigenetic effect which will retard the height or the wastage or the lifespan of a child. Wasting a child, stunting a child is inhuman. And therefore, I think in India, the proportion is so high that, my dear friends, I appeal to you that take note of it and try to see that there is no epigenetic fixation of dwarfism in children brain development. I thought this will be important. Also, a new point which was brought out, which needs to be highlighted again, the synthetic genomics and the dark genomes. And the as you call it, junkomics. And this thing is very little understood. And I think there is a need for working in these areas. And without these people go too far, I think we should be able to come into the reality of existence of these dark genomes. Therefore, the sequencing in the three-dimensional approach of looking at these big molecules and so on and so forth, the big genome Earth project, which is already operating, all these become so very relevant for us to be followed up today. I will only say that please reintroduce yourself to genetics teaching, learning, and practicing in such a way that you are able to capture the various points which were raised today, including the points, of course, the PPP-FRA, a unique provision to strengthen the humanity and to the pride, to save the pride of the farmer as well as his real prosperity. Finally, educational reforms, institutional reforms, investment reforms, policy reforms, because no science will operate in a vacuum. It has to have a policy framework under which the science of discovery and the science of delivery will work together. And if this is the case, I always say that there will be this social responsibility of science and the corporate social responsibility. It was being discussed in the seed sharing and the farmers' rights, how the private sector is doing or not doing enough and what could be done. And I think if these two things are kept in mind, the science social responsibility and the corporate social responsibility to serve the humanity at large, probably many of those questions will be solved. And again, as Mahatma Gandhi said, that any commercial interventions which the private sector or any person or any system brings into, if this is coupled with dishonesty of any kind, that is a big sin, and we should not be able to tolerate that. The honesty is the other name of science, and therefore science with honesty and trust has to be pursued, and nothing is much better than genetics to undertake that responsibility. So with these words, friends, I wish you all the best, and I wish you the best of science to serve the best of humanity. Thank you very much.
Thank you, sir. I would now request Dr. Vinod, Organizing Secretary, to present formal vote of thanks.
Respected Professor R.B. Singh ji, Dr. Prabhu, Dr. A.K. Singh, Dr. IRI, Dr. S.R. Rao, Dr. D.K. Yadav, Dr. Rajveer, Dr. S.R. Bhatt, Dr. S.C. Gulati, all the members of Genetics Club, delegates, students, and friends. So after 3 days of this celebration, we have reached to the final stage, concluding stage. And I think it is my privilege and honor to propose the vote of thanks. A large number of people have involved in organizing this event. So I think it is time to say thanks to all of them. As a part of this celebration, we plan to organize this 3-day commemorative symposium, and it was named Tending Mendel's Garden for a Perpetual and Bountiful Harvest. And as I said earlier also, this title was suggested by Dr. S.R. Bhatt, and it's a beautiful title, summarizes everything. Thank you, sir. And first and foremost, I would like to extend my heartfelt thanks to Professor R.B. Singh ji for readily agreeing to
chair the sessions, both inaugural session as well as this valedictory session. Sir, you are always available whenever we need you, and we thank you very much, sir. I also extend my wholehearted thanks to Dr. Trilochan Mahapatra, DG ICR, and Secretary Dear for delivering the inaugural address in spite of having health problems on a very pertinent and relevant topic, genetics and sustainable food security in India. I convey my sincere thanks to Dr. R.P. Sharma, Chairman, 15th Genetics Congress Trust, and Dr. G.P. Singh, President, Indian Society of Genetics and Plant Breeding, for their wholehearted support and co-hosting this event with Genetics Club. This event could have, this entire event could be organized successfully only because of the support and guidance of our director, Dr. Ashok Kumar Singh, who is also the chairman of the organizing committee. From very beginning, he was looking after each and every aspect, including technical program, arrangement, everything. So without his support, I think we had only 3 or 4 months of time for organizing this event. And without his support, it would not have been possible. So we are just grateful, thankful to him for providing all the support. And the success of this symposium depends to a great extent on the speakers. I am happy that all 3 days we had very eminent speakers who delivered very interesting and enlightening lectures. I extend my sincere thanks to Dr. R.K. Singh, former principal scientist at AJ Philippines, for his very interesting lecture in the form of Hindi poem explaining Mendel's laws. It was very interesting, sir. I thank Dr. R.K. Singh, who could not come here, but he made an online presentation. Our sincere thanks to Dr. R.K. Singh. Dr. B.D. Singh, again, a very ex-emeritus scientist from BHU, he delivered a very insightful lecture on Mendel's work and controversies. It was really many of the things I think I heard for the first time, and it was very enlightening lecture for all of us. We thank Dr. B.D. Singh wholeheartedly. And I also thank Dr. V.P. Sahi, Head Division of Plant Breeding, SHU, ATS, Allahabad, for enlightening us about the pre-Mendelian work on inheritance. That was a very good lecture on pre-Mendelian era. So that was a very interesting. Normally, we don't get that kind of information. And then I thanks to Dr. Ebenzer Haide from Bear Crop Science Limited for very interesting and insightful lecture on advances in maize breeding. I professionally thank Dr. P. Balram, ex-director, Indian Institute of Science, for sparing time for this symposium and delivering a very insightful lecture on Mendelian genetics and Darwinian evolution, bridged by the chemistry of DNA. I also thank Dr. B.M. Prasanna, Director, Global Maize Program, for his lecture on using molecular tools in making breeding programs efficient. I extend my heartfelt thanks to Dr. P.K. Gupta, Honorary Emeritus Professor, Chaudhry Charan Singh University, Meerut, for his informative lecture on Mendel to QTL and Earth Biogenome project. I thank Dr. Pawan Kedhar, Professor JNU, New Delhi, for his lecture, The Dark Genome, Emergence of Functional Genomics 2.0, which generated a lot of interest. I'm also thankful to Dr. C. Viswanathan, Head Division of Plant Physiology, for delivering a lecture on genome editing on a very short notice. I thank and appreciate Dr. S.S. Patil, ex-Dean of Agriculture, US Harvard, for his lecture on application of population genetics in leveraging plant breeding approaches. I'm thankful to Dr. Arun Kumar Singh, National Advisor, Rashtriya Bal Swasthya Karyakram, for a very interesting and thought-provoking lecture highlighting the role of genetics in society. My sincere thanks are due to Dr. N.K. Singh, National Professor, ICR NIPB, for delivering the lecture Accelerating Breeding Through Genomics. Thanks to Dr. Velu Govindan, Senior Scientist, CIMMYT, Mexico, for elaborating the role of rich genetics and breeding in mitigating global hunger. A special session was conducted today during the symposium for school children. The focus of talk were fostering genetic education and career opportunities in biology. I sincerely thank Dr. R.P. Sharma, Dr. S.R. Bhatt, and Dr. Akshay Talukdar from 15th Genetics Congress Trust for organizing this session. This session for school children and for their participation in this program was very important. And I thank Genetics Congress Trust for this initiative. I extend my heartfelt thanks to speakers, Dr. Arun K. Singh, Dr. N.G. Prasad, Staff Scientist, IISER Mohali, and Dr. S.R. Rao, ex-advisor DBT, Government of India, for their lectures during this special session. My thanks to all of you. Another special session was organized during the symposium on plant breeders' right by PPV-FRA under chairmanship of Dr. K.V. Prabhu. I also this generated a lot of questions and a lot of interest among the students. So this was very good actually session. And in fact, if we were having limitation of time, otherwise it could have continued this program. So that kind of interest this particular session generated. And thank you, sir, for taking initiative for this particular session. I also extend my thanks to chairman and co-chairs and co-chairs of different sessions for efficiently conducting the proceedings. My sincere thanks to Dr. P.K. Gupta, Dr. J.C. Rana, Dr. Akhilesh Tyagi, Dr. P. Balram, Dr. R.M. Singh, Dr. R.P. Sharma, and Dr. K.V. Prabhu and Dr. D.K. Yadav for conducting different sessions. I thank the conveners of different sessions, Dr. Firoz Hussain, Dr. Kiran Gaikwad, Dr. Gyan Prakash Mishra, Dr. Rajveer Yadav, Dr. H.K. Dikshit, Dr. K.K. Vinod, Dr. Akshay Talukdar, Dr. V.S. Hegde, and Dr. Shailesh Tripathi. My sincere thanks are also due to all the rapporteurs for taking notes of the proceedings. Thanks to Dr. Navinder Sami, Dr. Niharika Malik, Ms. Harshita, Dr. Prachi Yadav, Dr. Hari Krishna, Mr. Suman Datta, Dr. Neelu Jain, Dr. Murlidhar Aski, Ms. H.A. Bhargavi, Dr. Jayant Bhatt, Dr. Rajkumar Junjare, Mr. Govind Rai Sharma, Dr. Jyoti Kumari, Dr. Vignesh, Dr. Manoj, Dr. Ranjit Ellior, Dr. Rama Prasad, and Mr. K.P. Abhijit. In the organization of this symposium, tireless efforts were put by members of local organizing committee. I profusely thank all of them. Without their untiring efforts, it would not have been possible to organize this event. Dr. K.K. Vinod, I would like to mention especially, did hard work as a chairman of publication committee and ensured that the book of abstract is ready for time. Dr. Vinod was doing multitasking. In fact, he was sort of a nodal person in all the problems. Everybody was contacting him. So he has taken a lot of responsibility, and my sincere thanks to him. He has done really hard work for the success of this symposium. Dr. Gopal Krishnan, again, he did excellent work. He worked meticulously and developed the technical program. So now we are appreciating different sessions, but you see that when we are planning this symposium, these speakers were to be decided, technical program was to be decided. At that time, a lot of efforts were put by Dr. Gopal Krishnan, of course, in consultation with our director, Dr. A.K. Singh, and we identified very good speakers. That is the reason that we are having this very good symposium. Thank you, Dr. Gopal. And then Dr. Gyan Prakash Mishra and Dr. S.P. Singh both worked very hard for the registration of the candidates, for developing kits. In fact, both visited many places to get suitable bags and suitable kits and many things. So they were working day and night for that registration goes smoothly. And my thanks to both of them and their team members, because a number of people are involved. I'm not naming team members, but all those people who were involved in those teams are deserve the thanks. Then Dr. Firoz Hussain and Dr. Vignesh and their team worked hard to give publicity to the event. The kind of publicity which we are getting is because of this team, which was headed by Dr. Firoz, and they did excellent job. And in fact, a lot of letters were being drafted by Dr. Vinod, Dr. Firoz, and being exchanged, communicated to different people. So they deserve kudos. And then I thank Dr. Akshay Talukdar. In fact, he was a link between Dentist Club and Dentist Congress Trust. So he's our treasurer and he's trustee there also. So he's helping all our accounts. So thank you, Dr. Talukdar, for your help and assistance. In fact, for opening the account, he made so many rounds to a bank that you cannot imagine. Because he tried very hard to get the bank account number so that we can start getting funds. So thank you, Dr. Talukdar. And Dr. V.S. Hegde, again, he did a wonderful work. For last 3 years, he is here only from morning to evening, taking care of the stage and all the hall management things. So Dr. Hegde did wonderful work. And with Dr. Hegde, Dr. K.K. Mendel, he did a really hard work. And Dr. Mendel was looking after this management of this tent, pandal, and those things that water, if rain is there should not be any problem. So he did a lot of work. So Dr. Mendel really helped us in many ways. Then Dr. H.K. Dikshit and his team took care of transport needs of all the invited guests and delegates. And I'm sure nobody had any problem as far as transportation is concerned. Vehicles were available to almost everyone. So thanks Dr. Dixit. Then Dr. Bhardwaj and his team managed accommodation of all the guests, ensuring their comfortable stay. So Dr. Shailesh Tripathi, along with Mr. Amrendra Kumar and the team, they took care of online management of the event as well as poster sessions. Here we are also thankful to Dr. R.R. Burman, Principal Scientist, Extension Division, for his assistance. Dr. Pralay Bhaumik took care of food during the symposium to ensure that food is good and delicious, and I am sure all of you must have enjoyed the food. And you say thanks to Dr. Pralay and his team. My sincere thanks to Dr. D.K. Yadav, ADG Seeds, who was involved from very beginning in organizing this event. In fact, he was present in the very first meeting also, and in any subsequent meetings, we invited him and he was present. So he really was a helpful person for us. And he was also chairman of the Resource Generation Committee, Resource Mobilization Committee, and we are grateful to you because of your efforts only, we are able to organize this event. Thank you, sir. And thanks to Dr. Harita and her team, which included Chandana, Maniyari, Sunaina, and Harshita, for comparing the program during all these 3 days. So they are here only. Thanks to all of you. And I would also like to convey my thanks to various units of IRI. We are thankful to OLU, especially Dr. Markandi Singh, Dr. Manoj Khanna, for this decoration. Whatever floral decorations you are seeing here, it's taken care by our unit OLU and Dr. Markandi Singh. He was actually present almost on daily basis to see that all the flowers, bouquets, and everything is there. And I'm also thankful to the estate and protocol section of IRI, transport section, and all others who helped in organizing this event. I'm thankful to Dr. Anirban Roy and the team for the cultural program. They organized the 2 beautiful cultural evenings yesterday and day before yesterday. Those were really wonderful programs, and my sincere thanks to Dr. Anirban Roy and his team. And of course, Dr. Robin Gugoi also helped us in organizing these events. And my sincere acknowledgement to Dr. Rajveer, my colleague, for as head of division. He facilitated many things. We need permission of him many times. So, but he gave full freedom, and that's why everyone was working freely. And so, thanks to Dr. Rajveer. We are grateful to finally our sponsors because for organizing event you need funds, and we are really grateful to our sponsors, without whom it would have been challenging to organize such kind of comprehensive program. We appreciate the support of our gold sponsors, Indian Council of Agriculture Research and the PPB FRA. And here I just personally say my thanks to Dr. K.V. Prabhu, Chairman of PPB FRA. Within few days we got funds once we requested. So thank you, sir, for sponsoring the event. The contribution of silver sponsors, IFCO, Seedworks, Nuzi Voodoo Seeds, Rasi Seeds, Biocrop Science is gratefully acknowledged. They all supported us, and we acknowledge their assistance. We would also like to thank our double bronze and bronze sponsors, GEF UN Environment Program, Biodiversity International and CET Alliance, Sakata, Elora, Carter, JK Seeds, and Savannah. So I convey from my heart my sincere thanks to all our sponsors for supporting this program. Besides this symposium, as you know, is part of a celebration where we are celebrating 200th birth anniversary of Gregor Johann Mendel. And symposium was one event. Other event, parallel event, which was going on, was installing a bust of Gregor Johann Mendel in Genetics Division. And this event is exclusively a program of Genetics Club. Funds were raised by voluntary contributions from members of Genetics Club, plus some of our friends from outside our system. They also contributed. So it was because of those contributions that we were able to construct this. We got this kind of statue, this bronze statue, and this statue will be there forever. Yes, I will be there. May not be there for after some time. We will not be there, but this statue will be, Mendel's statue will be there. So that's really great, and this is a very good tribute to Mendel, that on his 200th birthday we have installed, we have unveiled a statue. The bust was unveiled yesterday by Honorable Director General Dr. Thilochan Mahapatra in the morning in presence of esteemed guests So this is another program that was going parallelly, and many of you delegates were present during unveiling of the bus. So I thank you all of you for contributing also. Right, sir? So for this, सुना नहीं, पहले बोल दिया वो पहले हो चुका है पूरा पैरा है. And this just for information that this bust was sculpted by renowned sculptor Professor Biman Bihari Das, and we honored him yesterday also. And in your kit you will get a miniature of that bust, so you must be familiar with that. So now, at least in all your homes or in offices, there will be Mendel. So that's our gift from on his birth anniversary. And I would also like to thank most important citizen of okay. And I think, sir, his discovery of law of principles was the most important discovery in biological sciences, because that gave the birth of a new science. Yeah. So, and it's pertinent that we celebrate this occasion. We should not have missed this occasion. And finally, I would also like to thank all the distinguished dignitaries and former faculty members of genetics for gracing this occasion with their presence. Thanks to all the delegates, faculty members, students for participating in this program in large number. Once again, I thank each and everyone for making this event a great success. And if I have missed any name, because a large number of people were involved, it was not possible for me to take all the names. So if I have missed any name, please apologize. Okay, if there is any deficiency, I apologize for that, and I wish you all the best. Thank you.
Thank you, sir. Thank you all for gracing the occasion, and we wish you a very happy and safe journey back home. Thank you.
तो नाम जोड़ भी जाना, नाम जोड़ भी जाना। मैं देख रहा था हम चलो।
तुम लोग बेटा बहुत अच्छा काम शो का है। ऑल द बेस्ट, गॉड ब्लेस।
ग्रीन पहनना तो